Chairman of Research Organization for Life Sciences, BRIN, Honorable Dr. Rati Asmananingrum, Head of Research Center for Biotechnology, BRIN, Professor Dr. Durk J.O., Professor Zihua Jiang, Mr. Zahir Ali, PhD, distinguished speakers and moderators, and all distinguished guests and participants. Good morning. Welcome to the second day of the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering, or ISIBIO 2021, Global Platform on Biodiversity and Biotechnology. Before we begin today's events, let me read the conference guide.
Recording in progress.
For presenter and participant are expected to join at least 30 minutes before the conference started, and all participants are expected to join all sessions in this conference. And please use the virtual background provided by the committee and rename your Zoom username according to the following formats for presenters: code of abstract or paper _name _institution. And for non-presenters, you could use the format P_name _institution. And please to make sure, mute the microphone, especially during the presentation of keynote speakers. And in the parallel session, presenter should activate the camera. And questions from all participants are delivered during the raise hand, Q&A, or chat feature in Zoom, and use the format name, institution, to name of the speaker, and questions. And every session will be led by a moderator. All participants should fill the attendees list and abide all the rules listed on the screen. Ladies and gentlemen, before we begin the plenary session, there would be Principle and Mechanism of Biotechnology Product Session 1. Now, we will hear presentation from Mrs. Dana Chow from Science Work, with the topic Accelerating Breakthroughs in Molecular Breeding with Innovative Genomic Technologies. So from Mrs. Dana Chow, the next 30 minutes is yours. Please proceed. Mrs. Dana Chow, are you here yet?
Yes. First, we will send a video of our distributor in Indonesia.
Introducing Biorad's CFX Opus real-time PCR systems. Modern connectivity. With more flexibility than ever, CFX Opus can operate in a completely wireless environment. In addition to Ethernet connectivity and direct data transfer via USB for standalone operation, connect with Wi-Fi for wireless access and data retrieval. Program data and notifications to be sent directly to your email. The new network drive allows you to save and share data locally with multiple users. CFX Opus systems can also link to Biorad's BR.io cloud platform. Remote setup, instrument monitoring, and data management and analysis capabilities easily integrate with CFX Opus systems, minimizing time needed at the instrument. For more advanced data analysis, Biorad's powerful CFX Maestro software works seamlessly with CFX Opus systems. A simple visual interface lets you quickly create biological groups, technical replicates, and set up standard curves. Generate publication-worthy graphics and figures in real time. Biorad re-envisioned their CFX Touch Systems block to produce its best thermal uniformity and accuracy while retaining compatibility with consumables. Uniform well plate temperatures ensure consistent data from well to well. At each scan position, the robust optic shuttle is centered above each well. The optical system automatically collects all well data so that information can be entered or edited when convenient. CFX Opus real-time PCR systems. Powerful, modern, uniform.
The proven unmatched sensitivity you've come to expect from Biorad's family of digital PCR products has a new addition, the QX1. Taking its place in a familiar workflow, The QX1 performs droplet generation, thermal cycling and droplet reading in a single instrument. The search for fully integrated and automated digital PCR is finally over. With 4-color analysis and a new multiplexing supermix, the QX1 is one of the most advanced digital PCR systems on the market. Operating with existing supermixes, assays and kits, the QX1 is designed for you. And just like our other systems, you can be confident in its precision and accuracy. Load up to 5 plates. Oil bottles are loaded and consumption is monitored by the system. Experimental setup and data analysis can all be done using the onboard touchscreen. A simple 5-minute setup to configure the plate is all it takes, and the QX1 takes it from there. Once a run has started, the plate handler moves a single plate from the inbox into the droplet generator section. There, the plates are prepared for droplet generation. The droplets are created through the combination of sample and oil to create an emulsion of approximately 20,000 nanoliter sized droplets. The plate then undergoes thermal cycling, in which 20,000 individual PCR reactions occur in each sample well to amplify the DNA. The plate is then moved to the droplet reader module. Here, the foil seal is pierced by the droplet reader, and droplets are taken up and read by the QX1 with improved optics, fluidics, and motion control. The QX1 features complete onboard software support. Data for 4-color detection and analysis can be viewed for each well on the newly designed 3-dimensional amplitude chart. 2D dimensional amplitudes can now be visualized in a 96-well plate format. And concentration calls can now be made automatically by the QX1 software.
The QX1 makes the workflow so simple. It's the future of automation.
Extremely easy operation and data analysis. Data looks great.
Multiplexing using 4 colors on the QX1 enabled us to gather more data than we could before.
Brought to you by Biorad, the world's leader in digital PCR. From preparing reactions to reading droplets, the QX1 is the all-in-one integrated system that will change how you do digital PCR.
The NGC chromatography platform is truly customizable and grows with your purification needs. Collect precious samples into bottles and tubes and easily access them during a run on the high-capacity NGC fraction collector. Improve reproducibility and efficiency through automation. Choose a method template in ChromeLab software. Automate optimization or assess multiple conditions in one method with multivariable scouting. Set up a run, push a button, and walk away for routine single or multi-column purifications. The pharma-ready NGC system has tools that enable compliance. The NGC chromatography system from Biorad, the one platform that takes you anywhere.
Good morning, ladies and gentlemen. Please allow me to introduce Ms. Dana Chow from our principal LGC Biosearch Technologies, who will present about accelerating breakthroughs in molecular breeding with innovative genomic technologies. Ms. Dana Chow, time is yours. Dana, sorry, we cannot hear your voice. Hello, Dana. No, we cannot hear your voice.
Can you hear my voice now? Sorry about that.
Yes.
Okay, thank you.
Please
All right.
continue.
Yeah, sure. Okay, Tika, can you see my screen? Okay, the presentation slide. Yep, all right. Okay, very good morning to all of you. Thank you so much for inviting me as a speaker for this event. My name is Dana Chow, and I'm the Field Application Manager based here in Malaysia. And I'm with LGC Biosearch, hoping to present my presentation to you. My topic is accelerating breakthrough in molecular breeding with innovative genomic technologies. Okay. Right. So as an organization, we strive to deliver science for a safer world, which is our group mission statement. In agrigenomics, we deliver it through our enabling our customers to overcome the macro challenges facing the world, such as increased population to feed, reduction in available farming land. In order to rise to this challenge, it is critical to apply genomics to uncover novel traits and then incorporate these traits into the desired germplasm. The more accurate selection is made, the faster genetic improvement will be delivered. But this requires data generation, which also require resources. So the critical question is how to generate the right data needed to make the most accurate selection with the least amount of resources. And answering this question will maximize effectiveness and accelerate agricultural science, enabling you and me to meet the challenges we face. Through the collaboration, our technologies help improve breeding cycle, allowing customers to take their discovery to make the market faster. Our integrated genomics tool enable you to achieve more with your breeding programs and accelerate your mission critical science. So LGC origin can be traced back to 1842, when the Laboratory of Board of Excise was founded in the City of London to regulate the adulteration of tobacco, which was prohibited under the Pure Tobacco Act. But over the years, through new product and development, partnerships and acquisition, our portfolio has now expanded to deliver also critical genomics tools such as extraction, purification, PCR, qPCR, and GS technologies applied to plant and animal breeding. Okay, so coming to the simplistic view of a breeding workflow. Okay, different phases and different analytical techniques can be used and are routinely used, as you can see here on this slide. Typically, starting on the left with array, microarray, whole genome sequencing that has a higher marker and low sample number, and moving through the targeted NGS to PCR that has high sample and low marker. So each technology here has its own trade-off, okay, and also such as its own trade-off, such as cost per sample, okay, data content, marker number, and time to result. These are critical components most of the researcher or developer, breeding developer routinely look at, okay? So example here, whole genome sequencing could get all the information you require, for example, back crossing, okay, right here, all right? So, but it is more expensive, a lot more expensive actually, compared to PCR, and slow to get to the data turnaround. Okay. Right, so many times we at Biosearch Technology here, we try to push the operating window wider to allow a technique that do more but still remain cost effective, accurate and fast. As we can see here, we have our TGBS, targeted GBS or multiplex PCR solution that is pushing the either way, okay? A lot of the ways that we do here is to partnership with our customer like you to develop and test this new technology. All right. In comparison of the technologies for the same application, one technology will win over another one, at least for one of this dimension: cost, speed, flexibility, and quality. Classically, when developing a new product, you will look at the 3 key factors, which is this 4 dimension. Normally, there is a trade-off. You can only choose not all, but some of them. You can be cost effective, fast speed, and quality, but you don't have flexibility. So genotyping lab requires high quality data to make those breeding decisions quickly and ideally at low cost. And at many times, this proved to be very challenging. It may require compromises and add in the flexibility requirement, such as marker panel change, and this can further complicate the whole workflow. Okay, so when it comes to PCR example here, PCR in agrigenomics, so usually the application that researcher would use or developer would use is endpoint PCR genotyping, and also qPCR. So for endpoint PCR genotyping, application that is usually used here is marker-assisted selection and marker-assisted backcrossing, and it would require the technology to be fast, flexible, and economical. All right, so for qPCR, something like adventitious, present, GMO, trait integration, copy number variation, and this is detecting if traits of event in interest present or quantifying level of presence. So some testing is actually required by regulation, other necessary as part of the breeding program. And for this, it requires to be sensitive and specific with higher quality at affordable price. So when it comes to endpoint genotyping technology, we at LGC Biosearch Technology here, we offer CAPS genotyping. In chemistry, as listed here, genotyping services. Okay, for us here, we have 3 components. We have CAPS assay mix. Okay, so we have a forward and a reverse primer. So the forward is the allele specific that has a universal tail end. And our proprietary technology with this universal tail end is combined with the master mix that has the frag quencher, frag cassette in quench here. Okay, so then we also have a DNA template right down here at C. Okay, so This template here is provided by you as our customer. So combining the assay mix and the proprietary master mix CAPS assay, so with the DNA template. So it goes through 3 rounds of PCR, thermal cycling. So total of it is actually 26 cycle, okay? And we generate a signal generation that can be read through endpoint, okay? So using the CAPS genotyping chemistry here, okay, so one of the case study that I hope to share with you here, okay, so rice blast is a serious plant disease worldwide and it causes losses up to 100%. It can be treated with chemical. Out of 100 race-specific are gene known and 20 have been cloned. Okay, another disease is the bacterial leaf blight, which can reduce yield by 5 to 85%. This accounts for 40% loss in the worldwide annually. These diseases is not able to be treated by chemical. Out of the 40 race-specific R gene known, 6 gene has been cloned. Okay, so these clone-resistant varieties are therefore a major goal for rice breeder to counter blast and BLB using single or multiple pyramided genes. Okay, so marker-assisted backcrossing has been used for the past 20 years and varieties incorporating resistant gene has been released in all these countries. So this is done on our CAPS genotyping chemistry here. Another example here that I hope to share with you is maize streak virus. Okay, a single loci that is found to be tightly linked to this particular disease resistant, and it only needed 3 capsid marker to identify the introgression into elite variety. These samples were screened with within a 1536 plate format using our SNP line high throughput genotyping platform. Okay, we will show you the photo later. All right, and over 50 plant sampling kits, our BioArc plant sampling kits will also send out to gather 5,000 sample material, and we extracted all this sample with our Octopure DNA extraction high throughput platform and genotype them 5,000 sample all within 4 weeks. That's the capability of high throughput. So the implication of this is the fast turnaround means that there is we only need to sample the resistant variety, taking it forward. So time and money saved. All right, so our customer can approach us running CAPS genotyping chemistry, whether in their own lab, okay, or send it to our largest genotyping service in the world in UK here at Hoddesdon. Okay, this is our genotyping service lab. So we routinely every day doing 1 million genotyping data point. Okay. So on top of that, if you run PCR, CAPS chemistry in your own lab over the years and you wish to take it to the next level into high throughput. So we also work routinely with our customer providing the instruments, DNA extraction. So, and using with our chemistry. CAPS is not the only chemistry that we provide for genotyping. We have the BHQ and BHQ plus probe here and also the master mix. Of course, we have here the PCR workflow technologies products that is for downstream and upstream as well. So we will go into the BioArc leaf collection kit in a moment, but we have other type of extraction chemistry. So we are all fully integrated here. So we have the quick extract, SpeedX, the Octopure, and also we have the PCR reagents and also NGS reagent as well. But of course, we are talking about PCR workflow here. So for high throughput instruments, we have the IntelliCube, Nexar, SNP line, array tape. All right. So coming to down here, PCR still dominates lower marker number workflow here. okay, when it comes to routine genotyping. So main focus here is speed and cost, of course, some flexibility here as well. And for NGS, it is unable to achieve this same price point per sample. You know, NGS is talking about having a certain coverage on especially like large genome size, so those can be quite costly. All right, but however, the market here, or in general sense, people are actually looking into higher density genotyping. Okay, so genomic selection is the thing that people are gradually moving on. Okay, so what technology does people use for genomic selection here? Okay, so genomic selection is a critical application to shorten the time to market. Okay, what does it mean by that? So if you know about breeding technologies here, so the people would look at genetic gains, okay, for market-assisted selection as an example. So for market-assisted selection, the genetic gain from it is actually like 0.4 to 0.6% per year. And to be able to address the food need in the future and the consumption need of the world in the future, statistics shows that it needs to be actually 4 to 6. So from 0.4 to 0.6 to 4 and 6 per year is 10 times. So how can we accelerate and meet this demand here? Okay, so we need to market and develop new breeds in a faster way. So genetic selection is actually showing potential and chance to accelerate this over time. Okay, so genomic selection accelerate breeding and provide potential solution as part of approach to solving global agricultural sustainability challenges. But genomic selection requires fast, flexible, cost-effective screening of thousands of markers. So this is the technology challenge. Okay, so when it comes to GS, for us here, we actually take the targeted genotyping by sequencing workflow, PGBS, which is an NGS technology. Okay, next generation sequence technology. So 6NIP here is TGBS, optimized for screening thousands of markers cost effectively in any sample number. Okay, so it is not GBS, not genotyping by sequencing. It is targeted. You need to know your reference genome and to design this panel. Okay, so it is optimized for several hundred to 10,000 markers, but scale up to 100,000 per sample. So it's cost effective because we designed the panel and in NGS platform environment. So it is flexible. So you design for this panel this time, and next round you can remove and add new panel, new marker into the panel, and it is lower setup cost. There is no setup cost for 6NIP here. So we designed the panel for you for free. So we just order it as a kit. Okay, so it allows de novo variant detection in the target SNP region. So there is like a 70 base pair read line here. So then you can detect additional SNP at the upstream and the downstream here. Okay, so comparing market-assisted selection to GS here, important point here is that like, although we're still looking at the same sample number, 100 to 10,000, so then we have here a low SNP number for 1 to 10,000 SNP, okay? And then we have here up to 3 to 50,000 here, okay? So GS is much quicker in that way. Okay, so we have it in the service process, okay, and then the data result. So we also have other product here, genotyping by sequencing by key gene, okay, and on top of that, we have our BioArc sample collection kit that helps you to collect sample quicker in the market, in the field there, all right, and with a BioArc kit, then we can have a better genotyping quality because the sample are properly dry. Okay, so of course we have our DNA extraction and purification platform. Okay, so as you can see for LGC Biosearch technology, we have focused on all this area of the breeding technologies. So we have here from WGS, whole genome sequencing, discovery, upstream analysis, down to a targeted GBS technology, and then move on to PCR routine QC technologies. We have our solutions for you. Okay, so there's a couple of ways of how technology development can drive cost, flexibility, quality, and time factors required to accelerate breeding technology to solve the global challenges facing the industry. So thanks to our customer in collaboration with their support and inputs, LGC Science for a Safer World. Thank you so much.
Thank you, Ms. Dana, for your presentations. And for the ladies and gentlemen, if you have any queries about our technologies, please don't hesitate to contact us, PT Sciencework. Thank you.
Thank you, Mrs. Maria and Mrs. Dana Chow for the presentation. For the next, we will hear a presentation from Mr. Revata Utama from Nusantics with the topic Indonesia as a Global Biodiversity and Biotechnology. Mr. Revata Utama, the next 30 minutes is yours. You may proceed.
All right. Thanks so much, Ms. Wiwit. Good morning, ladies and gentlemen. Let me just share a screen and introduce you guys to Nusantics. All right. So today's topic will be about introducing Nusantics and also Indonesia as the global biodiversity and biotechnology hub. Is it a far-fetched or a reality in the future? I think first of all, we'll just start with all these topics for this morning. And let me start with introducing who we are. So Nusantics is a holobiome startup. What does holobiome mean? I think we made a lot of mistakes looking at human as a single species, and now with the advance of genetic sequencing, we realize that we are not only a human, but also the microbiomes and the microbes that live on us, be it on skin, be it in the gut, or be it in your lung, respiratory organs. So genetics is part of the things that you can't really change because it's given by your predecessors, your parents, your grandparents, and it relates to your ancestry and also, the gender that you, the sex that you're given with, be it a male or female. While microbiome is something that is dynamics, where it's changed when and in where you were born, how you were born, be it through cesarean or even normal, birth, and then what you eat and drink also affects on your gut microbiome, the environment that you're living in, the air you breathe in, the kind of exercise that you do, and also the stress level. And when we talk about microbiome, we also need to know that they consist of the bacteria, the fungal, the archaea, and also the virus, which is very closely related to the pandemic that we are having right now. So pandemic accelerates biotechnology. We believe that the next technology is biology, not only digital, but it's also biology. And Indonesia should not waste the momentum because one of the things that we can do, we were established in 2020. And because of the pandemic, we helped the government to create the first local PCR test kit for COVID-19. And we released second generation in September 2020, where it's multiplex and increases efficiency by 3 times. And also in August 2020, we launched our BioScan service for skin microbiome. And recently, in March 2021, we released the air viral detection to help reopen the economy, where we tried to detect the COVID-19 concentration in the air and also give out the risk index for the infection in classrooms and also office. And as recent as June 2021, we released the gargle PCR method to increase the testing capability across Indonesia, not just in Java, but also in the rest of Indonesia. And our product has been used about 5 million test kits by Indonesian government and also the Indonesian market. We also have about 20% of our team members from universities, overseas universities. We consist of highly capable scientific team. These are the universities where we came from, and also we have the Professor Carl Whitworth here as our scientific board advisor. where he was we can call him basically the grandfather of qPCR and fast PCR systems. Let's talk about biodiversity because it's an ISIBIO event and we talk about biotechnology and biodiversity. So why does it matter? Biodiversity is something that we take for granted for a long time. We live in the ecosystem as humans. We always think of ourselves first and foremost, but we don't realize that ecosystem actually give us back all those things by giving us foods, provisioning function, fuels, medicine, and then it also we are actually part of the ecosystem where we help, pollinating, and also we have to give back to the nature by creating the cycle back where we don't just use them and also but give them the needs for the ecosystem to keep going. And the ecosystem itself also has regulating function, and also the most important part is actually cultural function for us, where we use cosmetics, we do rituals, we actually develop all these cultures because of the shapes of the environment that we live in. But we failed to value the biodiversity because as of 2020, we lose about 70% of the vertebrate species, 40% of the insect species, and also 50% of the plants. What we don't realize is that 50% of the world GDP is actually generated from nature, and 75% of pandemic is caused by emerging infectious diseases such as COVID-19 and also flu. And when we talk about a system like biodiversity, we have to really correlate it with second law of thermodynamics, where some things are irreversible. Once the entropy increases and the system actually collapses, there is no more life on Earth. That's why we have people like Elon Musk try to bring us to Mars. But what we don't realize is biodiversity is the key to ensure that everything is at peace, we live at peace, because all this vicious cycle of destroying nature is actually going to increase a lot of zoonotic diseases, extreme weather, and also resources scarcity. With that, it will lead to global conflict. But when we talk about biodiversity, we talk so much about the carbon metrics, where it's just about the cycle of where the carbon is absorbed and where the carbon is being produced. But what we don't realize is actually there's a lot of things that changes from the micro environment itself. So that's why it's time for Indonesia, where we have the world's biggest land and marine biodiversity. So let's take a look at the microbiome changes itself. For example, coral bleaching itself. When we talk about coral bleaching, before we see the visible changes in the color of the coral itself, there's actually a lot of changes in the microbiome diversity on the coral itself. And we realize that microbiome diversity itself reflects the microbiodiversity loss. There's a paper in Nature in 2020 that talks about the microbiome and the many changes before this coral bleaching is actually observed. And what we do here, we are trying to raise biodiversity awareness through whole biome concept, where we realize in human species, we have 30 trillion human cells, but we also have 39 trillion microbial cells, which consists of like 57% of our body. And they actually correlates to a lot of diseases out there, especially non-communicable diseases. Like, for example, in our skin, there's acne, eczema, psoriasis. In the oral microbiome, we have the dental caries, periodontitis. And in the lung microbiome, is linked to various diseases where the imbalance of microbiome will cause a lot of lung diseases like COVID-19, pneumonia, asthma. And gut microbiome, as you know, this is the most well-researched topic, and we have type 2 diabetes and stunting correlated to microbiome dysbiosis in the gut. So our work is centered around the interaction between humans and the microbiome itself. The key is holistic and accurate diagnostics, because we are not just looking at the human itself. We are, as I mentioned, microbiome is also coming from the environment that we live in. So we look at the air, soil, and marine. That's why we also have the air scan for COVID-19. And here are some of the surveys that we have built for the past 2 years. We have BioScan, which is the first skin microbiome check in Indonesia. We look at your bacterial fungal ratio and also the microbiome composition to see the right skincare for you. We also do a lot of research on ecobiome, especially the latest one will be air scan, where we work together with Commandicwood to scan the schools for COVID-19 presence and also give out the risk index. We did it in Cinema 21 and also Masjid Istiqlal to check whether there's a COVID presence and to protect the people who are doing the prayers there. We recently also worked with Pelataran, where we basically research on their air microbiome and also the environmental microbiome in the Pelataran area. So we believe by collecting all those data, we will be able to understand the microbiome composition and its effects on human environment and also the relation to the disease that we observe nowadays. So we would like to believe that these microbiome changes can be an indicator for the health of our planet, not just in humans, but also in the environment. So let's talk about Indonesian bio startup then. As we mentioned, the next technology is biology. So we have so much potentials, but lacking the execution and also the political will to do it. As we can see here, when we do go into the biotechnology, there's a lot of things that we can develop from there, especially from what we listened to just now. There's going to be a lot of improvement in agricultural side. We can build new therapies and cures by just looking into more of these microbiomes. We might be able to develop new food source, new food plant source by utilizing all these microbes and new materials and clean fuels. But the challenges are, there are a lot of challenges. As we can see in the market right now, there are two types of biotech startup. The first one is the pioneer and research oriented, where we build all this, we do go through all this workflow where we validate the tech and then we build a database and we find biomarkers and we develop device for diagnostics, for example. And then from understanding the problem itself, we'll be able to give a precision solution. So there's a lot of challenges that comes with this, especially with building databases where we have to, use, we have to build laboratories to collect samples and then process them. There's also the regulation where, all this lands is actually owned by the government and we need the ethical clearance to do all the research. But then there's also the type 2 where it's market driven. You can just buy things from outside, especially from China, and then you do the diagnostic service, for example, like what we have nowadays with the COVID-19 testing. Where do we stand for? Nusantics do the hard work, where we, for example, in the microbiome service, we do the validation ourselves. We build the sample collection method up to the detection ourselves, and we build the database right now where we can identify the biomarkers and also design the kit in the form of biomscan. We also do the production ourselves and we do the diagnostic service ourselves. In terms of COVID-19 pandemic, we don't need to build the database because we have the GIS-8 to do it. What we do is we identify the biomarkers so that our kit is safe from mutation. We can still detect up to the new mutation, the latest mutation. That is reflected in the BioCov19 up to the BioCov 3.0 that is going to be released soon, as soon as December. And the diagnostic service, we are going to do it as soon as possible because we believe in providing affordable service and also accurate service for COVID-19 testing. But we still need help from the government, especially because Indonesia, I think, as we mentioned, it's a big land and also we have a huge biodiversity. So I don't think we should be looked at as market only, but we also need to be an innovation driver. So to do that, we definitely need to collaborate with the government institutes like BRAIN, universities and diasporas, and also this other downstream companies like the BUMNs. So I think in conclusion, is it a far-fetched or realistic future? I think let's quote some of the statement that were made by Steve Jobs and Elon Musk. Steve Jobs mentioned you can't connect the dots looking forward, so you can only connect them looking backwards. So we have to trust the dots that the dots will somehow connect in the future. And Elon Musk also said, when something is important enough, you do it even if the odds are against you. So I'd like to believe that Nusantics will be able to achieve the universal biome index for us to monitor the health of human and also the planet through biodiversity itself. So I think for a closing statement, these are our websites. We have nusantics.com as our main website where you can take a look at our biome services and also personalized skincare that we have. We also launched the initial Indonesian microbiome initiatives recently, where we are looking to collect data from not just Indonesian people, but also the environment. And recently, we are involved with Commandicwood to basically reopen economy and reopen schools, where we do air scan and gargle PCR for schools. So I think that's it from us. I'll return it back to you. Thank you.
Thank you, Mr. Revata Utama, for the great speech. The following presentation, we will hear from Mr. Andreas Agustian from Elo Karsa with the topic Vaccine Development to Translational Research. So, Mr. Andreas Agustian, the next 30 minutes is yours. You may proceed.
Yeah, hello, good morning. Thank you, Ms. Wiwit, for the opportunity. I will try to share my screen. Okay, is my screen okay?
Yes, Mr. We can see it.
Okay, okay, let me start. Thank you for the ISIBIO committee or the chairman for the opportunity for PT Elo Karsa Utama to have the schedule for the presentation here. I would like to share some information regarding the vaccine development and translational research here. Okay, at the end of the presentation, I would like to invite all the participants to play some mini games at the end of the presentation here. I would like to introduce myself. My name is Andreas Agustian. I'm as the product manager from PT Elo Karsa Utama. And Elo Karsa Utama itself established in 1981. It's already almost 41 years now. Our head office in Jakarta and the regional office in Surabaya, Bandung, Bogor, and Yogyakarta. And our focus in the life science business, animal health, food safety, and also the medicals. Our vision is to become the leading distributor in life science and medical industry to the effective and synergy with the principles and achieving the best solution for our customers and also improving our customer satisfaction by consistently providing the high quality innovative products and a better service experience. And as our motto or logo is the serving you better. This is our distribution network. We are have the head office in Jakarta at the Kabayan Rama Street, and also we have some representative office in Bogor, Bandung, Yogyakarta, Surabaya, and also in Semarang. Please welcome all the customers to contact our representative in the area, and also we have some warehouse in Jakarta, Serpong, Surabaya, and also in Bandung. This is our value client from the industry, biopharmaceutical, also the animal health industry, pharmacy, and also the government institution, university, and also clinical lab. And this is our brand partner. The product we represent right now is the instruments, from the general, imager, and all the essential instruments that you gonna be needs in everyday lab, every day, and also the reagents starting from the chemicals, media, from the bacterial, and also for the cell culture, and also the testing kits, especially for the COVID-19, and all these consumables starting from the plasticware, glassware, and all the special one. For the life science itself, we also providing our solution from genomics, proteomics, cell culture, and also the animal research. Now for the vaccine development itself, typically we start from the pathogen profiling. We can use the COVID-19 as the example here. We do a lot of NGS, and also we do the viral characterization using the viral culture, and also some the Western blot or imaging itself. And then we try to design a vaccine here, starting from the factor, or using what kind of platform we want to use. For instance, the viral factor or the mammalian cells and so on. And also after that, when we get the protein, and then we try to do the expression, then we do the in vitro testing here, using maybe the nucleic acid, real time, or the in situ hybridization, and also doing some ELISA technique or Western blot, and also the insal Western. Regarding the insal Western, I will explain more later. For the cells itself, we can do the normal one is the plaque reduction neutralization test, and also some of the imaging. For the in vivo testing, typically we do serology, like ELISA and so on, and also the animal challenge here to see the pathogenesis, and also the imaging to see whether the biodistribution or the target is targeted the correct organ, or can protect the organ that we want to save. For instance, lung here for the COVID-19. And also we can provide the scaling up technology, starting from the upstream and downstream, and also through the QC process. Okay, our one of our solution for the viral acquisition systems comes from the Agilence technologies. We providing the adenoviral vector here, and also we do have the other plasmid called sure vector here. So in the previous time, maybe some of the researchers doing the molecular cloning, like doing the ligation, but here with the sure vectors, it's like playing your Lego. What is your gene of interest, and what kind of selectable marker do you want to use? Is it ampicillin resistant gene, kanamycin, or chloramphenicol? You can choose what kind of gene do you want to put in your plasmid. So it's just playing your Lego, and it's very simple to make a circularized plasmid in a single reaction, only 20 minutes. All right, I will next for the insal Westerns. So in virology, there is some well-known technique that can be used, like plaque assay or PRNT, and also some of the Western blot or the ICC, immunocytochemistry. Now, typically in cell Western workflow like this, when we do the culture cells, and then we stimulate or inhibit, typically for the vaccine, we put the antibody neutralization here, then the viral can be neutralized by the antibody, and then it can be inhibit for infecting the cells. And then the cells we can fix and permeabilize, do the blocking solution here, detect with the primary antibody, and then we detect with the secondary antibody. Now, the secondary antibody here, we put the label here for the fluorescence, so it can be detected with the imager. Then the imager will excite the fluorophore, and then we can get the result. So the benefit of using this insal Western to study the virus is to have this high sensitive, due to the low sample autofluorescence. Also the high accuracy of the assay, due to the antiviral antibody specificity. Also works with the virus with or without CPE. Some problems in the CPE in some virus and the cells model, that cannot show the CPE. And also we able to do the multiplexing, so we can do the normalization there, and also we can use the 96 well or 380 well and 84 well pass to get more throughput samples, and we can remove the subjectivity from the analysis. Also we provide the software to shorten the times for the analysis, And the other things, we can also do the on-cell Western. So what is the difference in these steps is we exclude the permeabilization steps. So we can do some detection on the cell surface protein, so we can detect on the receptor trafficking, for instance. Yeah, and then we can image. So in vitro application here for the virology, we can do the viral titration, we check on the viral replication, we can quantify the protein expressions, yeah, very easily, and then also the mechanism of actions and efficacy of the vaccines or the antiviral. Or we can go forward with the in vivo characterization with the delivery and the response. This is some examples or the parallel comparisons between the plaque forming units here for the plaque assay and the INSAL Western. And this graph showing the correlation is almost close, yeah, to each other. So instead of we counting the plaque number here, we can measure the fluorescence signals comes from the fluorophore or reflecting the amount of the proteins expressed protein from the viral, and then we can calculate very easily. Okay, this is also some examples for the non-CPE causing virus. Yeah, so we can detect the viral protein. Yeah, comparing with the other fluorescence technique. So the correlation is okay comparing with the conventional method. Now here also the some examples for the quantifying infectivity through viral replications doing the HTNV here comparing with the beta-actin, and then we can overlay here for the normalization. We can see how the viral replicates per days. Yeah. Here also some examples for the assessing the vaccine effectiveness. Yeah, using these examples here, vaccination with blood boost and the boost vaccination, and how we can compare the neutralizing antibody titer here very easily using the INSAL Western technique. So how we can achieve that one? We can use the ODC CLX imaging system here. This is the instrument picture. It can do the near infrared fluorescence. Why only near infrared systems? Because on the visible, we can show here cells. Yeah, cells we culture, and then without any staining dyes, when we expose or we capture the image with the visible fluorescence, it can emit a light. So in the visible fluorescence, there is some autofluorescence from the cells itself. So when we want to detect some of the target proteins, gonna be very hard, yeah, because due to the autofluorescence from the cells itself. Yeah. So this is the other DLX applications from the quantitative Western blot. Yeah, protein gels, in-gel Western. INSAL Western, we just talk about viral titration or the EMSA, gel chip assay. Tissue sections, maybe after we do the in vivo imaging, then we want to do the tissue section to check. We can do the scan on the ODC CLX and also the protein arrays. And the next is how to do the in vitro testing using the cells. Yeah, how to make your cells happy. Okay, I have the illustration here. The cells must live in the house. Yeah, we call it the culture vessel. Yeah, what type of house, how big the house, and also the media. What's the cells gonna eat? Yeah, what is the nutrition they need? The instrument is corresponding to the environments behind it or surrounding. Yeah, what is the CO2 concentration or the oxygen concentration and all the things. And after that, how do we analyze with the imaging here? Okay, so for the culture vessel here, we support for the thermo from NANG. Yeah, we have several type of the culture vessel. Also, if you want to upscale the production, yeah, somehow the for the in vivo testing, you gonna be required a lot of culture vessels, yeah, the bigger one. So we can provide that one also, the media, starting from the media basic and also the serum and also the differentiation medium or the serum free media. Yeah, we also providing the cell line here from our principals. Yeah, for the instruments, we can provide the CO2 incubator here, also the have the accessories for the cell locker, so no need to worry about the contamination for each segment or compartment. Yeah, also we provide the BAC to handle your cells, and then the centrifuge to process and the autoclave to terminate. Then for the imaging, we provide the microscope from the Perkin-Elmer here for the live cell imaging, and all the confocal or the high content imaging we have. I won't talk more about the high content imaging here with regards to the what can be done with this instrument for the for your vaccine research. Yeah. So high content screening itself is the imaging and analysis from image to number to the knowledge. So basically, it's image-based screening, yeah, measuring the biological activity in cells. So in this picture, we want to see, we can see the mitotic cells here, and also almost separate cells and all the mature cells. Yeah. So we want to discover things using the confocal with the high content screening. And how we can do the experimental with the high content screening. This is the workflow. We can sit, yeah, we can try to grow the cells, and then we can do the treatment. And after the grow, we can do the staining, and then after the staining, we can image and then do the analysis. And after the analysis, we can do the data analysis. Why there is two things, yeah, here, the analysis and data analysis. When we do the image analysis itself, we can separate which cells we want to analyze and which fluorescence we want to detect. And for the data analysis, we want to see the graph and the histogram about the effectivity of what we have done in the experiments, like the treatment. We put the compound or the antibodies that inhibit the interaction to the cells. Yeah. And what high content analysis can bring you to do more samples, yeah, because we do it in, typically we do it in the 96 well plate, yeah, in the microplate. So there is more samples and can be statistically relevant, reduce the bias and achieve higher physiological relevance and gain deep insight into our models, yeah, biology models here. And all can be done with the single software. Harmony software can do the analysis and data analysis. For the high content imager with confocal here, we can see the difference, yeah, between the wide field and the confocal. Confocal can bring more details on the fluorescence and a bit dim, but it can be, also give a sharp and better resolution. So what kind of models we can do with the high content imager? We can do the HeLa cells for the cell line. We can do the primer cells, co-culture, or the iPSC, and also the spheroid analysis, the FRET for the protein interaction, and also the zebrafish tissue. And again, we need image that contain information that be quantified. Yeah. And what is the other application? We can do the drug repurposing, development of new therapeutics, development of new vaccines with the high content imager from the viral, bacterial, and parasitic type of studies. And this is all the type of publication, yeah, from the viral disease, bacterial, parasitic, and also cancers. Now have a growing in stem cells, and 3D models also already done with the high content screening here. This is some example from the virology, yeah, using the high content screening here. So it can be determined in this research, they determine the colocalization at the cellular level, yeah, and also how the the receptor can inhibit the viral activity. And also with this instrument in the studies, we can see how host and viral interact each other. And also here for the COVID-19, how the cross-neutralization antibodies developed from the vaccination can inhibit all the circulating variants around the world. Yeah, this is the evaluating neutralization antibody and also calculate the neutralization activity using the Columbus software from Perkin-Elmer. This is the current publication for high content screening using the for the COVID-19. And then for the in vivo research, we can support for the mice itself, yeah, from Biolasco here. We can provide the inbred mice, the outbred animals, and also the hybrid. So please contact us if you have any inquiries. And also we can provide the in vivo imaging here. So what is the benefit of using in vivo imaging? We can explore different aspect of the disease. Yeah, we can monitor the disease development, characterize the mechanisms of actions from the pathogens, and also assess the therapeutic efficacy here. This is some of the examples. So the bioluminescence done here to monitor real time, yeah, for the COVID-19 spread in vivo, and then with the neutralization antibody design here, they can do the proliferation of therapy, and then the engagement here to reduce the viral load in the lungs, and then the monocyte also can be detected here, how the cellular immunity also responsible for the viral clearance. Here also some examples for the thermostable mRNA vaccine COVID-19. Yeah, hopefully it's gonna comes to Indonesia soon, I think. Yeah, so this is the development of the encapsulated mRNA vaccine targeting the RBD, and then it can induce the neutralizing antibody and also the cellular immunity. And then how these mRNA vaccines also give the protections to the animals model, and the thermostable vaccine is also already in the phase 1 clinical studies. Yeah, this is some of these examples. Yeah, the storage days and also the temperature to store, so it shows that it can be protected. So more than 12,000 publication in many applications for the IV system. And also we provide the 2D and 3D imaging. Yeah, so let me play here. Yeah, for the 2D, typically we can all detect the fluorescence or the bioluminescence from one side. Yeah, but the problem is how deep is these signals. But with the 3D tomography, we can detect how deep, because we do the 3D tomography to check where is the locations, and then also we can do localization and correlation to the mouse atlas on which organ this signal is detected. Okay, this is our system from Perkin-Elmer. So IFIS comes from the in vivo imaging system. Yeah, that's a brand from IFIS. So we have variant model here about what is capability of doing 2D, 3D, and also the X-ray. Besides the instruments, we also provide the reagents. So if all the attendees has the in vivo imaging system, it's on your lab, you can contact us and discuss further about the reagents we can do the research and which catalog that suitable for your research. Okay, for upscaling, we can provide from thermoscientific called Staff Factory here. It's very simple workflow with the constant result, because it's just upscaling like the apartment. Yeah, so this is the landed house and this is the apartment. Just increase the numbers of your flash here using the cell factory. No need to optimization, just do the scaling up with the cell factory, very easy. to upscale your culture, and also easy to downscale. And the other things is for the suspension cells, for instance, you can using our bioreactor system from our BioSmart. Yeah, it can be optimized to your cells for the single-use technology. And also for the sample QC, we want to introduce the MFI, stands for the microflow imaging from protein sample. Typically for your research in the QC, at the end your finished product, you need to count the particles, and then because the size of the particle itself can change the immunogenicity. Yeah, so typically when you do the in vivo imaging, the in vivo research, we want to analyze or detect the serology, for instance, is gonna be changed when it go to clinical trial. So better you can start with the MFI to check the particles for your formulations, and also for checking your vaccine final finished product, or in the in the process, yeah, in the downstream process, you also can do the the capillary electrophoresis, yeah, from Maurice, to separate the protein from the heterogeneity, yeah, because some of the protein may be produced and have their own isoform. And also for the size itself, whether it's already degraded, we can do the capillary electrophoresis using the SDS to check the size, yeah, whether it's truncated protein or not. Okay, that's all for my presentations. Maybe is there any open questions? And we have some gifts for the participants who are asking questions, and also we have some gifts for the participants who are willing to fill our link. Yeah, maybe is there any questions? Miss Vivit, maybe we still have time for Elokarsa?
Yes, we still have time. Mr. Andres, if anyone would like to ask questions to Mr. Andres Agustian.
Yes, please, you can put on the chat if you have any questions. If not, then we can play with the game. We have several door prize.
Oh yes, for the questions, Mr. Andreas Agustian.
Okay, is there any questions?
May I ask?
Yes,
oh yeah, Penny, please.
Okay, so I have a questions about the mice experiment that you did. I mean,
yes.
How did you induce COVID-19 to nude mice, if I'm not mistaken? How did you make a mice experiment for your this one for COVID-19?
This one?
I think it's more like to imaging. Yeah. Next, you can explain it this. I mean, for how did you make? Because you do it in mice, right?
Yeah. Okay. So in this research, I think, if I'm mistaken, yeah. So this is the research from Zhang here. Then they using the induced mice with the H2 receptor. So I think it's from the Jackson Labs who do these modifications, genetic modification. So these mice already express the human H2 receptors. Then it can be susceptible for the COVID-19.
So I got it. So you already, so it's not your own research. I mean, you use it for from someone else's research or something. Oh, I see.
And there is some of, I think there is one. Yeah, there is one like company that providing the animal model for COVID-19 research, yeah, to do the challenge because the mice already induced with these human H2 receptors, so it can be susceptible. Then after vaccination, we can do the challenge whether the mice is protected or not, something like that.
Oh, I see. About this mice imaging. So you use 3 different temperatures. So what is the influence between different temperature with the mice metabolite?
Yeah. So in this research, they do the bioluminescence for the what to say, the virus. Then the vaccine itself will be injected, and then will induce the neutralization antibody. Then when they inject with the virus, it will give the signals from the lungs, because the virus will target the mice lungs here. And then how they gonna be to be protected from the vaccination. Yeah, using the thermostable vaccination. So basically, this research more focused on the development on the encapsulated mRNA vaccine. So they want to develop the because we know, mRNA is very fragile, it's unstable, easily to fragmented, and how do we encapsulate the mRNA as the vaccine with the thermostable. So no need to have the low temperature storage, something like that. Just the
judging from this in vivo imaging, the fluorescence in liver is very high. Is it really effective to use this strategy? Because you target a lung, but a majority, the accumulation of the fluorescence is in the liver. Did they mention about this?
I think for this research, it only shows that the protections can confer the protection against the COVID. But for the organ itself, I need to check, Ms. Reni. Thanks for the confirmation.
Thank you very much.
Yeah, anything else from the chats? Okay, maybe Elokarsa, we play some games at the end. I will stop share, and then my colleague, Ms. Stephanie, will conduct with the door prize. Yeah, we have already several people that already fill the form. Yeah, thank you very much for your participation. Then we can spin here, and we have two prize here. Okay, we will looking for 2. Okay, please spin. Who is the winner? We will contact you after this. Okay, Miss Yati. Then the next one. Okay, Elo Rifki Virginia. Okay, thank you very much. We will contact because already submit the email and the phone. Thank you.
Thank you, Mr. Andreas Agustian, for the great speech, and congratulations to the winner. And you can contact Mr. Andrian for further information. Ladies and gentlemen, we have finished principal mechanism of biotechnology product session 1. Now, going to our next agenda, We will hear keynote speech from our keynote speaker, Professor Doug Jo from Asian Federation on Biotechnology, or AFOB, with the topic Enhanced Production of Recombinant Human Interferon Beta through Signal Peptide Modification. Now allow me to invite Professor Doug Jo. Good morning, Professor.
Good morning.
Good morning, Professor. Before we begin, allow me to read a brief CV of Professor Doug Jo, if you allow me, Professor.
Thank you.
Okay, so Professor Doug Jo earned his doctoral and master degree in Chemical Engineering Korea Advanced Institute of Science and Technology, and his bachelor degree in Chemical Engineering, Seoul National University, Korea. He is currently a professor in Sejong University, Department of Integrative Bioscience and Biotechnology. Professor Doug Jo research topic focus on bioreactor development for cell cultures, therapeutic protein production, including antibodies, cytokine, hormones, and many more. Cell culture process development, adult stem cell expansion, cell culture medium, including serum-free medium development, and gene therapy technology using virus and or non-viral delivery system. Now, without further ado, the time is yours, Professor.
Okay, thank you. Let me share the... Can you see?
Yes, we can see it, Professor.
Okay, thank you. Okay, thank you very much. I'd like to appreciate the organizing committee for giving me a great opportunity today to share our experience on the bioprocess and also cell line development. Okay, I'm Doug Jo from Sejong University. I work for Asian Federation of Biotechnology also. So I'd like to invite you, all of you, to Asian Federation of Biotechnology. Actually, membership is free, so you can join and enjoy your network with other people in Asian region. Okay. Today, my talk is enhanced production of recombinant human interferon beta through signal peptide modification. Actually, my background is engineering, and I used to have a strong interest in how to enhance the productivity from cell culture by bioreactor development or bioprocess development, media development, that kinds of thing I have a strong interest. But this time, today, I have a little bit different area. Recently, we have interest in this cell line development area, and we got some results. So I like to share this result with the participants in this symposium. Okay. Okay, what is the interferon beta? It is right now it's a biotherapeutics and mostly produced by animal cells, and it can be used for treatment of multiple sclerosis. And still, it has a huge size of a market in the global. And then actually, since it is a kind of biologic product, so it needs to have a high productivity to be competitive in the global market. So I have interest in productivity also in this case to get a high productivity for production of interferon beta from animal cell. But this time, not the bioreactor system, not the serum-free media, something like that. This time, focus on the cell line development, especially for vector engineering. But I'm not a specialist in the vector engineering, but we started with a small project to approach this area, and I'd like to share our experience with all of you. So as I told you, in order to produce a recombinant protein from animal cells, you need almost 3 steps. First one is cell line development that produce your product. And then after you get the cell line, you need to develop the cell culture process, especially for that cell line. You need some specialized bioreactor system, and you need to decide what kind of a culture mode you're gonna use, like a fed batch, profusion, whatever. And also you need to develop the media to get a high productivity. Maybe it's gonna be serum-free media or chemical-defined media. And then after you finish the cell culture, you need to purify your product. So if you optimize and develop the purification process, you can get a good productivity also. So most of my research area is focused on here, and most of my published paper is focused on this area. But this time today, I like to talk about cell line development, especially expression vector development through signal peptide. Okay, what is signal peptide? I think some of you already know that. And here we have a target protein, our target protein, and the signal peptide is a little bit earlier than your target protein, and it has 3 regions. This blue region is cleavage region. This sequence is you cannot change much because enzyme should cut this side. So if you change something, that enzyme cannot cleave this part by enzymatic reaction. And there is a hydrophobic region in the middle. There are hydrophobic amino acid to form the alpha helical structure, and there is an N region. This is positive charged. So this is very important part for especially the secretional protein. So I will show you one cartoon in textbook. So this is it. So this is a ribosome here, and the signal peptide is always translated first before your target protein. And then there is a signal recognition particle. They bounce together like this, and then they bind to a signal recognition particle receptor protein, and then your target protein can be translated and put into the ER rumen here, and then signal peptide can be removed and cleaved and removed, and finally you get the protein inside the rumen, inside the ER rumen, and then your protein can be followed by the post-translational modification, like cutting and then glycosylation or something like that. So the signal peptide synthesis is translation is the first step important for secretional protein for secretion. So this is important. And then when you look at the interaction between signal peptide and the signal recognition particle, they have an interaction in M domain and with the signal peptide through alpha helical. This yellow one is alpha helical structure. They interact through this alpha helical structure. So in signal peptide, alpha helical structure is mainly located in the H region. That is a hydrophobic region. So we thought that we can change or increase or decrease the hydrophobicity of the hydrophobic region, that is H region. But in this case, we decide to increase because some paper already show that. So we decide to increase the hydrophobicity of the H region. So when we increase the hydrophobicity, we decide to use leucine instead of other hydrophobic amino acid. And also from the codon usage table like this, we decide to use a CTZ codon for leucine. So this one is the signal, human IL-2 signal peptide example. There are 20 amino acid, and this part in the yellow box, this region is hydrophobic region. And if you look at closely, then you will find these blue ones are hydrophobic. Okay, let me close this first. Okay, so the blue one is a hydrophobic amino acid, and the red one is a polar amino acid, and the green one, the green one is basic amino acid. So in hydrophobic region, the H region, you still can find the non-hydrophobic amino acid. For example, here, serine and cysteine is here, and isoleucine and alanine is not that strong as like leucine in hydrophobicity. So in a previous study with the erythropoietin, not the interferon beta, in my lab, we changed the hydrophobicity by changing the amino acid in this hydrophobic region. For example, we replaced the serine to leucine, cysteine with the leucine, and both together with the two leucine, or 4 of them, not strong or not hydrophobic amino acid to the strong amino acid, this hydrophobic amino acid, all leucine like this. So this kind of a strategy we applied, and by something is wrong. Okay, so by doing that, in a previous study, we could get the EPO concentration, transient transfected EPO production was increased, like a 2.7-fold increase when you change hydrophobicity in signal peptide. So this time we would like to focus on interferon beta. Interferon beta is very difficult to produce. The concentration is very low, and it is very unstable protein. So it's very difficult protein. So right now, this time we try with interferon beta. So here, this is the signal peptide of interferon beta. Most of the the secretional protein has their native signal peptide. So interferon beta is a secretional protein, so it also has its native interferon beta signal peptide, which is this one. So, and when you look at the hydrophobic region, it's very similar to IL-2 we already done with previous research. So we follow the same approach. We apply same approach to interferon beta signal peptide. So we changed glutamine to leucine and the QIA to 3 leucine as we done in previous research. And then we see the result of this approach. So this is introduction, and then I will explain the material method. For this research, we used the 4 different cell lines, HEC293 cells and CHO cells. One is HEC293A, which is adherent cells, and HEC293F cell is a suspension cell. And we have 2 CHO cell lines, CHO-K1 for adherent and CHO-S, XP-CHO-S for suspension cells. Because we try this, because what kind of a cell line is good for interferon beta production, we need to decide. So we have to use for all of these 4 cell lines to make to do this research. For adherent cells, we use lipopectamine 2000 for HEC293A and CHO-K1. We follow this process. And for suspension cells, we use the polyethylenemine or XP-fectamine for XP-CHO cells. So by using this kind of transfection protocol, we could get the transient transfection of our vector developed, expression vector developed. And to make a stable cell line in the final, because we want to make a final cell line that produce stably, not transient. So we need to make a establish stable cell line. So we did two different kind of selection protocol. First one is Geoshin. So selection one, phase 1, 2, and we use MTX selection protocol. So we follow the separate the selection process, and followed by the limited deleting method to have a single cell clone. And then we actually developed sandwich ELISA method to analyze the interferon beta concentration. And most of the material, like expression vector, we use PCDNA 3.1, which one is commercially available. So there's no secret material. We use the commercially available product, and we used already known information to develop, to increase the productivity of the cells we produce. So result. So first one, we changed the hydrophobic region of interferon beta synapeptide. So we actually increased the hydrophobicity of the hydrophobic region like this. For example, here we change the amino acid like this, and then the results like this. This one is interferon beta synapeptide wild type. It's a native one. When we change it, the hydrophobicity by replace the non-hydrophobic amino acid to hydrophobic leucine, we could get this kind of result. And of course, when we use no signal peptide, there is no protein secretion. So that's pretty much acceptable. However, the data shows that the opposite to our expectation, interferon beta productivity was not successful. So what's gonna happen? So, but we have a good experience with the IL-2 signal peptide. So we decide to, okay, maybe the interferon beta signal peptide is not that strong. So we decide to use the IL-2 signal peptide. So we went back to the IL-2 signal peptide that we already succeed in EPO. So we did the same approach. And then this one is, number one is a wild type interferon beta signal peptide. And the number two is a wild type IL-2 signal peptide. And then we, tested different, combination of the hydrophobicity fortified signal peptide of IL-2. And then it did not increase much, but still we could get the 1.5, 1.84 increase compared to the native interferon beta signal peptide. So, okay, we could get almost twofold increase. Is that enough? Actually, if it is a bioprocessing optimization process, 1.8-fold increase in productivity is pretty much acceptable and very successful. However, we are talking about the cell line development and vector development. So we could not satisfy with this kind of small number. So what else we can do? And then we learned that the project sequence is very important to get high productivity in vector engineering. And the Kojak sequence is the GCC RCC sequence. So R is a purine sequence, and so it can be adenine or guanine. So ACC, ACC or GCC, ACC or GCC, GCC sequence before the initiation codon. So if you put this sequence in there, Kojak sequence, we learned that you can get higher productivity. So we decide to apply this sequence in our vector. So this is the result. So when we do experiment with the HEC293 adherent cells, we could get, this is the number one is IL2 modified, SCIA changed to 4Ls. So this is a modified IL2 signal peptide. When we put the Kojak sequence in there, we could get about 3.5-fold increase, but just by adding the Kojak sequence. It's good. And compared to the wild type, the interferon beta native signal peptide, we could, this number is about 6.5-fold increase. So this is good. And in the HEC293F cells, this is suspension cell, we did similar experiment, and then we could get the 3.5-fold increase. And then actually HEC293F, the suspension cell, has a more productivity compared to the two HEC293A cell line. So compared to these two, we could get 3.2-fold. So compared to the initial interferon beta native signal peptide in HEC293A, we could get 21-fold increase in Kojak modified IL2 signal peptide produced in HEC293F cells. So this is good. We could get 20-fold increase. So compared to the previous one, 1.5, 1.8-fold increase, we could get the pretty much increase in productivity. So this is good. And also we did in CHO cells, because CHO cell is mostly widely used in industry, biopharmaceutical industry. This is also important. So we could get good increase, 9-fold increase with the Kojak sequence, and the 3-fold increase in Kojak sequence, especially in CHO-S cells, and this one is CHO-K1. And then the concentration difference, CHO-S cell line, we could get the between these two, the difference is about 104-fold, more than 100-fold. So if we think about from the native interferon beta signal peptide, it is more than 1,000-fold actually. When we use XP CHO-S cell line with Kojak and modified IL2 signal peptide, we could get 1,000-fold increase before and after. So this is amazing. And then, okay, then we try to find is there any other way to increase the productivity. We already have a modified IL2 signal peptide. So we want to change a little bit more because we have in here, this is our modified signal peptide. We have 4 extra leucine, and originally there are 3 leucine here, and then we change it 4 amino acid to leucine. So we have 7 leucine in serial. So so we want to see if we change the number of leucine in this hydrophobic region, what's going to happen. We just curious about that. So we change that. So this one is our modified signal peptide with the Kojak sequence, and then we changed the number of leucine. This one is 5 and 6, 7, 8, 9. So we changed the leucine number, and that, but we could not get the enough productivity as we have already. So it seems like the productivity produced by our hydrophobic region modified IL-2 signal peptide has optimal, very close to optimal condition. And actually this one, number 4, has a 7 region, and our the modified one also 7 region, but difference is a codon different. So codon is a little bit optimized already in our signal peptide, but CTG might not be best in this occasion. So that's the another thing. We could get the better productivity in our signal peptide. Okay, and we actually this is the hydrophobic region. That's why we have interest. But there's another reason. This is a basic reason, and we cannot change all of this, but there is one polar amino acid, so glutamine here, and that here is a methionine. So we try to see, because the barrier between this is not clear. So we decide to change the glutamine to leucine to see the effect, and also we decide to change it both two to leucine like this. But the results show that if we change the M or M2 to leucine, we could not get the good productivity compared to the original one. So we should not touch anything about this basic region, and we should keep the size of the hydrophobic region as same as we have. So it seems like we have a optimal optimized the condition of the signal peptide already. And the next step is to make a stable cell line. So we start with the geosine selection system. We did with 400 microgram per milliliter of geosine, and we could get some cells survived already. And then we did the second phase selection system. And then after that, we could get, this one is signal Kozak sequence and signal peptide optimized, modified system, but it's a transient gene expression, transient transfection. And after we make a stable, selection system, second phase, we could get this one. And then when we compare these two, there's almost a 12-fold increase compared to the transient transfection and the stable pool, not a single clone. It's a stable pool cells. We could get a 12-fold increase. So we select this pool to isolate single clone. So we did limited dilution cloning procedure. And then we finalized the three, most productive clones, 3 of them, and then we finally cultivate them and then compare the productivity. So we have 3 different clones, so Z1, Z2, G3 clone, and then we could see the G3 clone shows the highest productivity, and like this number, and this number is compared to the transient gene expression system, this clone gives us a 35-fold increase in productivity. So actually, this the increase in productivity is more than 1,000, because we already know that when we use the native interferon beta signal peptide, this one is already 1,000, and then we have extra 35-fold increase. So it's pretty much quantum jump from the beginning to the current stage. And also we did the MTX methotrexate selection phase, and we could get the very similar productivity like this. So conclusion. So we, in order to develop the highly productive cell line, we have to touch the signal peptide vector engineering. From interferon beta wild type, we use IL-2 wild type, and we change the signal peptide in hydrophobicity, and then we put the Kozak sequence. And when we did the IL-2 signal peptide, we could get 1.5-fold increase, and then 1.8-fold increase when we modify the hydrophobicity. And when we add the Kozak sequence, we could get the 6.5-fold. And then use HK E93F cell, 293F cell line, we could get the 21-fold. And then also when we did the same procedure with the CHO cells, we could get the 104-fold increase in productivity. So this is pretty much high productivity compared to the signal peptide from interferon beta. So also when we do the stable gene expression, that means we can establish a stable cell line, cell clone. We could get the 35-fold increase from transient gene expression. We could get the 35-fold increase, and also when we use the MTX selection system, we could get 28-fold increase in productivity. So today I show that how we successfully increase the productivity in cell line engineering. Especially we touch the signal peptide, we change or modify the hydrophobicity, and we put some the important sequence, Kozak sequence there. And then after we finalize the selection system, we all together we could get the more than 1,000-fold increase compared to the non-treated original native signal peptide applied cell culture system. So this kind of thing, I don't know, but maybe some of these finding we got from here can be applied to the developed cell lines producing recombinant therapeutic cells, therapeutic proteins, which is a kind of major part of biopharmaceutical industry in these days. So this is the end of my talk. Thank you very much for your attention.
Thank you, Professor Dugjeo, for a very great presentation. And let us give a round of applause to Professor Jugde, and you can also give reaction.
Thank you.
Thank you, Professor. Ladies and gentlemen, we will continue to our next agenda, which is plenary session with speakers, Professor Zihua Jiang from Department of Animal Sciences, Washington State University, USA, and also Mr. Zahir Ali, PhD, Center for Desert Agriculture, King Abdullah University of Science and Technology. This session will be moderated by Professor Siti Subandia. Now, before we begin, allow me to read a brief profile of Professor Siti. Professor Siti Subandia earned her doctoral degree in plant pathology, the United Graduate School of Agricultural Science, Gifu University, Japan, master degree in plant pathology, Faculty of Agriculture, University of Queensland, Australia, bachelor degree in phytopathology, Faculty of Agriculture, University of Gadjah Mada, Indonesia. She is currently a professor in Faculty of Agriculture, University of Gadjah Mada, where previously she was also a faculty of member in 1983 to 2017. She was honorary professor in the university, she is currently the honorary professor in the University of Queensland, Australia, guest professor in Gifu University, Japan, and visiting professor in Shizuoka University, Japan. Her research interests are in the area of diagnosis and management of plant diseases and molecular plant protection, and also agriculture biotechnology. Now, without further ado, Professor Siti, time is yours to lead the session.
Okay, thank you very much, Ibu Wiwit. A very nice introduction of me. And good morning, good afternoon, or good evening for everybody in this meeting. I hope everyone is stay healthy, and we have already having better condition. The pandemic already declining. Yeah. Okay. Thank you very much for this opportunity for me to lead this talk. And the first speaker will be Professor Ji-Hua Jiang from USA. Yeah. Can I read the CV of Professor Jiang? Okay. Professor Ji-Hua Jiang is a full professor at the Department of Animal Science, Washington State University, as a faculty member of the University of Guadeloupe in 1997 to 2002. And he was doing postdoctoral at Roslin Institute in UK, 1994 until 1997, and then as a faculty member at Nanjing Agricultural University in China since 1986 to 1997. So his research interest is focusing on the studying of the origin of phenotypes using state-of-the-art genomic and transcriptomic approaches in combination with bioinformatics and cell embryo biology. So it's very interesting field. So Professor Ji-Hua Jiang, sorry, you have 30 minutes to present your research, and we are looking forward for your great talk. Okay, thank you, please.
Can you hear me?
Yes.
Okay, wonderful. Just to let you know, we have kind of the heavy wind outside, or you hear some kind of background noise. Sorry about that. First of all, thank you so much, Professor Dr. Subantia. Hopefully I can pronounce your name correctly.
Just call me Siti.
Oh, okay, Siti.
Siti is easy.
Okay,
Yeah.
Professor Dr. Siti, for your nice introduction. I would also take this opportunity to thank the conference organizer, Dr. Bao, for inviting me to give a talk today. My topic today is biodiversity. Does RNA diversity matter? As you know, biodiversity has been a hot topic in biology for more than 35 years. Aims to study variety and variability of life on Earth. As you can see here, the research field mainly focus on three areas: genetic diversity, species, ecosystems. No doubt, the number of species on Earth is enormous, while the ecosystems are very, very complicated. So therefore, my talk today would focus on genetic diversity, particularly on RNA diversity.
The committee haven't presented the PowerPoint.
So, can you see everything clear? Okay.
No, we cannot see it.
Can you see my slides?
I cannot see in my laptop.
This is clear in my.
Oh, sorry. What's wrong with
Can you see my Can you see my slides?
Yes.
Yes, we
Oh,
can.
sorry. I cannot
Okay.
see in my. Oh, sorry.
Oh, sorry about that. Okay, the question here is why RNA diversity? First, we can simply classify our human genome, for example, into the exonic, intronic, and intergenic regions. Based on ENCODE studies, it estimated that only 7% our human genome is silent. So that means that the remaining 93% of our human genome are actively transcribed and associated with at least one primary transcript. As you can see here, that includes 1.2% expressed from protein coding exons, 39% derived from introns, 3 UTRs, and also 44% mapped outside of the protein coding genes. That's kind of one thing we need to know. Second, in addition to protein coding genes, you can see here there are several types of non-coding genes, such as SNRNAs, RNAs, tRNAs, long non-coding RNAs, microRNAs, and pRNAs, for example. Third, you can see here a gene often use more than one transcript. And for example, alternative polyadenylation can occur at the tandem 3' UTR sites, alternative terminal exons, intronic sites, and exonic coding sequence size. So what's the consequence if you look at these, these alternative transcripts? For the first top two, you can see these two transcripts just have different lengths of 3 UTR regions. And they do code for the same proteins. That means no protein change for first two alternative transcripts. However, you can usually there are lots of regular elements residing in the 3' UTR regions. For this case, for example, and the short version of transcripts may lose the microRNA size, so the transcript should stay much longer than longer one. So from here, you can see, so-called quantitative effects when two transcripts are used in the process. So again, if we look at bottom 3 alternative transcripts, they may code for different proteins. So these proteins may have a different property, such we'll see so-called qualitative effects on gene function. In some case, protein coding gene can be converted into non-coding gene. As you know, obviously, non-coding gene will execute epigenetic effects. So anyway, so you can see here when a gene uses different isoforms, it may functions quantitatively, qualitatively, or epigenetically. So anyway, so gene overall use of alternative transcripts makes a finite genome capable to coordinate an infinite phenome. Okay. Okay, somehow I got something here I could get fixed right away. So you can see this slide shows you a typical structure about mature RNA. So in this case, if you would like to profile a gene, everything from the 5' end to 3' end. So you can use RNA-seq, conventional RNA-seq, or iso-seq. However, if you would explore how alternative transcription start size or alternative polyadenylation size are used in response to different conditions. So you need to use either 5' end sequencing or 3' end sequencing methods. So here, I just show you our methods to profile alternative polyadenylation sites. That's called whole transform termini site sequencing, or just WTTS thing. As you can see here, our WTS-seq methods uses total RNA as starting materials. The assay involves fragmentation, poly-RNA enrichment, first strand cDNA synthesis by reverse transcription, and the second strand cDNA synthesis by PCR. Actually, I want to tell you a few unique features about our WTTS-seq methods. So you can see here, actually, our WTTS-seq methods as both 5' adapter and 3' adapter simultaneously by during a reverse transcription process. That's number one. Number two, as you can see here, poly-A RNA fragment are enriched using oligo GT25 bits, and also poly-A cDNA. Okay, poly-A plus cDNA molecules are enriched by using RNSi and H treatment. Third one, where you can see our primers here, adapters. So basically, we use so-called poly anchored primers to do PCR. So that's avoiding any PCR amplification detour. Lastly, we try to avoid any over amplification. So basically, that helps us to maximize spectrum coverage. Now, let me tell you a few advantages our WTTS-seq methods over the conventional RNA-seq method. So first one, you can see here, compared to RNA-seq methods, our WTTS-seq can directly measure the diversity and the dynamics of APA patterns. So you can see here we use the embryos at the stages 6, 8, 11, 15, and 28. Use of frog embryos, and the same samples derived at stage 6, 8, and 11 were also done used for RNA-seq analysis. So from the top part, you can see here you see two APA size. You can call them, one distal one, another one proximal one. So you can see at stage 6 and 8, distal APA site is more abundantly expressed than the proximal site. However, things are changing. So I would say, at 11 and 15, you can see the proximal site the proximal side is more abundantly expressed than the distal side. However, at the stage 28, looks like both sides are kind of balanced. However, if you look at RNA-seq, RNA-seq profiles, you cannot see any such kind of changes. Obviously, if you look at transcripts, there are pretty long ones. There are pretty, short ones. For example, so this is Xenopus trabeculus ribosomal protein like genes 34 RPL 34 ribosomal protein L34. So this gene, okay, basically you look at express mature RNA. It has just 449 base pair, so it's pretty short. So you can see, look at the RNA-seq data. So that's kind of, obviously that's not well presented in the data set. So basically, conventional RNA-seq is biased against 3' ends of transcripts. So you can not see clearly their expression levels. However, if we look at our WTTS-seq, and that's pretty clear, and the gene expression increases along these developmental stages. Now that in our genome there are overlapping genes, our design is very unique, which our WTS-seq just a sequence from the polysize. So that's kind of the strand-specific sequencing. So as you can see here, you can clearly assign reads to the overlapped genes based on the color. However, if you look at conventional RNA-seq data, and we have no clear way to assign reads to each gene. So as I said, our WTA-seq uses strand-specific sequencing, so that reads can be assigned to overlapping gene easily and clearly. And also, obviously, as you can see, APS size can be extended from exon regions to introns. They might be located in introns or expressed from the antisense strand. There are cases. So if you look at RNA-seq data, basically you have no way to identify them. If you look at our WTS-seq data, so that's pretty clear. For example, here is a GCP1A gene. As you can see, APS site is expressed at starting at stage 11, and that's pretty dominant at stage 28. Well, and here this slide shows you comparison of transcriptome distributions. As you can see, RNA-seq data produce pretty wider of the wider distributions than the our WTS-seq. As a consequence, RNA-seq analysis would exaggerate the numbers of differentially expressed genes. So for RNA-seq data, you may see some false positive results. Here's the last one, based on, so usually for RNA-seq data, and you need to have at least, RNA-seq requires at least 30 million reads per library. However, for our WTS-seq, usually we use, we just require 5 to 10 million reads. So overall, our technique will save some sequencing cost. Okay, now I would use a few examples to tell you some interesting story. The first one I want to show you the relationships from gene knockout, APA, and muscle mass changes. So here, as you know, MPK alpha 1 and MPK alpha 2 are two alpha subunits of adenosine monophosphate activated protein kinase, which serves as cellular sensors in regulation of many biological events. here we investigate MPK, MPK-R1 knockout and MPK-R2 knockout mice on muscle mass changes. So the knockout mice was produced by Violet and colleagues in 2003. If I remember correctly, the deleted exon 2 for each gene, and so these genes are located. AMPK-R1 is located on chromosome 15, and MPK-R2 resides on chromosome number 4. So we used muscle samples to profile APA events. So as you can see, our analysis shows that down-regulated, you can see you can see upregulated and down-regulated genes, between two knockout mice. Actually, we use the wild type, as a control, as a control. So for the pathway analysis, so we used the either upregulated gene or downregulated gene for pathway analysis. At the end, we observed that downregulated APA size in MPK alpha 1 knockout, okay, knockout, and are downregulated, while many genes are upregulated in alpha, MPK alpha 2 knockout mice. So at the end, you can see MPK alpha 1 knockout mouse have much smaller, muscle area, while MPK alpha 2 mice produce much larger muscle area. So kind of you can see here, profiling APA size is a sensitive and a powerful approach that can be used to detail the information flows from a genome to transform to proteome and explain the altered phenotypes between knockout mice and wild type animals. So here I use frog, embryos and young males and growing females to tell you a story about development. APA.
Can you have slideshow, Professor John? Slideshow.
Sorry.
To have bigger view.
Okay. Yeah. Thank you.
Yes, that's better.
Okay. Okay. So here you can see we use the embryos derived from, again, from 5 stages, 6, 8, 11, 15, and 28. So therefore, adults, so we have 2 stages, kind of young animals and growing animals. One question here, I put it in a box here. So first question, are males and females genomically equal? That's kind of a question here. As you know, in mammals, males and females differ in sex chromosomes. Males have XY sex chromosomes, and the females have XX, sex chromosomes. As you know, in order to balance the dosage differences between sexes, one of the X chromosomes in females is inactive, but with 15 to 30% of its genes activated to counterpart the genes held by the Y chromosome in males. So I would say males and females are genomically equal. However, gender difference exists. Gender difference affects many different processes. So as you can see from the top left side, so that basically tell you that's kind of principal components analysis revealed kind of relationships among these samples. And based on color, you can clearly see where embryos are located, where the few young males and young females, growing males and growing females are located. Overall, you can see the APA transcriptomes of young animals are more variable than growing adults. That's kind of one thing you can see there. And then you can see overall male frog APA transcriptomes are more divergent than females. You can see if we look at the 6 growing males and females, obviously 6 females are clustered, together, while you can see pretty separations among 6 male animals. Why? So here I just use one pathway to tell you the story. So if we look at the genes involved in sex differentiation, that's pretty interesting here. Do you see among 110 genes involved in sex differentiation, only 20 are common to both males and females. As you can see here, 6 specific genes are involved in male sex differentiation, while only 30 genes are specific to female sex differentiation. And I don't think I have time to go through pathways in details, but here you can see a story. You can look at a pathway, and I can, the question here: why do females live longer than males generally? So also you can see a specific pathway for telomere maintenance. So you can see females have much better situation in telomere maintenance than males. So that I don't I don't think you need me to explain further. So that tells a story why females lives longer than males. Well, so let me probably need to speed up a little bit. So here you can see kind of issue obesity is a huge issue in the United States. The current obesity rate is for 42% in the US. So here we look at APA events in response to high fat diet. And you can see here, high fat diet triggered more genes upregulated in hypothalamus. As a consequence, so these animals ate more food and grew more body weight. So here's is another interesting case about cannabis exposure, APA, and brain functions. As you know, marijuana use has been legalized in many states in the USA. And so based on kind of reports, and 48 million people used marijuana at least once in 2019. Okay, that's a big number. That's a big number. So we use rat and also one-time exposure, that's acute exposure of cannabis for 10 minutes. So as you can see here, and hypothalamus responded to cannabis exposure pretty quickly. So as you can see, many genes were down-regulated in response to cannabis exposure. So if you look at the pathways, so basically cannabis exposure impairs learning, memory, mode, moment, sense, and thinking. That's pretty reflect what people feel when they use cannabis. This kind of last example, that's also a big issue in dairy. Okay, so that's try to tell you a story about pathogens, APA, and endometritis in dairy cows. Endometritis is one of the most devastating reproductive diseases in dairy cattle, resulting in annual loss of more than $50 million in the United States. So we screened dairy cows and collected samples from healthy and healthy and unhealthy cows. So basically, we look at here. We have collected 18 samples, and we could, based on APA profiles, we are able to classify them into clinically healthy animals and conditionally healthy dairy cows, clinically mild, mild endometritis cows, and clinically severe, severe endometritis case. As you can see here, and lots of pathways related to immune response are upper regulate. I want you to pay attention. So for healthy cow, you can see the pathway called cilium moment is dominantly enriched. So in order to keep cow healthy. Basically, psyllium moment function has to be normal. So anyway, these are kind of stories tell you how APA events in response to internal, external, and environmental challenges. So here is my kind of summary. And so if you look at the genome annotation, so we know how many number of genes in our genomes. However, if we look at numbers of alternative transcripts, the number is much higher than number of genes. Based on our research, I can tell you that alternative transcripts rather than genes are minimal functional units expressed by genomes. So I guess use of alternative transcripts makes sense. Makes sense. If you look at as I said, if you look at our phenome, okay, that's a phenome. That's pretty involving, and you can see many phenomes. However, you look at our genomes, that's kind of the limited. So basically, use of alternative transcripts makes it possible for finite genome to coordinate infinite phenome. So overall, I would say alternative transcripts serve as bridges between genomes and phenomes. And if we want to understand, create, or maintain the biodiversity, so we need to pay attention to the use of alternative transcripts. The last things I would suggest here are hypotheses to be tested. Loss of iron diversity results in extinction of organisms. Thank you so much for your attention.
Thank you very much, Professor Xiang. So we have run out our time, and we will invite our second speaker, Mr. Zahir Ali, PhD. I think he's available now, and so we will have a question and answer after the second presenter finishes his talk. Is it okay? Okay. So, Dr. Zahir Ali, please.
Hi.
Hello.
Can you hear me?
Yes. Okay, thank you.
Yep.
Okay, welcome, Dr. Zahir Ali. Yeah.
So, yep.
Can I introduce you? Yeah. Can I introduce you first? Okay.
Yes.
Mr. Zahir Ali, PhD. He got his PhD from Plant Molecular Biology and Biotechnology, Gyeongsang National University in South Korea, and then his master at Quaid-e Azam University, Islamabad in Pakistan, and then his PhD, his BSc was in the University of Peshawar, Pakistan. Okay, his work experience as a research scientist at KAUST, Saudi Arabia, and then postdoctoral fellow to 2013 to 2018, right? At KAUST, Saudi Arabia also, and then lecturer at Department of Biotechnology, University of Malakkat, Pakistan in 2003-2007. So he has very good experiences. So his research interest is in developing tools for plant genome engineering using specifically the CRISPR-Cas system And similarly, he likes to explore plant potential to be used as biofactories for the production of various bioproducts, drugs of therapeutic importance. Okay, very, very nice. Please, Dr. Zahir Ali, you have 30 minutes for your presentation. Thank you.
Yep. Thank you. I will share my screen. Can you see my slides?
Yes, it's coming.
Yep. Yep. My name is Zahir Ali. Good morning. Here it's morning. I don't know what time is there. Anyhow, it's really a good opportunity for me, and especially Dr. Rekno. He told me to participate in this one, and I'm really thankful for this, making this event. So actually, I'm a plant genome engineering scientist. But as you know, the recent spread of the COVID, and actually the system was ready. We were using it for plants, but the system can be used for any pathogen or any virus. So we developed something also for the COVID-19. So I will present today our recent work, which is just accepted, and I will just present this work for this event. So the title is Bioscan. We call it Bioscan, a CRISPR-Cas9 based lateral flow assay for detection of pathogens. Why a Bioscan? Like, for example, as we know, the COVID-19 pandemic, it's just started like 2 years back, exactly 2 years back. And then here you can see that actually in October 2020, the cases were just 34 million. But just in 1 year, the cases become very high, and you can see like 234 millions were there in October. And confirmed it globally from this pandemic in October, it was in October 2020, it was like 1.08. But in October 2021, it is 4.8 million. It's a huge, and still in some countries, still the lockdowns and all these things and all the lockdown measures are there, and still the people are suffering. But the main thing is like how to constrain a pandemic. So this was the call from World Health Organization, like they call it the 3T call. What is basically 3T call? It is like test, test, and test. So they just have like, they just say, we have a simple message for all countries, test, test, and test. Why test and test, test, and test? We make it a little bit more elaborate. Here you can see, like, for example, if there is a test that is affordable, fast, sensitive, and available to all the people, then you can trace. And if you trace, you can identify the cases and you can isolate the cases. And then if these are isolated and identified, then you can provide the treatment, supportive care, and in symptomatic cases. So the currently available method, which is normally used everywhere in the whole world, and it's a gold standard method, which is the qPCR method. What they do, like they just get the nasopharyngeal swab from the sample, and then they extract the RNA, and then they do the qPCR, and then they provide the results in the format of whatever. And here you can see that this is possible only in labs. It's not possible for individual. So what are the drawbacks of the RT-qPCR method? The main problem with this one, like you need a centralized lab where you need very specific designs and operations, and this is really a complex mechanism. And then you need the specialized people, like they should be trained very specifically for the qPCR run and for the result interpretation. Like the operators are not that much easy. Then the next one is like you have to purchase all the system, and as well as you have to hire the highly trained people, so which make it very costly. And normally, if you see the results are like from sample to the result outcome, there is a 24-hour gap, which is a very long time, especially in the case of pandemic like SARS-CoV-2. So what we think that a point of care, a simple test, are needed to accelerate the COVID-19 diagnostic, as we see very long lines when the people are going for the tests and all these things, even like by individual, or we see these lines everywhere when the pandemic started and when the pandemic was there. So these things, there is a huge need for point of care test to accelerate the COVID-19 diagnostic. An alternative should be there, which qPCR cannot do, because the qPCR, there is a limitation of the equipment, the labs, the people. So what we think, we want to develop a point of care diagnostic test. To start with, we look with the World Health Organization recommendation. They have an assured criteria for point of care diagnostics, which is like, which means it should be affordable to end user and affordable to health system. And S stands for like sensitive, avoid false negative, and the S specific, avoid false positives again. And user friendly, they should be simple. Like the qPCR, people cannot use by themselves. So they should be user friendly, and the people are maybe the simple clinics, they can detect the virus. It will be very easy, especially in pandemic cases. And they should be rapid and robust. Like the results you should get like 24 hours is a lot. The result should be get like within 1 hour or maximum by 2 hours. And equipment free, like there is like everywhere or every setup, or especially the developing countries, they don't have these sophisticated equipments and trained people. So the test should be equipment free, and it should be deliverable to the end user. So we are following this criteria to make a point of care diagnostic test. And if it's affordable, sensitive, specific, user friendly, and rapid, this can enhance the testing facilities for the pathogens, specifically the SARS-CoV-2. So to follow this one, recently the CRISPR-Cas system, which is immune system of the bacterial system, bacteria and archaea, this comes to the diagnostic, and this is called as like the next generation of point of care diagnostic tests. Basically, what is cluster regularly interspaced short palindromic repeats? This is actually the immune response of the bacteria against viruses. So what happened in the bacteria is basically like when the virus attacked the bacteria, it sent its genome into the to the bacterial cell. And what happened in the bacterial genome? The bacterial genome have a repetitive sequence. That's why it's called like clustered regularly interspaced. So it's like there is a cluster which is regularly interspaced. So there is specific region that are repeated again and again, and there are specific protein related to these sequences, which is the case nucleases. So what happened when the virus is there, the bacterial cell is playing a very good play with the virus. It's just take a small piece of the virus, use it as a memory and keep it in the genome. So when next time the virus come, it have a memory for this virus. So in the next step, this is the immunization against the virus. Like the bacterial cell keep a specific sequence as memory against this virus. So in the next step, when the virus come, like next time the virus attack, so here this sequence is expressed as single guide RNA, which is the memory of the virus. And this protein is produced, is sgRNA, and this protein make the complex, and it will cut the virus genome. So the virus cannot attack. So this is really a good mechanism which is present in the bacterial system and archaea system. And then this system is the targeting of the virus. And this system was recently adopted for genome engineering because you need only simple sequence of the gRNA, and then you can cut, or then you can use this system for guiding this protein to any nucleic acid. So the system is very simple, and it can be engineered very well for genome targeting, for the virus targeting, or for genome engineering in any species like mammalian cells, plant cells, bacterial cells, virus, whatever, for the virus resistance or pathogen resistance, and even diagnostics. So we want to take advantage of this simplicity. Like if we have a gRNA and if we have the case protein, we can use this for the detection of the virus as well. Not genome engineering and not virus resistance, but for the detection of the viruses. So to do that, if we look, like recently there are thousands of publications which show like how the Cas9 system is used for genome editing. It's need very simple. The requirements are very simple. You just need a 20 nucleotide sequence of the genome, which is followed by the 3 nucleotides like NGG. This is called as the PALM sequence. And this, if you have a 20 nucleotide followed by the 3 NGG, then you can target this sequence. You just design RNA, which is complementary to this target sequence, and then your Cas9 will make a complex. And then if you have this one, then there will be a cut in the genome. And once the genome is cutted, then because there is a random mutagenesis from here, and then there will be insertion and deletion in the genome, or the gene will be knocked down. It can be used for the repair as well if you provide the repair template. It can be also used for diagnostics because here you can bind your protein to a specific sequence of the nucleic acid. So the system is very simple and it can be adopted. But how to adopt this system for engineering of a diagnostic test? For that, we develop a mechanism which we call the BioScan. And that is a CRISPR-Cas9 based lateral flow assay for detection of pathogens. And what is BioScan? What is our hypothesis and what are the components of the BioScan? It's very simple. As we know, the lateral flow assay, which is commercially available, it have mainly 3 regions. Like, for example, it have the gold nanoparticles, which is conjugated with the antiform antibody. It have a streptavidin strip, or the streptavidin is impregnated into the lateral flow. And another line, which is called as control, is the antiform antibody. So this is the lateral flow strips, which is commonly available, commercially available, and it can be used. So we think that these commercial strips, they can be used for the SARS-CoV-2 detection very easily. But what we need a dCas9, which should be biotin level, because we know that biotin can interact with the streptavidin. So if this is the case, so we have the biotin and we have streptavidin. So this biotin can localize the case protein here at this region. And then if this is the case, then we can design a gRNA, which is complementary to the SARS-CoV-2 genome. And then the complex will be localized on the streptavidin strip. And this guide RNA we can easily make in vitro. This is just like you have a DNA sequence, then you can do the in vitro transcription. And if you produce the protein, then it can be used with the lateral flow strip. And if this is possible, you just need a simple heat block, or even water bath can be used with a specific temperature, because we don't need any amplification, any PCR amplification. It can be coupled with the isothermal amplification. And we just need the simple pipettes, or the simple and tubes and strips. So with this whole setup, you can do very easily the test for the SARS-CoV-2 or any pathogen. Next, we produced like how to produce the BioScan reagent. So we have a system where we put our DCAS9 with AV tag, and we have in the same cassette, in the same plasmid, we have another protein which is responsible for biotinylating this AV tag. So the biotin is there in the system, in the bacterial cell, and then we can purify the BioDCAS9. So to confirm, we induce the system and then we produce these proteins. So here you can see that the size of the DCAS9 is like 180 kDa, and here we can see very clearly that yes, the protein is produced. But the next question is like, is it biotinylated? So we use the Western blot and using the anti-biotin antibody. So using the anti-biotin antibody, here we confirm that yes, this protein is biotinylated. So if this is the case, then we can produce the gRNA, which will guide us to the SARS-CoV-2 genome or the SARS-CoV-2 nucleic acid. So we did the in vitro transcription. Now we have all the components, like the guiding sequence to the SARS-CoV-2 genome, and we have the BioDCAS9, and we have the lateral flow strip. So the whole concept or the working principle is that actually if you have the farm level primers, then you can amplify, first you can use the reverse transcription to convert the SARS-CoV-2 RNA into DNA, and then by the isothermal amplification, you can produce the DNA in 10 to 15 minutes in a single isothermal temperature, no need of any PCR. So we are using RPA method to amplify the DNA. And once this DNA is there, so we have a guide sequence to this DNA, and this is coming from the SARS-CoV. This DNA is made from the SARS-CoV, which is complementary here. So the complex will come here, and once this complex is on the DNA, in the next step, the complex will be bound with the gRNA and with the Cas9. And then we have the ends as farm level, and where we have the anti-farm antibody, which is coupled with the gold nanoparticles. So this whole complex will be made. And the next step, because we are using the lateral flow step, so if you put this complex on the sample pad, it will flow through, and if you have the SARS-CoV-2 genome or the SARS-CoV-2 is there in the sample, it will be stuck here because of the biotin-streptavidin interaction. But if not, it will flow through. So here is just a representation of the positive results. So in positive result, you can see on the test band that the appearance of the color band is here because the fragment will be accumulated or localized on this side. And some of them, like for example, the remaining nanoparticles with the gold in the form, they will move here. But in the negative, there is no band appearance here because this complex is not there, the SARS-CoV-2 is not there, and here on the test line, you will see nothing. So to follow this one, we have a simple sketch that you just collect the saliva from a patient. It can be done with the saliva or nasopharyngeal, but saliva is much more easy and that you can just spit in a tube. And the next step, a simple RNA extraction protocol we developed. It's a 5 minutes RNA extraction protocol. And then you can incubate at 42 degrees C for RT-RPA, which will convert the RNA into DNA and also amplify the DNA at 42 degrees C. So we don't need a PCR, but we just need a simple heat block or a water bath. And here you are using the anti-farm, the farm labeled primers. So the DNA will be labeled with the farm. And then we are providing this complex. So this complex will be made in the tube, like the BioDCAS9, sgRNA, and farm level DNA complex. So then this complex is applied to the sample pad, and the lateral flow will show you that you have a positive result or you have a negative result. So following this simple procedure, we try to develop our system for the SARS-CoV detection. So like this is just the general image, and here is the real result. We select two targets, like target 1 and target 2. And when we are using the specific gRNA, we can detect the target. But when we are using a non-specific gRNA, there is no target detection. Means our system is working very properly for target 1, and then we conform with the target 2, another nucleic acid sequence. But the next question is like the different controls. So like, for example, no SARS-CoV-2, no gRNA, no protein, and also the different like SARS-CoV-2 genome and the influenza virus genome. So here you can see that only the SARS-CoV-2 is detected, but all other negative control, they are clean on the test line. So means our test is very specific only for SARS-CoV-2 because of the complementarity of the sgRNA that we are using, the specific sgRNA for SARS-CoV-2. So to confirm that this can be used for the SARS-CoV-2, we select different region. Like for example, we want to amplify a short sequence from the SARS-CoV-2 genome. Here, this is the genome of the SARS-CoV-2, where we have ORF1A, ORF1B, and the spike protein, the envelope protein, membrane protein, and engine. So we select different region, like one in each gene. And here you can see that when we were amplifying these regions, two regions, which is like number 8, which is here from the ORFB, and engine, which is this one, the set 1. Here they are amplifying, or we are detecting these regions very well. Comparatively, these other regions, they were not that much efficient. And the main reason is like the primer binding. So the primer binding is very well at these two regions, but maybe the primers are not binding, or the efficiency of the primer or amplification is not that much good. So then we just follow only with this specific set of the primer to follow it for the detection of the SARS-CoV-2. But one of the main question is like, what is the sensitivity of this test? Because the CDC, they provide a CT value that only this much virus should be detected by a point of care test. So we put different amount of the copy number, like for example, 0 copy number of the SARS-CoV-2, 0.4 copy number of the SARS-CoV-2, 2 copy number of the SARS-CoV-2, 4 copy number of the SARS-CoV-2 genome, and 20 copy, 40 copy, and 400 copy. So here you can see our system is very sensitive, and even 4 copies of the SARS-CoV-2, like generally one microliter contain more than 1,000 copies in the patients. But here you can see that our system is very sensitive, and we can detect 4 copies per microliter very easily. This is just the conversion of this figure into the graphical format. So this shows that our system is detecting it very well. Then we validate our system with the clinical sample. So we collect like up to 94 sample from the clinical patients. And here you can see that on the test line, most of these clinical patients we can detect. Only 3 samples, like sample number 54, 69, and 73, they were not detectable. And these are the negative control, are the people who were recovered from the SARS-CoV-2 and they don't have the SARS-CoV-2 anymore. Next is the question, like there are different variants of the SARS-CoV-2. So is it possible that we can detect the SARS-CoV-2 variants? But these variants, as we know, that these are just point mutants. And the total system is dependent on the gRNA sequence. So let's say, for example, alpha variants have a deletion of 2 amino acids, 69 and 70. So if you design an sgRNA for this sequence, only the mutant, the alpha mutant, but not the wild type. So this can easily detect, or you can predict the prevalence of any specific virus or specific strain present in a specific region. And you can follow the distribution or the prevalence of the specific variants. So we detect, like for example, the alpha, beta, delta, and one of them is common mutation in all of them. And then we confirm that our system can specifically detect the alpha, beta sequence and delta sequence very well. So next, we confirm that like the next one of the biggest requirement of the point of care is it should be compatible with point of care, like specifically at room temperature or 4 degrees C, because at point of care, like for airports, stadiums, you cannot use -20 and -80 for storage. So we confirm that can our system work at room temperature, and can we store the reagent for a long time at these two temperatures specifically? For that, we confirm that yes, the system is working. And also the quick extraction of RNA, like for example, can we extract the DNA in 5 minutes? And yes, so we did. We here you can see that the system, when they are stored for weeks, like for example, the components, you can see like for first week, second week, third week, and fourth week, you can see the reagents are still working and they can be used very well at room temperature. There is no need of these storage conditions. And here you can see that when we are extracting the RNA by our method in 5 minutes, the system is working. So as a conclusion, I will say that bioscan reagents can be easily produced in the lab, because all the system you can make easily in any lab, in any molecular biology lab. But this is just the reagent production needs the lab. Other things don't need the lab. Bioscan is comparable in sensitivity with the commercial RT-qPCR because the sensitivity is very high. We see like 95% sensitivity. Bioscan doesn't require sophisticated lab equipments, no need of lab equipments, and it can be done with the simple point of care setup. The other thing is like Bioscan follow the World Health Organization assured criteria. It's affordable, sensitive, specific, user-friendly, rapid and robust, and it's equipment free. And you can deliver it into the users, and we are trying to develop it for like personal use, like if it's available in market, in clinics, so people can do it by themselves. No need to go to the labs. And bioscan detection assay can be performed in less than 1 hour. So like, for example, you collect the sample and then you treat the sample and then you got your results within an hour. No need to wait for like these 24 hours and also wait for the main labs. So this is our team, the bioscan team. This is Professor Magdi Mahfouz. He is our principal investigator. And Shiva, Nurhan, Ahmad, and Ten and Rashid. This is the bioscan team, and this picture was taken when everything was locked down and only these 7 people were allowed to come to the university. So I like this picture a lot. And thanks for your attention and question.
Okay, thank you very much. Great talk. So can we have applause for Professor Ji Huan Jiang and Dr. Zahir Ali? Thank you. And now we have about 15 minutes, even 16 minutes to have some question from the audience. You can raise your hand, or you also can write your questions in the chat box. Okay? Anyone? Any questions? Hello?
Hello, can I ask? Hello? Hello, can I ask question?
Yes, please.
Okay, hello, Prof. Zahir. Okay, I'm Pir Muhammad from Malaysia. So thank you for your presentation just now. Okay, I just started to learn about CRISPR. Okay, so I just want to ask your your opinion based on your experience. Okay, I feel to design the gRNA is the guide DNA is one of the most critical component of a successful CRISPR. So based on your experience, what's your advice to design a good guide DNA?
Basically, like the gRNA design is not a big problem at all. It's very simple. You just need the NGG sequence, like any like AGG, GGG, CGG, or like you just need NGG sequence and then 20 nucleotide. The only thing, like all the gRNAs, they are not working equally. So you have to search out in your gene which gRNA is working better. You can select the top strand, you can select the lower strand. And the sequence is the same sequence as you are targeting. So because you will design, it will be in vitro transcribed or the system transcribed, and it will become complementary to the lower strand. So the design is very simple. Specifically, if you are designing for Cas9, the Cas9 one is very specific and very simple compared to the Cas12 or the Cas13 and 14 systems. The Cas9 one is very simple and it's very widely used. So you just need 3 nucleotide, which is NGG sequence. 20 nucleotide, you can select any sequence. And NGG, the presence of 2 Gs in the genome, it's very common. So any target can be designed. The system is very simple. Like simplicity is there, and it's really robust. The Cas9 is really working very well.
Okay.
Thank you.
Okay. Thank you, Dr. Zahir. Anyone else?
Actually, I have a question for Dr. Zahir. And what's the cost for one test?
Okay. So actually, the good thing is like point of care. Again, there is one thing is affordability. Like it should be affordable. So like if you look at the lateral flow strip, so the lateral flow strip is the main cost. It's $3. The rest of the reagent we can produce in the lab. And like, for example, once you in vitro transcribe it, you can, like one in vitro transcription is enough for 3,000 samples. And one production of the DCAS9, biotin DCAS9 is enough for 10,000. So what we are expecting, it should be under $10. So this is what we are expecting, that the people can afford by themselves, like not by the governments or not. So it should be available in clinics, just like a normal test, like a pregnancy test. So the people can just take it and use it by themselves.
Great. Okay. Any more questions? We still have enough time. Come on. Okay, I would like to have a little question for Professor Chihua Jiang. So, on your APA analysis, in that case, when we would like to use something like antisense for stopping or knock down any unfavorable gene, maybe for the pathogenesis or whatever for the pathogen, how can we have the benefit from your APA pattern?
So basically, you can use our WTD-seq to profile APA changes in response to internal, external, and universal challenges. So that's kind of the tool to look at the kind of mechanisms involving relationships from genome to phenome. Basically, if you look at kind of for biodiversity research, in the past, probably many people have focused on DNA, variation. However, RNA, variation has not paid lots of attention. So obviously, and we have not thought about to use APA to do kind of the pathogen diagnosis. However, basically, I just show you a few examples, for disease, so we can see but I couldn't have time to explain everything in details. However, our technique can be used to study kind of the progression of diseases. In human, like cancer is a huge issue. Obviously, cancer is not a kind of one-day event. That takes time to develop. And so like from, something, you start something there and to the kind of pretty, kind of level 4 stage something, there should be lots of changes. So basically, we can use this sensitive tool to monitor kind of the progression of diseases or defects or some disorder.
Okay, thank you. Very nice. From the audience, if there is any question, we still have some time. No more? No more? Yes, question for Professor Silva. How to maximize the transcriptome coverage? Primers and adapter are designed for that.
Okay, that's a great question. That's a great question. When we started to develop our methods, we have tried different primer combinations, and so I mentioned that in my talk that we use so-called poly-anchored primers to avoid PCR amplification detour. That's the thing can help us to maximize the transcriptome coverage. That's kind of one thing you can do. Next one, try to use less cycles of PCR amplification. So as you know, so for our genome, we just have two copies, have everything. However, for transcriptome analysis, things are not equal. Some genes are expressed abundantly. Some genes are rarely expressed. So if you do many cycles of PCR amplification, that will favor abundantly expressed, APA size, not a rarely, expressed APA size. So that's the thing we need to pay attention. So in terms of protocol, can be used for plants. I will say yes. Use of alternative polyannulation is an evolutionary conserved event from animals to plants. Yeah, you can see lots of papers published using plant as materials. So yes, our assay will work on plants. So that's not a question. If you have RNA, to the RNA as materials, just use. Also depends on your kind of sequencing platform. So at the moment, our assay is based on ion torrent sequencing platform, not Illumina. So kind of tricky issue associated with the Illumina platform, because Illumina platform requires so-called diversity libraries. So look at our assay, so we have, our reads begins with several T's. So that's also, these are not a diversity library. So that's kind of a unique library. So Illumina system cannot process reads for you. So that's a problem. That's why we use ion torrent system. However, not a problem. We can adjust the parameter design and see. So one day definitely we can use Illumina system to do API profiling. So that's not a problem. Even bacteria, fungi, I guess, many organisms, they use alternative polyadenylation. So definitely these methods can be used for different organisms.
Okay, thank you. So we have still only 4 minutes left. If there is one more question, it's okay. No more? Okay. If there is no more questions, so thank you very much for Professor Xia Hua Jing and Mr. Zahir Ali, PhD, for your very interesting talk, and hopefully we can have more communication outside there. Yeah, okay, thank you very much, and give applause.
Thank you, it's my pleasure. Thank you.
Thank you.
Yeah.
Bye.
Thank you.
I send it to the committee.
Okay, thank you, Professor Siti, and also Professor Zihua Jiang and Dr. Zahir Ali for a very insightful session. Ladies and gentlemen, next we'll have a photo session with all of the participants. So if you could please kindly open your camera in Zoom so we could take a picture together. Okay, so the picture will be taken by the host, and there are 5 slides. Okay, so are we ready? On the count of 3, let me help for the first slides. Give your best smile to the camera. 1, 2, 3. The next slide. 1, 2, 3. The third slide. 1, 2, 3. The fourth slide. 1, 2, 3. And the last slide. 1, 2, 3. Thank you for your participation.
Okay.
Thank you.
Okay.
All right. So ladies and gentlemen, we would like to remind you to please kindly fill out the attendee list of the first session that has been informed in the chat. For the next agenda is Principle and Mechanism of Biotechnology Product Session 2. We will hear a presentation from Mr. Pandu from PT Bioscience with the topic Next Generation Sequencing to Advance Microbiology Study in Indonesia. Mr. Pandu, the next 30 minutes is yours. You may proceed.
Hi everyone, thank you for the time. Okay, let me share my screen first, and then please, I want to talk in Indonesia. Okay, here. Pertama-tama, terima kasih kepada Bapak Ibu yang sudah menyempatkan hadir dalam acara ISIBIO kali ini. Izinkan saya memperkenalkan diri terlebih dahulu. Saya Safitriani, merupakan Senior Field Application Scientist dari PT Pandu Bioscience, di mana PT Pandu Bioscience sendiri merupakan eksklusif distributor dari Next Generation Sequencing, yaitu Illumina System. Seperti yang telah disebutkan, kita akan membahas mengenai Next Generation Sequencing to Advance Microbiology Study in Indonesia. Oke, jadi di sini kita juga memiliki untuk giveaway untuk para partisipan yang hadir di dalam sini. Saya memberikan waktu kepada Bapak Ibu untuk bisa melakukan scan QR code-nya atau bisa langsung hubungi langsung di link yang sudah disediakan di layar Bapak Ibu sekalian untuk mendapatkan giveaway. Adapun giveaway-nya terdiri dari wireless charger, flash disk, and many more. Ini dia. Merupakan PT Pandu Bioscience. Untuk PT Pandu Bioscience sendiri berlokasi di Jalan Raya Pondok Gede nomor 10B, Duku Kramat Jati, di mana untuk informasi lebih lanjut mengenai PT Pandu Bioscience itu bisa diakses melalui info@bioscience.com atau di www.bioscience.com. Untuk produk dari PT Banu Biosense sendiri, kita merupakan eksklusif distributor dari next generation sequencing, terutama di Illumina system, seperti yang sudah Bapak Ibu ketahui sebelumnya. Lalu kemudian kita selain ada Illumina, kita juga memiliki Beckman Coulter untuk terkait dari liquid handling automation, dan ada beberapa dari molecular region juga terkait dari kit ekstraksi maupun kit PCR lainnya. Dan yang terakhir itu juga kita ada 10x genome untuk buat profiling single cell. Lalu selain itu juga kita ada beberapa brand yang kita distributor, yaitu ada Verogen untuk buat forensik NGS solution, ada Shimatsu protein analysis, Major Science General Lab Instrument, dan juga kita merupakan kerjasama dengan IDT, Archer, dan CARDX untuk LA typing. Apa yang kita sediakan untuk buat solusi dari Bapak Ibu ketika ingin melakukan research terkait apalagi terhadap dari biologi molekuler, banyak dari produk bidang keilmuan yang sudah kita bisa cover, baik dari onkologi, reproductive health, genetic disease, microbiology, agriculture, dan molecular. Yes, yang mana saja yang bisa di-cover, yaitu sudah bisa di-cover dari universitas, dari academic research center, pharmaceutical company, genome center, biotechnology, hospital, dan sampai ke government. Nah ini dia. Jadi topik yang akan kita bahas adalah bagaimana NGS memberikan solusi untuk di bidang mikrobiologi tentunya, yaitu adalah salah satunya dengan menggunakan metode whole genome sequencing, atau kita sebut biasanya familiar dan disebut total genome organism, yaitu satunya adalah dengan menggunakan culture based whole genome sequencing. Nah, ini salah satu contoh yang diambil dari Journal of Clinical Microbiology pada tahun 2018. Salah satu paper yang dibuat ini adalah ketika melakukan whole genome sequencing dengan menggunakan culture base untuk buat bakteri Clostridium difficile, yang dimana untuk dari sini kita bisa melihat bahwa terdapat analisa pohon filogenetik yang mana menghasilkan adanya kesimpulan, yaitu adanya transmisi dari AMR genes atau antimicrobial resistant di antara farm animal and human melalui dari bakteri Clostridium difficile ini. Nah, untuk teknologi ini total whole genome sequencing lah yang menjadi solusi ketika Bapak Ibu ingin melakukan tracking adanya AMR genes. Nah, ketika melakukan culturing atau melakukan kultur untuk melakukan whole genome sequencing, apa sih konsiderasi untuk buat bakteri dan viral whole genome sequencing? Untuk bakteri sendiri, kita memastikan apakah kultur kita pure monoisolate atau tidak, lalu bagaimana genome size-nya. Lalu pastikan kalau ketika kita melakukan kultur, dia 100% target organism. Lalu kita lihat di bagian virus. Ketika di bagian virus, kita lihat apakah dia berupa RNA atau DNA, apakah dia berupa single stranded DNA atau double stranded DNA, lalu apakah dia bentuknya host atau culture medium dari DNA atau RNA. Jadi kita bisa mengetahui dari sini, ketika kita mengetahui apakah dia pure isolate atau tidak, kita bisa melakukan metode selanjutnya untuk bisa mendapatkan hasil yang memuaskan, seperti itu. Nah, ini dia salah satu pipeline atau salah satu metode ketika Bapak Ibu ingin melakukan next generation sequencing dengan menggunakan Illumina system, yaitu kita bisa lihat di sini ada isolat dari virus, lalu kita bisa bagi yaitu apakah virus tersebut berupa RNA atau berupa DNA. Untuk DNA balik lagi tadi ada single strand atau double stranded. Nah, dari sini kita bisa lihat untuk buat RNA virus, kita perlu melakukan reverse transkriptase terlebih dahulu. Setelah dapat dari si DNA-nya, lalu kita bisa melakukan preparasi. Adapun tahap preparasinya dengan menggunakan kit Illumina, yaitu bisa menggunakan Illumina DNA Prep, lalu instrumen yang digunakan pilihannya antara NextSeq atau MySeq, lalu dilakukan data analisis. Untuk buat DNA-nya juga sama. Pertama, kita bisa melakukan ekstraksi terlebih dahulu untuk bisa mendapatkan material genetik berupa DNA. Terus kemudian kita bisa melakukan preparasi seperti pada RNA, yaitu dengan menggunakan Illumina DNA Prep, lalu instrumen yang dipilih yang ingin dilakukan apa, lalu dilakukan data analisis. Nah, ini dia library preparation atau
kit yang digunakan untuk buat melakukan whole genome sequencing dengan menggunakan Illumina system, yaitu dengan kita sebut kitnya yaitu adalah Illumina DNA Prep dengan metode augmentation atau pemotongan. Karena Illumina mengusungkan teknologi yang disebut short read sequencing atau pembacaan dengan jarak pendek seperti itu, maka dibutuhkan pemotongan terlebih dahulu. Nah, untuk itu, tahapan pemotongannya itu dilakukan dengan menggunakan enzim transposom. Illumina DNA Prep ini memiliki teknologi yang disebut sebagai bit link transposom, jadi transposomnya itu terlink dengan bits, sehingga hasil pemotongannya lebih seragam dan memudahkan para peneliti untuk melakukan data analisis untuk selanjutnya. Jadi Illumina DNA Prep bersifat versatil, robust, jadi mudah digunakan, reproduktif, dan fleksibel untuk buat berbagai jenis aplikasi yang akan digunakan. Nah, ini prosesnya, jadi berupa hasil dari genom DNA yang dihasilkan baik dari blood, saliva, atau dari genomic DNA hasil ekstraksi, kemudian input DNA-nya kemudian akan dipotong, lalu setelah dipotong baru kita lakukan preparasi untuk kemudian dilakukan sequencing. Seperti itu. Nah, untuk buat berapa sih input requirement-nya ketika melakukan next generation sequencing ini, itu dibutuhkan konsentrasi sekitar 1-500 nanogram, jadi sangat fleksibel sekali ketika Bapak Ibu memiliki material DNA yang sangat kecil sampai yang besar, baik dari culture maupun unculture, seperti itu. Lalu hasilnya nanti ketika di akhir tidak perlu melakukan normalisasi atau penyetaraan konsentrasi, karena di dalam teknologinya, di dalam library preparation ini, konsentrasinya sudah disetarakan, sehingga nanti proporsi setiap sampel ketika dilakukan sequencing itu akan seimbang, dan hasilnya akan lebih baik. Jadi ketika melakukan studi mikrobiologi, salah satunya adalah dengan menggunakan teknologi yang disebut sebagai, atau metode disebut sebagai metagenomic. Nah, untuk metagenomic sendiri, pembaginya ada dua, di mana kita mengetahui yang disebut pertama adalah targeted sequencing dan shotgun metagenomic. Untuk targeted sequencing, itu jadi kita mengamplifikasi terlebih dahulu untuk target amplikonnya, itu ada disesuaikan dengan studi yang ingin dilakukan, salah satunya adalah 12S, apakah 16S, CO1, atau 18S, dan ITS untuk fungi. Lalu untuk buat shotgun metagenomic, kita melakukan sequencing untuk seluruh DNA yang kita dapat. Jadi total genom DNA dari lingkungan yang kita ekstraksi, terus kemudian langsung kita lakukan sequencing, baru lakukan analisa untuk selanjutnya. Nah, bagaimana dengan 16S metagenomic sendiri? 16S metagenomic banyak familiar digunakan dengan menggunakan instrumen yang disebut sebagai MySeq system. Sudah ada protokol dari Illumina sendiri untuk terkait dari 16S metagenomics itu sendiri. Nah, dilakukan sequencing dengan menggunakan 500 cycle, atau kita biasa lakukan dengan 2 kali 250 sampai 2 kali 300 cycle, di mana untuk regionnya kita ketahui untuk 16S sendiri memiliki panjang 1500 base pair, yang mana untuk regionnya, untuk wilayahnya itu kita hanya mengamplifikasi di versi 3 dari gene 16S, seperti itu. Lalu kemudian dilakukan analisa data. Untuk analisa datanya itu bisa menggunakan MySeq Reporter atau longkaran manager dari instrumen. Jadi di dalam instrumen Illumina itu sudah diinputkan dengan bioinformatics software. Bapak Ibu bisa langsung mendapatkan data ketika melakukan sequencing, atau bisa menggunakan base pair sequence hub, yang mana base pair sequence hub ini merupakan software analisa data berbasis cloud. Jadi dibutuhkan akses internet untuk buat melakukan analisa data itu sendiri. Untuk protokolnya sudah disediakan oleh Illumina, tinggal mengikuti prosedur yang ada. Nah, ini bagaimana workflow-nya ketika melakukan 16S RNA sequencing, yaitu dilakukan sampel prep terlebih dahulu untuk bisa mendapatkan total genom dari lingkungan. Lalu setelah kita mendapatkan total genom dari lingkungan, kita hanya mengamplifikasi di version 3 sampai version 4 region amplification. Jadi panjang base pair-nya hanya sekitar 300 sampai 600 base pair. Lalu kemudian baru dilakukan tahap preparasi. Nah, setelah dilakukan preparasi, indeks yang digunakan itu disebut sebagai indeks Nextera dari Illumina. Lalu dilakukan sequencing menggunakan MySeq system seperti ini penampakannya. Lalu dilakukan analisa data dengan menggunakan base pair sequence hub. Adapun softwarenya yang bisa digunakan atau aplikasinya yaitu Kraken atau 16S metagenomic. Hasilnya, ini merupakan tampilan salah satu ketika Bapak Ibu melakukan analisa sistem S metagenomik, kita bisa mendapatkan profil mikrobiota dari lingkungan atau dari sampel-sampel yang dilakukan untuk buat sistem S metagenomik. Jadi memudahkan para peneliti untuk bisa melakukan profiling bakteria, sudah bisa mendapatkan komposisi bakterianya dan sudah bisa mendapatkan dari taksonomi atau klasifikasi golongan spesiesnya seperti itu. Nah, jadi ketika kita melakukan sistem S amplikan metagenomik, kita harus mengetahui apa saja sih keterbatasan ketika kita melakukan studi seperti ini, bahwa akan ada selalu kemungkinan sequencing error di dalamnya, sehingga membutuhkan optimasi atau melakukan pengulangan ketika melakukan sequencing seperti itu. Kita ingin mengetahui apakah komposisi di dalam lingkungan tersebut sudah sesuai atau tidak dengan komposisi bakteri yang kita temukan. Lalu adanya keterbatasan dalam melihat species level identification seperti itu. Terus kita juga bisa melihat kalau misalkan abundance atau komposisi berapa persen rates yang didapatkan ketika melakukan sistem S metagenomik ini dia bersifat relatif, jadi tidak bersifat absolut. Lalu ada kemungkinan dia menghasilkan PCR bias. Jadi bagaimana sih untuk bisa melakukan sistem S atau melakukan metagenomik, seberapa dalam kita bisa melakukan sequencingnya, bagaimana saya menentukan rates ketika kita ingin melakukan sistem S ataupun shotgun metagenomik, itu tergantung dari jenis sampel yang akan kita lakukan ataupun juga dari komposisi sampelnya seperti apa. Lalu kemudian kita juga bisa melakukan hipotesis terlebih dahulu, bisa mengetahui apakah kita ingin mengetahui di lingkungan tersebut harusnya sudah ada komposisi apa saja bakterinya di dalam lingkungan. Lalu kemudian kita juga bisa melihat apakah rates tersebut cukup atau tidak. Di sini sudah ada beberapa paper yang menstudikan mengenai berapa sih jumlah rates yang dibutuhkan ketika kita ingin melakukan sistem S maupun dari shotgun metagenomik itu sendiri. Nah, selain itu, di sini juga metode yang bisa dilakukan dengan menggunakan metagenomik adalah untuk fungi metagenomik, yaitu dengan menggunakan reagent ITS. Nah, untuk Illumina ITS protokolnya, ini merupakan pipeline yang dilakukan dari collecting extract sample, lalu dilakukan library prep reagent yang bisa digunakan itu sebenarnya bebas, bisa dari manapun. Nanti yang penting cocok untuk di mesinnya, salah satunya adalah kita juga menyediakan dari Illumina, lalu baru dilakukan organism test, terus dilakukan workflow-nya. Di sini juga sudah kita sediakan protokol untuk melakukan ITS metagenomik. Lalu dilakukan normalisasi atau dilakukan wet lab, lalu setelah selesai preparasi kita melakukan sequencing dari instrumen yang paling kecil itu ada ISIC sampai yang paling besar itu Novasic, lalu dilakukan report dengan menggunakan base-pair sequence hub, yaitu aplikasinya dengan menggunakan
Mr. Satriani, are you here with us? Or that it seems like the connection from Mr. Satriani is rather bad. So perhaps another person from Pandu Bioscience, if you could please continue with the presentation, or while we're waiting for Mrs. Satriani to join us. For Mrs. Aulia from Pandu Bioscience, are you here with us? Okay. Mrs. Safitriani, it seems like she already joined us. It's still unmute, Mrs. Safitriani.
Yeah, sorry for the interruption. So I can continue my presentation.
Yes, please proceed. Thank you.
Okay. Ya, terima kasih Bapak Ibu. Mohon maaf atas gangguannya. Jadi seperti yang sudah kita jelaskan di sini sudah ada gambaran mengenai bagaimana para peneliti melakukan ITS metagenomic seperti itu. Bisa terlihat dengan menggunakan ITS metagenomic pada Base Pair Sequence Hub, kita bisa mendapatkan profil dari fungi yang sudah dihasilkan dari suatu sampel lingkungan. Lalu kita bisa melihat komposisi atau seberapa banyak fungi yang bisa yang terdeteksi ketika kita melakukan ITS metagenomic ini. Nah, ini untuk buletinnya. Jadi kita di Illumina juga memiliki application note terkait dari bagaimana profil dari fungal sequencing and classification with the ITS metagenomic, bagaimana prosesnya, terus kemudian bagaimana juga tahapannya dari awal sampai akhir, terus hasilnya seperti apa. Nah, selain itu juga dari tadi kalau misalnya kita bahas mengenai ITS dan Cysteine S, maka di sini juga ada yang disebut sebagai shotgun metagenomic. Jadi kalau misalkan untuk buat 16S dan ITS, itu kita hanya mendapatkan amplikonnya. Jadi prinsipnya kita mengamplifikasi terlebih dahulu region amplikon yang ingin kita dapatkan, terus kemudian baru kita melakukan sequencing atau NGS. Sedangkan kalau shotgun, hasil sampel dari lingkungan, kemudian kita lakukan sequencing untuk seluruh total genom di dalam lingkungan tersebut. Jadi kita misalkan dari soil atau dari tanah, kita ambil, kita ekstraksi, terus kemudian total genom dari tanah, kita langsung bisa melakukan preparasi dengan menggunakan kit yang dari Illumina, baru kita bisa mendapatkan seluruh informasi terkait genom-genom apa saja yang didapat di dalam suatu lingkungan tersebut. Seperti itu. Nah, ini dia keuntungan dengan menggunakan shotgun metagenomic. Keuntungannya adalah bahwa shotgun metagenomic ini culture independent metodologi, jadi kita tidak perlu melakukan culturing terlebih dahulu. Kalau misalkan saya mau ingin mengetahui bakteri apa saja yang didapat di dalam suatu lingkungan, kita harus culture terlebih dahulu, lalu kita pisahkan setiap organisme di dalamnya dengan menggunakan shotgun. Kita tidak perlu melakukan culturing. Dia juga relative abundance of microbial taxa seperti yang pada Cysteine S metagenomic ataupun ITS. Metagenotyping artinya kita juga bisa melihat ada mengidentifikasi adanya gene loss, gain atau mutation di antara population cell atau microbial species. Terus kemudian kita bisa melihat dia hipotesis free testing. Jadi kita bisa mengetahui seluruh informasi di dalam sampel tersebut, apakah mikroorganisme apa saja di dalam sampel tersebut, apakah mikroorganismenya terdapat archaea, bakteri ataupun yang lainnya gitu, apakah sudah sesuai atau tidak, lalu bisa dilakukan pathogen detection, lalu juga bisa menghasilkan kelebihan-kelebihan lainnya. Nah, ini dia untuk buat application note dari shotgun metagenomic. Untuk kit yang digunakan adalah yang disebut sebagai Next-DNA Flex atau kita sebut sebagai sekarang Illumina DNA Prep, di mana untuk Illumina DNA Prep ini bisa melakukan preparasi untuk buat shotgun metagenomic dengan cara tadi, total genom yang sudah kita dapatkan langsung dilakukan pemotongan. Setelah dilakukan pemotongan, tambahkan indeks atau ditambahkan barcode, baru kita lakukan sequencing. Hasilnya itu bisa kita lihat bahwa dengan menggunakan NGS Illumina, hasilnya terdapat keakurasian data maupun klasifikasi untuk bisa melihat adanya perbedaan komposisi diversitas antar sampel di dalam sampel tanah. Jadi efisien dan komprehensif workflow untuk melakukan shotgun metagenomic dengan menggunakan Illumina system memudahkan para peneliti untuk mendapatkan informasi terkait taksonomi, diversity comparison, juga bisa memungkinkan untuk dilakukan de novo assembly. Jadi kita bisa salah satunya bisa mendapatkan total genome baik dari organisme-organisme yang terdeteksi di dalam suatu sampel lingkungan, lalu dilakukan functional profiling. Nah, untuk aplikasi-aplikasi atau software bioinformatik yang sudah kita gunakan, itu sudah ada berbagai macam dan disolusikan dalam base-pair sequence hub. Nah, ini salah satunya yang kita lakukan dengan menggunakan shotgun metagenomic, yaitu adalah dengan menggunakan sampel tanah, soil metagenomic. Ini bekerja sama dengan Perkin Elmer, dilakukan fully automated DNA extraction dengan Semagic 360 instrumen, DNA soil kit, lalu dilakukan tadi menggunakan Illumina DNA Prep atau teknologi yang disebut sebagai bit link transposon, dilakukan sequencingnya dengan menggunakan Nexus system dan analisisnya berupa Kraken. Yang kita lihat di sini, ketika Bapak Ibu melakukan shotgun metagenomic, maka visualisasi hasil akhirnya bisa kita lihat seperti ini. Jadi dia diklasifikasi, kita bisa lihat komposisinya. Di sini sudah ada organisme, lalu di sini sudah bisa dibagi organisme apa saja yang terdeteksi dalam sampel tanah tersebut, seperti itu. Hasil akhir solusi dari slide saya yang ingin sampaikan adalah untuk microbiology workflow solution dari NGS itu sendiri, bahwa ketika kita melakukan studi mikroorganisme dengan menggunakan NGS, kita bisa melakukan berbagai pipeline yang dilakukan, yaitu salah satunya untuk mendapatkan apakah kita ingin melakukan total genome sequencing, apakah ingin melakukan targeted sequencing atau S DNA sequencing, atau ingin melakukan shotgun metagenomic. Tadi shotgun metagenomic jadi mengambil semua informasi di dalam dalam satu sampel, lalu kita melakukan sequencing, lalu dilakukan targeted sequencing virology untuk virus, atau dilakukan RNA sequencing. Hasil akhirnya material genetik yang kita dapatkan, terus kemudian kita lakukan amplifikasi ketika pipeline-nya berupa targeted metagenomic atau targeted amplicon. Terus kemudian setelah didapatkan target DNA yang kita inginkan, lalu dilakukan preparasi libraries, di mana untuk library preparasinya itu juga disediakan oleh Illumina. Salah satunya adalah Nextera XT ataupun Illumina DNA Prep. Ada dilakukan sequencing, instrumen paling kecil yaitu adalah ISIC, terus kemudian yang di sini yang paling populer yang digunakan di Indonesia salah satunya ada MySic system, lalu kemudian dilakukan data analisis dengan menggunakan base-pair sequence hub dengan aplikasi-aplikasi yang disesuaikan dengan metode yang digunakan. Itu dia untuk metode dari NGS itu sendiri. Adapun kita mendistribusikan Beckman culture. Untuk Beckman culture-nya merupakan produk yang digunakan untuk genomic solution. Jadi kita memprovide para peneliti untuk melakukan ekstraksi kit dan juga untuk buat purifikasi. Di sini Beckman culture memiliki beberapa jenis produk, yaitu salah satunya DNA isolation kit, ada RNA isolation kit, total nucleic acid isolation from FFPE tissue. Adapun juga kita Beckman culture juga menyediakan untuk Ampure Bit. Sekali lagi bisa saya ingatkan kepada Bapak para Ibu yang ingin mendapatkan giveaway dari tim kami, bisa melakukan scan pada QR code yang tertera di layar, atau bisa akses di link yang sudah disediakan di layar Bapak Ibu. Adapun nanti untuk buat pemenangnya akan diinformasikan kepada tim kami. Demikian presentasi dari saya. Jika ada pertanyaan, dipersilakan. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
I apologize for the connection problem. So for Mrs. Safitriani, is your presentation is done? Okay, it seems that you're still muted.
Oh, sorry. Yeah, I ended my presentation and thank you for the attention.
Okay, thank you, Mrs. Safitriani from Pandu Bioscience. I'm sorry for the previous mistakes. Ladies and gentlemen, Before continuing to the next agenda, there would be some documentary videos from our partnership that we can watch. So let us watch the video together.
Halo, kami dari PT ITS Science Indonesia. Hari ini kami akan memberikan penjelasan singkat mengenai pengujian porcine. Mengapa perlu melakukan pengujian porcine pada produk pharmaceutical? Beberapa macam bahan baku atau obat, termasuk tablet, kapsul, injeksi, dan sebagainya, dapat berasal dari produk hewani atau turunannya. Contohnya seperti gelatin, yang sering dipakai sebagai bahan utama kapsul atau stabilizer pada vaksin. Selain itu, antikoagulan seperti heparin juga dapat berasal dari porcine. Sedangkan berdasarkan Undang-Undang Nomor 33 Tahun 2014 tentang jaminan produk halal, produk yang masuk, beredar, dan diperdagangkan di wilayah Indonesia wajib bersertifikat halal. Oke, jadi bahan apa saja yang dapat mengandung porcine? Kebanyakan senyawa tersebut merupakan antikoagulan, digestive supplement seperti enzim lipase, amilase, insulin, dan sebagainya. Wah, cukup banyak ya. Tapi tenang saja, pengujian porcine ini tidak sulit kok. Ada beberapa metode yang dapat digunakan untuk mengecek kandungan porcine pada produk. Yang pertama, lateral flow device atau test pack. Metode ini cepat dan mudah, tanpa perlu banyak peralatan laboratorium yang canggih, tetapi sensitivitasnya rendah. Metode 2, ELISA. Waktu preparasinya dibutuhkan 2-3 jam. Preparasinya cukup mudah dan dapat menguji dalam jumlah banyak dalam satu kali running. Dibandingkan lateral flow device, ELISA lebih spesifik, tetapi masih memiliki batas deteksi sensitivitas yang sedang. Metode 3, real-time PCR. Metode ini adalah golden standard untuk pengujian porcine pada produk pharmaceutical dengan limit deteksi yang sangat rendah 1-5 kopi DNA. Pengujian porcine pada produk pharmaceutical sebaiknya dilakukan dengan metode PCR karena bahan baku yang diuji telah melalui proses pemurnian atau pemanasan tinggi sehingga kemungkinan yang tersisa hanya materi genetiknya saja. Selanjutnya, tahap real-time PCR ada 3, yaitu preparasi sampel, ekstraksi DNA, dan amplifikasi dengan menggunakan mesin real-time PCR. Untuk lebih jelasnya, mari melihat alat real-time PCR dari Roche, Light Cycler 96. Alat berkapasitas 96 well ini memiliki 4 channel fluorescence yang dapat digunakan sekaligus dalam satu kali pembacaan. Sebelum memulai proses PCR, profil temperatur dan protokol PCR dimasukkan ke dalam mesin. Setelah itu, tekan tombol start. Secara perlahan akan terbentuk grafik sinyal seperti ini. Sinyal ini terbentuk dari proses denaturasi, annealing, dan elongasi yang berulang, sehingga fluorofor yang terkandung di dalam master mix dapat berpendat dan tertangkap oleh sensor mesin. Sinyal yang didapat kemudian dianalisis menggunakan software Light Cycler, sehingga terlihat sampel mana yang positif atau negatif. Demikian penjelasan singkat mengenai pengujian porcine dari kami. Sekian dan terima kasih.
Halo, kali ini saya mau memperkenalkan alat laboratorium deteksi engine. Ingenious Tree. Sebelum pengenalan alat, apakah Anda pernah mengalami kesulitan saat mendokumentasikan hasil gel electrophoresis atau plot Anda di ruang gelap? Sistem dokumentasi gel secara manual terdiri dari ruang gelap, kamera, dan transluminator sederhana. Dokumentasi gel secara manual seperti ini kadang menyulitkan dan belum lagi kalau hasil foto gel yang didapat ternyata blur. Dengan Ingenious Tree, Anda dapat memilih pencahayaan sinar UV, sinar biru, atau putih sesuai dengan pewarna yang Anda gunakan. Selain itu, software Genesis akan membantu Anda memilih pengaturan cahaya dan filter yang tepat sehingga sangat memudahkan untuk mendokumentasikan gel fluorescence ethidium bromida atau cybersafe DNA atau protein gel dengan composite blue atau silver staining. Untuk penggunaan mode otomatis, Anda hanya perlu memilih tipe gel yang sesuai, kemudian Software akan secara otomatis mengatur aperture, zoom, dan fokus serta posisi gel yang sesuai sehingga gambar gel terekam secara optimum. Software Genesis dilengkapi dengan database, tipe gel, dan pewarna yang tersedia di pasaran. Jadi jangan khawatir tidak bisa menemukan pengaturan yang sesuai. Sangat mudah. Hubungi tim ITS Science Indonesia untuk info lebih lanjut. Terima kasih. Stay safe.
Since 2019, PT Wadia Prima Mulia has continued to develop in meeting the needs of basic laboratories for Indonesian territories. Until now, our company has been trusted as an authorized distributor for 11 companies that produce the best laboratory equipment in the world. Our highly skilled and experienced sales team spread across Indonesia from Medan, Jakarta, Bandung, Semarang, and Surabaya. Our products. Sales services, including application and technical support, installation and training, also service and maintenance.
In the life sciences, researchers are actively pursuing studies aimed at understanding life, the causes of disease, and achieving breakthroughs in regenerative medicine. To observe and accurately measure intracellular processes and structures, researchers need a microscope that combines sensitivity and speed. Increasingly, scientists require the capability to view cells and tissue at high resolution, as well as their larger context with macro to micro capability. All of these advanced features must be easy to use, so that even novice users can acquire high-quality data quickly and simply. Olympus created the FV3000 series to satisfy these needs. This next generation of confocal laser scanning microscopes combines leading-edge imaging features with renowned Olympus optics.
Microtomy requires my full attention and dedication to make sure that I deliver the perfect slides. And with the Tissue Tech Auto section, I started getting the job done easier, faster, and with better quality. The automated block alignment makes sure precious tissue is preserved optimally. It is also a great help for making recuts, while the perfect alignment helps me produce even more slides. Trimming and sectioning are done with high precision, each section the same exact thickness. Thanks to the easy operation of the auto section, every technician in our laboratory is quickly up and running. Whatever the need, we are all confident to deliver the perfect slide each time. My name is Mark Shanti, and I'm empowered by Sakura.
All right, ladies and gentlemen, for the next, we will hear a presentation from Mrs. Sri Hayuni from Biotech on end-to-end solutions for biopharmaceutical workflow. And for Mrs. Sri Hayuni, the next 30 minutes is yours. You may proceed.
Hello, good afternoon, everyone. My name is Reha Yuni, and I'm going to share my screens first before I start my presentations. Here you go. All right. So first of all, I'm going to apologize because I'm going to deliver these presentations in Bahasa to make everyone in this room would understand and better about my my presentation. So allow me to deliver this presentation in Bahasa. Selamat siang semuanya. Perkenalkan, nama saya Sri Hayuni. Terima kasih untuk kesempatan yang diberikan kepada saya mewakili PT Merck Indonesia untuk mempresentasikan topik berjudul End-to-End Solution for Biopharmaceutical Workflow. Di sini presentasi ini akan saya saya coba. Just give me a second. I think I have a technical problem. Okay, just sorry. It seems like the PPT is closing. I'm going to reshare again. So sorry about this technical inconvenience. Okay. Oh, just give me a minute.
Do you want me
To
the
have a big-
end to end?
Yeah.
Yeah.
It's about to. Okay, cool. All right, cool. Does my share screen?
Yes.
Can you see
you
my
can.
screen? Okay, thank you.
Yep.
Okay, cool. Yeah. Sorry for this inconvenience, everyone. So, yes, I'm just starting my presentation again. Jadi presentasi ini akan dibawakan oleh saya saya Hayuni M. Biotech selaku Cell Culture Manager dari tim research PT Merck Chemicals and Life Science. Baik, sebelum saya memulai presentasi, saya ingin memperkenalkan sedikit mengenai portfolio PT Merck Life Science yang saat ini sangat ingin kita branding sebagai Life Science Company setelah mengakuisisi beberapa brand seperti Sigma Aldrich. Di kita sendiri ada beberapa divisi, yang pertama healthcare, kemudian performance material, dan life science. Khusus kebutuhan laboratorium Bapak Ibu, baik itu dari akademi maupun juga industrial, produk yang akan kita usung kebanyakan berasal dari industri life science bisnis di Merck sendiri, di mana di sini saya dan juga beberapa tim saya akan mendukung Bapak Ibu untuk kebutuhan laboratorium dan juga penelitiannya. Khusus yang healthcare, biasanya ini lebih ke arah obat-obatan yang sudah memiliki hak paten, dan khusus untuk performance materials, industri ini bergerak di bidang-bidang yang lebih ke arah coating, kemudian juga beberapa electronics yang mendukung teknologi seperti OLED. Nah, untuk bisnis life science sendiri, di sini kita ada 6 brand utama yang biasanya sering didengar oleh Bapak dan Ibu. Yang paling utama kali ini adalah Sigma Aldrich. Jadi ini sudah diakuisisi lebih dari 3 tahun yang lalu, dan produk kita ke arah life science lebih banyak diusung dari brand Sigma Aldrich ini sendiri. Nah, kemudian di kita juga ada Miliki, di Miliki biasanya ini mensupport lab water solution, baik itu di laboratorium maupun juga industri. Kemudian juga brand yang sudah cukup lama, sudah mengakar di PT Merck sendiri ini adalah Supelco Analytical Product. Jadi biasanya memang chemical classics. Kemudian juga kita ada Millipore. Kalau Millipore ini dia sudah lama kita akuisisi under brand of Merck. Dan untuk Millipore sendiri ini produk-produknya lebih ke arah preparation and separations. Kemudian juga kalau untuk media-media bakteri sebagai kisi gitu ya, itu lebih banyak brandnya ke arah Millipore. Nah, yang terakhir ini kita ada SAFC dan Bioreliance, di mana kedua brand ini lebih ke arah mendukung raw materials untuk industri yang dilakukan oleh customer-customer kita. SAFC mungkin banyak yang berhubungan dengan biopharmaceutical juga, dan Bioreliance ini mungkin lebih ke arah biopharmaceutical yang mengarah ke biologics maupun juga vaccines yang rekombinan, di mana nanti mungkin kalau misalnya Bapak dan Ibu ada kebutuhan untuk engineer cells untuk menghasilkan protein tertentu atau antibody, monoklonal antibody, kita memiliki tim yang khusus untuk memberikan paten engineer cell line yang atau stable clone yang bisa memproduksi produk biologics ataupun biosimilar yang Bapak Ibu ingin produksi. Baik, without further ado, I will present the portfolio that we can support for the biopharmaceutical. Herein saya ingin memperkenalkan sedikit produk biopharmaceutical atau industri biopharmaceutical apa saja yang bisa kita support. Jadi dimulai dari compound atau industri yang biologics yang menghasilkan compound, ini dia small molecule, biasanya dia lebih kecil dari 1 kilodalton. Ini merupakan salah satu industri biopharmaceutical yang saat ini sedang sangat booming juga. Salah satu produk yang terkenal mungkin Lipitor dari Pfizer ya untuk high cholesterol. Kemudian juga new biopharmaceutical pharmaceutical seperti monoclonal antibody MAB ini dia berupa therapeutic protein. Biasanya untuk ukuran dari monoclonal antibody MAB ini sendiri berkisar dari 25 sampai 800 kilodalton. Dan ini salah satu produk yang dari Merck sendiri di life science-nya sudah luncurkan adalah Keytruda, ya ini berupa checkpoint inhibitor for cancer. Kemudian juga saat ini biopharmaceutical lebih ke arah vaksin ya, terutama untuk SARS-CoV-2, di mana vaksin yang dimanufacture juga bukan hanya berasal dari inactivated gitu ya atau live attenuated vaccines, tapi sudah lebih ke arah mRNA, kemudian juga peptide. Bukan peptide vaccines tapi kayak therapeutic atau rekombinan vaccines gitu, sehingga untuk bisnis-bisnis biopharmaceutical ini kita memfokuskan beberapa produk yang ingin di-highlight untuk kebutuhannya. Nah, di bisnis kita sendiri karena kita memiliki 6 brand pilar yang menjadi dasar dari bisnis ini, maka dari mulai end to end, dari mulai R&D sampai biopharmaceuticalnya kita dapat dukung Bapak dan Ibu. Nah, secara umum untuk prosesnya sendiri kita akan membagi industri biopharmaceutical ini menjadi 3 kelompok, yaitu di bagian discovery, development, dan manufacturing. Nah, untuk produk-produk yang bisa kita support mungkin saya compactkan dalam slide saya di slide yang ini dan juga slide-slide berikutnya, di mana produk-produk highlight yang pertama dari untuk stages discovery sendiri mungkin ada beberapa stages ya, berupa biasanya berupa disease target identification, kemudian identify dan isolate target yang sesuai, kemudian candidate design, jadi termasuk PEG glycylation dan sebagainya ini masuk ke sini. Kemudian di karakterisasi ini juga masih dalam discovery step by step, dan ini merupakan produk yang biasanya kita dukung. Jadi untuk discovery sendiri tahapan awal yang paling sangat sederhana itu adalah teknik kultur sel, di mana di sini Sigma Aldrich sendiri sudah sangat populer, sudah sangat tua, berakar gitu untuk mensupport produk-produk yang berhubungan dengan culture cell. Jadi dimulai dari koleksi-koleksi sel lainnya. Jadi di sini kita untuk collection of the cell line itself, we have a variety of brands. For instance, for cell line we have ECAC cell line. Kemudian untuk primary cell we have cell application inks. Kemudian in millipore we usually support for primary and also stem cell. Nah, kemudian untuk teknologi terbaru juga ada induced pluripotent stem cell. Di sini kita biasanya membawakan atau mendeliver sel yang berasal dari ABISC. Di sini induced pluripotent stem cell yang sudah yang didistribusikan di sini merupakan stabil dari iPSC yang berasal dari donor-donor dan dimodifikasi dari lab-lab di berbagai belahan dunia. Nah, selain sel line, kita juga memiliki sera, media, antibiotik, growth factor, freezing medium, the cell disattachment factor yang sangat mendukung proses biopharmaceutical Bapak dan Ibu. Di sini mungkin saya highlight saja produknya. Jadi dimulai dari yang FBS. Kalau FBS sendiri, Sigma Aldrich sudah mendeliver FBS yang memiliki GMP certified. Kemudian juga untuk proses biopharmaceutical sendiri karena memang sangat rentan ya terhadap virus, kemudian juga other kontaminan yang bisa merubah atau mempengaruhi end product dari biologik itu sendiri, biasanya sangat rentan untuk virus, sehingga untuk FBS yang kita sarankan untuk biopharmaceutical itu adalah yang sudah gamma irradiated. Jadi kalau sudah gamma irradiated biasanya dia sudah eliminates virus-virus yang biasanya menginfeksi bovine itu sendiri. Jadi ada yang GMP manufacture dan gamma irradiated khusus untuk industri biopharmaceutical. Nah, kemudian juga karena industri biopharmaceutical ini nantinya akan dipakai oleh manusia, tentunya di Indonesia sangat concern dengan isu halal. Nah, untuk cell disassociation factornya sendiri biasanya menggunakan trypsin, di mana trypsin tersebut mengandung kandungan porsin, sehingga untuk tahapan awalnya kita sarankan tidak menggunakan trypsin gitu, pencacah sel yang berasal dari porsin. Di sini produk kita sendiri ada yang disebut sebagai trypzean. Jadi trypzean ini merupakan trypsin yang diambil dari gene pankreas bovine yang diekspresikan di jagung. gitu. Untuk hasil disattachment-nya sendiri bila dibandingkan dengan kompetitor, trypsin memiliki kekuatan cell disattachment atau enzim yang lebih kuat dibandingkan dengan other competitor, sehingga sangat convenience untuk kebutuhan biopharmaceutical Bapak dan Ibu. Nah, terakhir untuk cell freezing mediumnya sendiri, tentunya proses freezing merupakan hal yang paling krusial ya untuk di industri biopharmaceutical. Nah, di sini kita biasanya mensarankan cryostore. Cryostore ini adalah jenis cryopreservasi yang sudah ready to use, sehingga nanti pada perlu mix lagi ya serum, kemudian medianya untuk memfreezing selnya. Di cryostore ini sendiri dia sudah ready to use, dan rate of survival sel ketika dibangunkan jauh lebih tinggi bila dibandingkan dengan kompetitor atau produk lainnya. Nah, untuk produk-produk ini juga media, kemudian juga cell dissociation factor, kemudian cell freezing mediumnya juga ada yang serum free. Jadi kalau misalnya Bapak dan Ibu ingin melakukan ingin melakukan production biopharmaceutical product yang bebas dari animal komponen, dari awal kita sebenarnya sudah ada beberapa produk yang mendukung serum free environment untuk perkembangan kultur pertama atau stabil sel yang ingin Bapak Ibu kembangkan di laboratorium. Nah, selanjutnya tidak kalah penting juga adalah filtranya. Di sini kita bisa bawa millipore, di sini ada Stericup, kemudian juga beberapa syringe filtrasi yang biasanya digunakan dalam praktik kultur sel untuk menghindari kontaminasi. Terakhir, untuk labware alliance-nya sendiri, dari kita memiliki brand partner premium dari Corning, kemudian juga dari brand sendiri, di mana kedua brand ini bisa mendukung konsistensi hasil dan juga pyrogenik atau endotoxin free untuk alat-alat kultur atau consumable yang biasa Bapak Ibu gunakan di laboratorium masing-masing. Nah, selanjutnya biasanya selain dimulai in vitronya dari kultur sel, mungkin juga untuk studi tertentu akan ada asai yang lebih advance berupa 3D cell culture. Jadi bukan sekarang ini yang lagi happening bukan hanya cell culture yang biasa yang ditempelkan di plastik, tapi juga ada yang 3D cell culture. Nah, kita juga sangat support untuk kebutuhan Bapak dan Ibu mengenai 3D cell culture, di mana seperti yang kita ketahui 3D cell culture membuat asai-asai atau studi yang Bapak Ibu lakukan untuk sel, misalnya untuk penyerapan obat atau mungkin untuk bahkan untuk menghasilkan organ seperti kolon atau alveolus gitu ya untuk SARS-CoV-2. Nah, ini biasanya didukung dengan 3D cell culture technology. Nah, di kita ada 2 produk unggulan yang biasa kita sering sarankan gitu ya untuk studi disease modeling. Yang tertarik untuk 3D, yang pertama hydrogel. Jadi kalau di kita itu ada hydrogel yang sering digunakan itu adalah yang max gel sama yang ECM gel. Jadi berdasarkan dari testimoni beberapa customer yang sudah menggunakan ini, dengan menggunakan max gel dan ECM gel ini dapat meningkatkan proliferasi dari selnya sendiri, juga hasilnya sangat konsisten dan sangat bagus untuk studi disease modeling yang 3D. Selain hydrogel, kita juga memiliki 3D organoids portfolio, di mana Bapak dan Ibu bisa memimik organ gitu, jadi mengembangkan sel yang biasanya berasal dari induced pluripotent stem cell untuk dikembangkan menjadi organ. Jadi kalau yang ini adalah gambar 3D organoid berupa kolon, sehingga ketika Bapak dan Ibu berhasil mengembangkannya, kalau untuk studi absorption obat gitu, dia lebih memimik in vivo dibandingkan dengan hanya 2D cell culture teknik yang biasa dikembangkan di lab. Nah, selanjutnya dalam proses discovery juga nanti akan ada teknik di mana kita harus memodify selnya atau ya gene editing. Di sini saya ingin highlight gene editing kita berupa CRISPR di Merck sendiri. CRISPR gene editing memiliki portfolio yang cukup lengkap. Saya bisa infokan seperti itu. Contohnya seperti gRNA atau guide RNA, kemudian Cas9. Di guide RNA sendiri kita memiliki guide RNA yang hanya berupa protein maupun juga guide RNA yang sudah melekat pada plasmid. Selain itu kita juga kita juga memiliki sintetik gRNA atau guide RNA yang memiliki efektivitas lebih tinggi dengan gRNA yang biasa. Nah, untuk Cas9 sendiri, untuk enzimnya sendiri gitu, Cas9-nya sendiri juga sudah ada yang tercantum di dalam plasmid maupun juga sudah dalam vektor berupa lentiviral. Nah, produk unggulan dari Cas9 kita ada yang Cas9 GFP protein. Di sini adalah Cas9 yang sudah track melalui GFP, sehingga nanti ketika si Cas9 ini melekat dengan target binding yang berada di sel kita, maka dia akan menghasilkan fluorescence bahkan sebelum transfeksi, karena kalau biasanya menunggu transfeksi membutuhkan waktu yang cukup lama gitu ya, beberapa hari. Nah, ketika si Cas9 ini sudah bertemu dengan meminimalisir waktu kita untuk melihat apakah transfeksi kita ini berhasil apa enggak. Kemudian juga kita memilih Bapak dan
It seems like there's a trouble from the presenter. We're waiting for presenters perhaps to wrap up the presentation because there's 1 minute left.
Sorry, how long is the presentation for this, Mark?
1 minute left.
1 minute left. Okay.
Oh, I'm sorry, 10 minutes left.
Yeah, so sorry about the connection. I'm going to reshare my screen again. Share screen, yeah. Okay, where were we? Okay. So I'm talking about the CRISPR before. Jadi kita memiliki custom oligos. Selanjutnya untuk produk highlight CRISPR ini sendiri kita memiliki all-in-one CRISPR plasmid, di mana Bapak dan Ibu sudah memiliki vektor plasmid yang di dalamnya sudah terdapat Cas9 dan juga guide RNA. Jadi ini sudah all-in-one, Bapak dan Ibu tidak perlu lagi khawatir untuk satu persatu masukin kemudian mix. Nah, di CRISPR ini sendiri apabila sudah oke nih bagian gRNA dan Cas9-nya, nanti mungkin perlu melakukan transfeksi gitu ya, transduksi. Nah, di sini saya menyarankan extreme gene 36 di sini. Mana extreme gene 36 ini merupakan reagen transfection yang khusus untuk melakukan plasmid DNA dan sangat cocok untuk aplikasi CRISPR atau Cas9 yang akan dilakukan oleh Bapak dan Ibu. Highlight produk ini sendiri adalah performance-nya yang bagus sehingga memiliki higher transfeksi dibandingkan dengan produk sejenisnya, kemudian juga low cytotoxicity, dan yang paling penting itu dia free dari animal derived component, sehingga apabila Bapak dan Ibu ingin melanjutkan ke biopharmaceutical downstream, dia jadi lebih aman untuk menggunakan extreme gene 360 ini. Nah, lanjut masih ke arah discovery yang nyerempet-nyerempet sedikit ke development. Setelah melakukan teknik berupa gene editing, tentunya kita perlu menganalisis. Nah, jadi ini ada beberapa produk analisis yang bisa kita dukung untuk kebutuhan Bapak dan Ibu. Jadi mungkin yang paling gampang kita bisa melakukan immunocytochemistry, yaitu berupa antibody ya, fluorescent antibody. Nah, antibody ini sendiri kita juga ada edisi yang, jadi kalau untuk Zoomec ini sendiri dia merupakan antibody yang bisa tahan di room temperatur gitu, sangat bagus. Nah, kemudian juga memiliki sangat banyak varian gitu di portfolio produknya sendiri. Dan ini merupakan rekombinan dari interspecies gitu ya, tapi bisa men-tailor atau menyesuaikan dengan kebutuhan antibody yang ingin Bapak Ibu gunakan di lab. Nah, selain itu kita juga memiliki life cell imaging reagents gitu. Jadi untuk beberapa reagen yang mendetek perubahan variabel berupa warna, kita memiliki beberapa kit reagensnya. Kemudian juga untuk analisis proteinnya atau antigennya kita bisa menggunakan SMC X-Pro dan juga Milliplex Kits untuk produk-produk Luminex. Jadi kita juga mendeliver produk-produk Luminex. Nah, selain antigen, di sini juga ada sel asik onyx, di mana ini merupakan live imaging dari sel yang biasanya kita gunakan untuk studi disease modeling. Kemudian juga ada beberapa cellular assay dan juga Scepter 3.0. Nah, cellular assay ini berupa kit ready to use, yang di sini MAK kit ready to use khusus untuk beberapa analit yang biasanya digunakan Bapak dan Ibu untuk penyakit tertentu. Jadi misalnya Bapak dan Ibu tertarik untuk sedang melakukan penelitian onkologi. Nah, kami akan dengan senang hati memberikan brosur MAK Kits karena biasanya dalam brosur ini akan berisikan komprehensif analit apa yang biasanya digunakan untuk onkologi gitu, maupun dan juga penyakit lainnya. Jadi di sini hanya gambarannya kecil saja, cuman kita memiliki beberapa penyakit yang sering diteliti oleh peneliti secara globally dan juga di Indonesia. Kemudian bahkan ada juga analit khusus untuk drug discovery ya, seperti NAHDS atau mungkin untuk melihat senyawa metabolit tertentu gitu, bisa dengan menggunakan ingin lebih ke arah protein gitu ya, misalnya melakukan penelitian berupa monoklonal antibody. Di sini memiliki beberapa workflow berupa ekstraksi dan purifikasi, konsentrasi, sampai analisis western blot-nya kita memiliki produknya. Untuk ekstraksi, produk yang saya highlight di sini Celitic kits, di mana Celitic ini ekstraksinya lebih aman, lebih tanpa merusak selnya, sehingga nanti hasilnya akan lebih. Kemudian untuk purifikasinya kita juga memiliki produk concentrate protein. Nah, mungkin yang saya ingin tampilkan di sini adalah Emicon state cell, di mana di sini kita bisa mengkonsentrasikan protein dalam jumlah yang lebih banyak. Dan yang terakhir untuk analisisnya itu ada western blotting ya. Kalau untuk western blotting, tapi kita punya alat berupa Snap ID2. Jadi di sini biasanya western blotting kegiatan Bapak dan Ibu bisa sampai 4-24 jam. Ini bisa disingkat saja dalam waktu 30 menit gitu, karena teknologinya bisa menggunakan vakum. Nah, terakhir untuk gel precast ini kita ada TruePage, di mana dalam untuk TruePage ini sendiri Bapak dan Ibu tidak perlu lagi membuat gel precast sendiri, sudah ready to use dan diklaim 10 kali lebih stronger atau lebih kuat dibandingkan precast gel yang tersedia di market. Nah, selanjutnya juga mungkin ini highlight beberapa teknologi saja gitu yang kita punya. Kita juga punya automated 2D electrophoresis, di mana di sini apabila menggunakan SDS biasa ya, ini mungkin cuma bandnya hanya segini. Dengan menggunakan automated 2D electrophoresis, Bapak dan Ibu bisa melakukan analisis SDSP secara otomatis. Jadi tidak perlu tambah-tambah reagennya secara manual, tapi langsung menggunakan alat. Dan yang terpenting dari sini adalah analisis atau band separation yang biasanya di SDS page itu hanya dalam turun ke bawah gitu dia molecular weight. Kalau di sini dia sudah menggunakan PA dan molecular weight. Jadi seandainya Bapak dan Ibu memiliki protein yang biasanya berada dalam satu band yang sama dengan menggunakan PI atau isiokinetik, nanti dia akan ke arah samping dulu pemisahan separationnya lalu ke arah bawah sehingga nanti untuk protein-protein yang jumlahnya sedikit atau protein yang memiliki band bersama, nanti separationnya akan kelihatan lebih jelas dengan menggunakan automated 2D electrophoresis yang kita punya. Kemudian juga untuk analisis antigen tadi sudah saya sebutkan. Di sini kita bisa ada SMC X Pro. Ini merupakan ultra sensitivity di mana bisa mendeteksi sensitivitas antigen sampai dengan femogram. Kemudian untuk Luminex platformnya sendiri, gitu dia menggunakan Luminex di Taylor di support Luminex dan SMC X Pro. Nah, yang terakhir untuk teknologi yang ingin detection yang ingin saya
It seems like there's a problem with Mrs. Sri Hayuni.
plate-nya iya, sudah waktunya habis ya Mbak ya?
No, you still have 3 minutes left.
Okay,
the
Chairman, thank
connection
you for
is
the
started.
confirmation.
Yes, please proceed.
Okay, thank you. Okay, so the last part I think I was just summarize. Jadi untuk manufacturingnya sendiri kita juga memiliki banyak produk yang bisa mensupport. Jadi biasanya kalau untuk manufacturing sudah masuk ke tim Bioreliance, tapi untuk beberapa reagen kimianya, misalnya untuk vaksin maupun juga biologics lainnya gitu ya, biosimilar lainnya membutuhkan preservasi, kemudian surfactant, adjuvant, antibiotik, stabilizer, dan acidity regulators. Ini merupakan portfolio produk yang kita miliki. Silahkan bila Bapak dan Ibu tertarik bisa mendiskusikannya dengan saya sendiri ataupun tim Merck, area Bapak dan Ibu untuk mendapatkan konsultasi yang lebih lanjut mengenai preservasi dan juga chemicals lainnya yang bisa mendukung produk akhir biologics Bapak Ibu kembangkan di laboratorium. Nah, mungkin sebagai highlighting saya sudah menyebutkan bahwa kita merupakan one step solution. Nah, di sini saya akan sebutkan bahwa produk Merck sendiri itu dapat dilihat dari MQ level. Jadi ini adalah quality segment dari produk kita. Biasanya di setiap produk ketika Bapak Ibu dan login ke Sigma Aldrich, di situ akan dijelaskan berapa quality levelnya. Jadi MQ 100 sampai MQ 600. Nah, MQ 100 adalah komponen yang paling komponen yang paling rendah gitu, karena dia hanya melakukan pengecekan berupa basic quality gitu ya, tapi tetap menggunakan manufacturernya masih dengan menggunakan ISO 9101 gitu, 9001. Nah, ini mungkin cocok, tapi ketika Bapak dan Ibu sudah mulai melanjut ke arah biopharmaceutical atau manufacturingnya, mungkin bisa mengecek produk yang berlabel MQ 500 dan MQ 600. Jadi misalnya Bapak dan Ibu mau menggunakan sodium fosfat gitu. Sodium fosfat ada yang MQ 100, tapi kalau sodium fosfatnya sudah mau ke arah sarankan menggunakan sodium fosfat yang menggunakan MQ 600. Jadi kita memang support memiliki wide variety dari reagen chemicals berdasarkan quality segmentnya sendiri. Nah, terakhir apabila Bapak dan Ibu memiliki ingin cost atau menghemat budget gitu, kita juga memiliki kemampuan untuk bulk packaging. Jadi di sini Bapak dan Ibu bisa membeli packaging atau pre-pack yang lebih besar sehingga nanti cost untuk bahan-bahan kimia yang Bapak Ibu gunakan di biopharmaceutical tersebut di-cut dengan sangat efisien. Silakan menghubungi ke untuk mendapatkan penawaran produk untuk Bapak dan Ibu. Baik, ini saya terakhir hanya masuk ke quiz time gitu. Nah, untuk quiz time sendiri ini akan silakan menjawab pertanyaan tercepat dan mengirimkan emailnya ke saya dengan format nama, institusi, kemudian jawaban gitu. Nah, untuk pertanyaannya sendiri adalah sebutkan salah satu satu saja teknologi detection yang dimiliki Merck yang tadi sebutkan, walaupun dengan presentasi yang terburu-buru karena limitation waktunya. Jadi silakan Bapak dan Ibu bila memiliki jawaban untuk pertanyaan tersebut silakan kirim ke email saya. Nanti di situ untuk 5 orang tercepat akan saya berikan gimmick atau hadiah dari tim kita. Baik, saya rasa itu saja presentasi saya. That's all my presentation. So sorry about rushing the time. I have a technical problem, etc. So yes, thank you very much for your attention and hope to see you again.
Okay, thank you, Mrs. Sri Hayuni, for the presentation. Now, ladies and gentlemen, before continuing to the next agenda session with invited speakers and parallel session, we will have a break for an hour, and we will start the session at 1:30 p.m. Jakarta time. And at the end of the event, there would be announcement for 6 best speakers. So stay tuned and see you after the break. Thank you.
kucing deh.
Ladies and gentlemen, welcome back to ISIBIO 2021. In the following hours, we will have the first parallel session that are divided into breakout room. But before that, there will be the session with the invited speakers. So for room 1, there will be Dr. Puspita Lisdianti. At room 2, there will be Dr. Fir Muhammad Abdul. And at room 3, there will be Professor Yong Soo Kim. Parallel session 1 will start at 2 p.m., parallel session 2 at 2:40 p.m., parallel session 3 at 4 p.m. Jakarta time. And for all presentation of parallel session will be conducted according to the rundown given by the committee. And to enter the room, you could choose the square icon, breakout rooms, and on the toolbar right below and click join of the room that you choose. And now you may enter the breakout room. Thank you.
Hello.
Yes, Mr. Yong Soo Kim.
So I can start now.
You could choose the room that you are assigned to. Professor Yong Soo Kim, you could choose the room 3.
Room 2. So how can? Oh, I see.
Room 3, if I'm
room
not
3?
mistaken. Yes.
Okay, okay, here, room 3. Okay.
Suryadiantina. Hello, once again. Miss Suryadiantina, are you here?
Yes, Mbak Sandy.
Okay. Well, ladies and gentlemen, welcome back. Welcome to the break room, room 1. Now we have the agenda is a parallel session, but before we start, I will guide you for the first session is invited speaker, and the second session is a parallel session. This session will lead by Ms. Surya Diantina, SPM, SCPSC. Now time is yours.
Thank you, Mbak Shirley. Distinguished ladies and gentlemen and all participants, welcome to the afternoon session of this second day. It's a pleasure for me to serve you as a moderator on this room 1. So we will start with our first agenda. For the invited speaker session, please allow me to invite Dr. Puspita Lisdianti with her topic on microbial bioconversion technology to produce oligosaccharide from plant biomass for contribution to circular bioeconomy. Ibu Puspita Lisdianti, are you already here? Okay, so first I will read Ibu Dr. Puspita Lisdianti's curriculum vitae. She worked at National Research and Puspita Lisdianti got her PhD in agriculture chemistry from Tokyo University, Japan, where she also obtained her master and bachelor degree. She was formerly director of Research Center for Biotechnology, LIPI, from 2019 until 2021 period, and has been a researcher at LIPI since 1987. Her specialty is at microbiology. So everyone, I can see Ms. Puspita Lisdianti already here now. Please welcome Ibu Puspita Lisdianti.
Okay.
Ibu Puspita Lisdianti, time is yours.
Yes. Thank you very much, Dr. Surya Diantina. Can you hear me clearly?
Yes, me.
Okay. I would like to share my presentation. Oh yeah. I'm sorry. Again. Could you see my presentation?
Yes.
Yes, we can see, but if you can please put into the slide. So yeah.
Okay.
Yes. That's great.
Okay. Yeah. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Okay. I will leave my masker first.
Voice is too small, too low.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Today, I would like to share with you about the my title of presentation, microbial conversion technology to produce oligosaccharide from plant biomass for contribution to circular bioeconomy. Actually, this research, this topic is accumulation of our research together with Ibu Nani Rahmani from RC Biotechnology LIPI, Dr. Sri Puji Raharti, retirement from research unit of clean technology, LIPI, and then also Dr. Yopi from National Standardization Agency. Okay, the content of my presentation, I have 5 topics. The first is about microbial conversion technology and circular bioeconomy. And then the second, I would like to share with you our research on inulin fructotransferase that obtained from Indonesian microbes, then can convert inulin to fructooligosaccharides. And then the third topic is mannanase, enzyme mannanase that can convert mannan to produce mannan oligosaccharide. And then the fourth is xylinase that convert xylan to xylo-oligosaccharide, and then summary. Okay, let's move to the first topic about microbial bioconversion technology. Microbes is everywhere, and then microbes it's called at cosmopolitan. Yeah, if we found microbes from tropical countries, and then we also can found microbes even in the subtropical countries if the habitat, the nutrition in the habitat is same. And in a single grams of fertile topsoil, about 1 centimeter cubic, may have 3 billion of microbes. Yeah, it include bacteria, actinomycetes, archaea, fungi, protozoa, and others. Microbes also found in a plant biomass, leaf litters, fruit, tubers, and so on. Yeah, their function, yeah, their function in habitats, yeah, in nature as a decomposer, also as a degradation. Also, they produce bioactive compound to keep the health of their habitats. Microbes are using carbon, yeah, for their growth, or microbes need sugar. Sugar, it means 6 carbon, yeah, for their growth. And then if we if we look at the role of biomass, we have abundance of biomass, more than 792 gigatons of carbon in biomass are stored in the world forest and plantation. This is more carbon that in all remaining oil stocks and twice as in the atmosphere. Forest or plantation act as a sink carbon, accumulating carbon as they grow. Because of that, but carbon can be reduced and be finished, yeah. And then now the companies, they applied triple bottom line concept. The triple bottom line, as shown in the right figure, is a sustainable business concept that measuring the value of a company's success using 3 criteria. People, we have to manage the social, yeah, or the people, and then also we need to manage the planet. We have to care about our planet, our environment, and but also of course we for the profit or economy, yeah. And then this is the potential biomass in Indonesia. As a biomass feedstock, the lignocellulosic materials are better for utilization because there are non-edible and no competition problems between food, since there are abundant cellulosic materials from agricultural waste fraction in tropical countries. Yeah. The potential biomass in Indonesia, you can see in the table. Rubber wood, we have rubber wood, logging residues, sawn timber residues, and also palm oil residues, sugar residues, rice residues, coconut residues. We have a lot of abundant of biomass, then still they still not use it, yeah, or still not explore yet. The concept of a key technology for the microbial bioconversion technology, actually there at least there are 5 key technologies. The first, we have to we have to pre-treat the biomass. Biomass contain of a lot of sugar, yeah, contain of a lot of carbon. Biomass hydrolyzing, the second key technology is a biomass hydrolyzing process using enzymatic reaction. And then the third, we have to also understand about the microbial breeding for fermentation process. And the fourth is a bioprocess engineering or fermentation process. And of course, efficient separation technology of the target and integrate and target and product. Integration of these 5 key technology becoming important. The availability of biomass and microbial resources as unlimited resource will support the microbial conversion technology, which will be an issue to produce various product type from local bioresources. And this product could be more competitive in the future. Yeah, And then, but we have to know a lot about circular bioeconomy. Circular bioeconomy offers a conceptual framework for using renewable natural capital to transform and manage our land, food, health, and industrial system with the goal of achieving sustainable well-being in harmony with nature. Yeah. Circular bioeconomy, we have to manage social, yeah, and then also environment, and of course of the economy. Yeah. Microbial conversion technology is one of the key point to make a circular bioeconomy, because the uniqueness of microorganism, in microorganism, they produce 1,000 of biochemical reaction by enzyme that can run at the same time to convert various kind of biomass as a substrate. Mankind also use this ability to produce several bio-based products. First, in the form of food. Yeah, we know, yeah, tempe, tape, nata, de coco, and also functional food and functional beverage. Later, also in the form of pharmaceutical, fine chemicals, and of course, biofuels. Those microbial conversion, also known as microbial biotransformation, can play a key role in the development of new bio-based product follow biorefinery approach. Functionally, biorefineries integrated with circular bioeconomy will play a guiding role in the context of sustainability. Biomass are potential and renewable feedstock for biorefinery system. Okay, I would like to move to our experience on the production of fructooligosaccharides by using inulin as a biomass, and then we found the enzyme, yeah, inulin fructotransferase from actinomycetes that can convert inulin to fructooligosaccharides. Yeah. The first, we did the screening of actinomycetes from our collection, from INACC collection. We choose 100 isolates, and then we grow the culture in the ISP2 agar, and then we put it, inoculate them in a inulin induction medium with inulin, and then we extract the enzyme. Yeah, the genera of actinobacteria, not only from Streptomyces, but also from rare actinomycetes. And then for the enzyme reaction, in various kind of pH and also various kind of temperature. And then we proceed the reaction, and then the enzyme that we found from actinomycetes can convert inulin, yeah, inulin from dahlia tubers, yeah, that we extracted inulin from dahlia tuber, and then we apply our enzyme, and then the product is dihydrofructoanhydrate. Yeah, this DFA3 is useful for the absorption of calcium that can be used as a probiotic and then for therapeutic in for the osteoporosis. Yeah. We try to produce the DFA3 crystals, yeah, using nonomura, actinomycete in a scale of 30 liter. And then we can get the crude DFA3 about 3 liter and then ready to commercialize. This work already have a granted patent granted. If we try to clone the enzyme, yeah, clone the enzyme, the inulin fructotransferase from nonomura, and then clone to Ecology, and then the activity can increase, and then also can resist to high temperature, high pH, and then also high temperature. Yeah, the enzyme activity of the recombinant inulin fructotransferase is quite high compared to other commercial product. The second one, I would like to show the third topic, the second research, yeah, that I would like to share is about manganese. We screen a lot 500 actinomycetes from Biotechnology culture collection, now become Indonesian culture collection, yeah. And then we found one strain, Kitasatospora, that can produce manganese in a lot of activity, in a high activity. Yeah, we found one strain, Kitasatospora, can produce manganese, and then we produce the enzyme purification and characterization of the enzyme, and then analyze, and then the final product is a mannan oligosaccharide. We use a lot of substrate here, plant biomass, palm kernel cake, or also perang potato that that compose a lot of glucomannan. Yeah, this is the result, yeah, from our paper. Our isolate can make a clear zone, and then can also convert locust bean gum, also ivory nut, also konjac glucomannan, and also can convert porang potato, and then the final product is a mono oligosaccharide. Also, we try to clone the enzyme and then measure the activity, and then the activity still high in the production of mono oligosaccharide from mannan polymers. Yeah. They are stable in various pH, and also have activity, still have activity even in a high temperature. Yeah, that's optimum, also optimum temperature that will be useful in the bioprocess. And then the third result in salinase. Actually, sugarcane, has composed of a lot of salinase. Sugarcane can be used as a bagase, and then we have to treat bagase first to be a powder, and then we screen actinomycetes that can grow on the medium contain sugarcane bagasse only, and then we found some actinomycetes, and we also compare to other to the substrate only, and then we produce we can get the crude enzyme supernatant of silinase. We continue our research, and then we found two kind of new enzyme found from Kitasato that high salinase producer. The name of enzyme is GH10 and GH11. And then after we did some enzymatic hydrolysis, we can find some find silo-oligosaccharide, with low cost. In summary, I would like to indicate that plant biomass composed of various kind of complex polysaccharide. We have to treat the plant biomass first to be useful of polysaccharide like inulin, mannan, silan. Actinomycetes produce enzyme that could convert various kind of biomass to useful oligosaccharide. It means a microbe can convert, can have, can convert the biomass to useful bioproduct. Oligosaccharides are important industrial product with major uses in human and animal nutrition. The discovery of enzyme from actinomycetes that can convert plant biomass to produce fructooligosaccharides, mono-oligosaccharide, silo-oligosaccharide could contribute to circular bioeconomy in the future. Thank you very much.
Thank you, Ibu Puspita. It's very interesting presentation. So all participants will have next 10 minutes discussion. We are invite you to comment or question. You can put your question or comment in the chat columns or directly by press the raise hand button on the reaction icon. So we will have around 10 minutes for discussion. That was very interesting presentation from Ibu Puspita. Yeah. Please, if you have any question, you can put on the chat column. Okay, thank you, Ibu Puspita.
Thank you very much.
Maybe everyone thinks your presentation was very clear, and we can clearly understood about the application of the microbial bioproduction technology for human life and environment. Thank you for your presentation and explanation. So now maybe we can move forward to the next parallel session one.
Okay. Thank you very
We will
much,
have.
Dr. Surya. Yeah, thank you very
You're
much.
most welcome, Ibu Puspita. So we will have three parallel speakers in this session. For the three speakers, it's a request for all presenter to open your video camera during the parallel session. So the first speakers we will have Bapak Rahmal Yusuf and followed by Ibu Wahyuni and Ibu Tiwit Widowati. I will check if all the three speakers are already here in this room. Bapak Ramal Yusuf, are you already here? Can you please confirm?
Yes, I am here.
Okay, nice.
Thank you.
Yeah, so for the first 10 minutes, we will hear Bapak Ramal Yusuf presentation with his topic on application of mold and soil microbes to increase growth and yield of chili pepper. Bapak Ramal Yusuf, time is yours.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, today I'm going to talk about one of our experiments. Is about using molds and microbia on the chili pepper. Let me share the PowerPoint first. So here we go. So the title is Application of Molds and Soil Microbes Increase Growth and Yield of Chili Pepper. So our team is a big thing, I think. Sri Anjar Lasmini, Idam, Salapu Pagio, Nur Hayati, Muhammad Yunus, Flora Pasau, Burhanuddin Haji Nasir, Rosmini, Nur Khasana, Luluk Hayati, and myself, Ramal Yusuf. So on behalf of the team, we would like to thank to the committee who accept our paper. So to begin with the background, chili is one of the horticulture product which have a potential economically. The problem with this plant is they still have low production. Based on the literature, why that's low, the reason is maybe related to the soil fertility or less water, or may relate to the climate. So then we have idea, our team have idea to increase that by using soil molds and soil microbia. For the soil molds, we using one is natural, is rice straw, and another one is synthesized molds. And for soil microbia, we using Azotobacter and Pseudomonas. So that's two microbia we're using in this experiment. So this study, the aim is about to determine the better combination between molds and biofertilizer in increasing the both growth and yield of chili pepper, especially in dry land. So here the treatment, we have 7 treatment, including one control. So that's without any straw. And we have M1, M2, M3 until M6, I think. So we combine between straw mulch and silver black mulch with the Azotobacter or Pseudomonas. So you can see here from M0 to M6, that's a treatment. For the preparation for the microba and mulch, we write detailedly in our paper. So if you like to see the detail, you can read our paper. The idea is just we collect the soil from the field, then we bring to our laboratory to growing that microba, then apply to the plant. The same with the another procedure. For example, for the harvest, we mention detail in our paper. So here the result, if you can see, for the vegetative parameter from plant height, number of leaf, number of branches, and then leaf area, most of the treatments show that using mulch and Azotobacter and Pseudomonas, it's different with the control, but there is, for example, in plant high, with the treatment with black silver mulch, still similar with the control. But for another treatment, like number of leaf, number of productive branch, I think most treatment of straw mulch with additional of Azotobacter and Pseudomonas have the highest result, but some of the result is not different with the another treatment. If you can see from number of branch productivity, for example, straw mulch with the Azotobacter and Pseudomonas have the number of leaves about 18.0 plus minus 0.3, but this is not different with the treatment with using black silver and Azotobacter. Another result show for the time to flowering, number of fruit, and fruit weight, we can see treatment with a mulch, straw mulch, I mean, with the Azotobacter and Pseudomonas have the highest result. For example, the number of fruit, they produce 221.8 plus minus 6.7 fruit, higher with the another treatment, but this is not different with the treatment, for example, straw mulch with Pseudomonas. The same with the parameter for the fruit weight. Straw mulch with Azotobacter and Pseudomonas have the highest fruit weight, 231.1 plus minus 3.0, and some of them is different with another treatment, but totally different with the control. And the last parameter for this talk, I think chlorophyll content, similar with another parameter, treatment with straw mulch with Azotobacter and Pseudomonas have the highest leaf chlorophyll content, and that different with the control, but still similar with another treatment. For example, black silver mulch treatment combined with the Sotobacter and Pseudomonas. They are still similar result. And we come to the conclusion that a combination of straw mulch with the Azotobacter and Pseudomonas at the same time show better result than another treatment on the plant height, number of branch, number of leaf area, number of fruit weight, growth content, and time to flowering. I think that's all my talk. Thank you very much for your attention. If you have any question, you can ask me. Thank you very much.
Thank you, Bapak Ramal Yusuf.
Yes,
For our participants, we will hold your question or comment until the discussion session. So we will have 3 speakers first for this session, and then we can continue the session after all the 3 speakers' presentation. Okay, for the next presentation, we will have Ibu Wahyuni. Ibu Wahyuni will deliver a presentation on expression analysis of candidate structural genes involved in starch biosynthesis pathway of cassava from manihot esculentus storage root. Ibu Wahyuni, please welcome everyone. Please welcome Ibu Wahyuni. Time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good day, ladies and gentlemen. Today I will present our research results entitled Expression Analysis of Candidate Genes Involved in Starch Biosynthetic Pathway of Indonesian Cassava Storage Roots. My name is Wahyuni and I am from Research Center for Biotechnology Group. Cassava represents one of the sources of carbohydrates. The storage root contains 65 to 90% starch. The application of starch covers a diverse product, and these applications are based on the amylose and amylopectin ratio. Moving back a bit, starch molecules comprise of amylose, a linear glucose molecule, while an amylopectin is a multi-branched glucose molecule. Starch is biosynthesized from sucrose. Then by a series of enzymes, the sucrose is changed into amylose and amylopectin. The aim of this research is to evaluate the amylose content and gene expression of starch genes in Indonesian cassava cultivars. Prime materials for this research are 9-month-old storage roots from 5 cassava cultivars, the replica RP1, Adira4, Manti, Singkong Tali, and Kristal Merah. The storage roots were filled and deep, and then they are extracted for their starch and for the total RNA extraction. The total RNA then was subjected to cDNA synthesis and qRT-PCR. The amylose content of cassava is around 22 to 25% of amylose. Amylose content in Kristal Merah is categorized as high amylose, since the native amylose content in cassava is around 20 to 24%. And one of the cultivars with 27% of amylose has already been classified as high amylose as reported in Morante, which is screened a natural high amylose cassava oils. Before we profile the starch gene expression, we select 3 housekeeping genes we choose to normalize the expression of target genes. We use these genes, elongation factor 1 alpha, beta tubulin, and zinc finger protein. The CP values of 3 genes found in a stable expression in all cultivars. However, the range of CT value of the singular protein was the closest range as those observed in CT values of gene targets. Therefore, we use zinc finger protein as the reference gene for normalizing the expression of gene targets. It's the expression of 8 genes involved in starch biosynthesis that we organize them according to the pathway described by Fatima et al. The first gene was sucrose synthase. It catalyzed sucrose into ADPG, which are the precursors for amylopectin and then amylase. However, we could not obtain the amplification effect, so then we just split the start the sucrose synthase for the long. Two of GBSS genes, the granule-bound starch synthase, were expressed in different profiles. This gene enhanced the formation of amylose from ADPG. GBSS2 was less expressed compared to GBSS1. This may suggest that there was an accumulation of amylose in storage roots of cassava. This result is in agreement with previous report, and then the report indicated that GBSS1 is the key gene in amylose accumulation in storage root. Meanwhile, the GBSS2 are the genes that regulate the amylose accumulation in stem and leaves. The next gene is starch synthase gene, and we detect the starch synthase gene higher levels in crystal merah and singkong biru, while in Adira4, Manti, and Rapita RP1 is gene at low level. However, we cannot correlate this profile genes. This gene we studied here is one of the 8 primary genes of starch synthase. Probably this gene is not the causal genes involved in biosynthesis of starch in plant. And further exploration of other family genes of starch synthase are needed in the future. The next gene is starch branching enzyme. We include 3 genes, the SBA21, SBA22, and SBA3. These genes were highly expressed in all cultivars. However, in singkong Thai, the expression of these genes were the lowest. The SBA21 also the lowest expression in crystal merah. And then this compared to the 3 cultivars, Adira4, Manti, and Revita RP1. The low level of SBA21 expression may be caused the limiting factor for amylopectin biosynthesis in crystal merah. Thus, gives extra flux to the amylose accumulation rather than the amylopectin accumulation. ABA enzyme is responsible for the branching amylopectin. This is the further genes encoding enzyme for amylopectin accumulation. ABA expression were lower than the other gene expression, and the maximum levels of this gene the maximum expression level was around 0.5, which was detected in Adira4 and more or less in Revita RP1. The low expression of this gene may correspond with storage root initiation as reported previously. So it was reported that GPA gene was expressed highly at storage root development. The last gene is glucan water dekinase. This gene was expressed highly in Singkong Valley than other cultivars. This gene has been reported as an ortholog gene of cassava storage root millet. This gene regulates plant retardation in both physical and storage root food. So this may one factors explaining the small size of Singkong Valley storage roots compared to the other cultivars. The storage root retardation growth is marked by the visible vascular cambium ring in the middle of the storage root of Singkong Valley compared to the other cultivars. So the conclusion of our results that the GBS-S1 was relatively expressed in all cultivars, suggesting the biosynthesis of starch or amylose occurred in all cultivars. Low expression of SPA21 may limit the amylopectin accumulation and associated with the amylose accumulation is in crystalline. More storage root in Singkong Valley may associate with glucan water dekinase 1 expression, which may cause a delay in storage root development. This study gives an insight of amylose content and starch biosynthetic gene expression profile, and further research on spatial and temporal distribution of starch accumulation in multi-organ Indonesia cassava plant should be considered. And we would like to say thank you for your attention.
Ibu Yunis, very interesting results. And at last presenter, please allow me to welcome Ibu Tiwit Widowati with her presentation on bioproduction of indole acetic acid by endophytic bacteria of Bacillus strains isolated from chili and its potential for supporting the growth of chili seedling. Please welcome Ibu Tiwit.
Good morning, ladies and gentlemen. Thank you for the opportunity. I'm Tiwit Widowati from Research Center for Biotechnology, National Research and Innovation Agency. Today, I will present my research entitled Bioproduction of Indole acetic acid by endophytic bacteria of Bacillus strain isolated from chili or capsicum annuum and its potential for supporting the growth of chili seedling. As introduction, endophytic microorganisms are communities that live and colonize the plant interior without causing disease symptoms. The microorganism communities establish a mutualism interaction, enhancing the host plant nutrient uptake and inducing plant resistance for biotic and abiotic stress. Endophytic bacteria synthesize plant hormone such as indole acetic acid, indobutyric acid, gibberellin, cytokinin, and abscisic acid that support growth and root development and absorption of nutrient, water, and mineral. Plant growth promoting endophytic bacteria has been found to colonize different plants including crops, horticultural, aromatic, and medicinal plants. Chili is one of the horticultural crops that have high nutrition and economic value for export and domestic consumption. This study aims to investigate the IAA hormone production by the endophytic bacteria, especially Bacillus strain, isolated from chili, and determine the effect of endophytic bacteria, Bacillus, on the growth of chili seedling. Isolation of endophytic bacteria using plant-based method. The sample used are fruits, leaf, stem, and roots of chili. After surface sterilization, the sample was sliced into two sections and placed aseptically over nutrient agar containing nystatin. screening for IAA production using colorimetric assay. The isolate with IAA production more than 1 microgram per milli were molecularly identified. DNA verification was done using the colony PCR method. The isolate that identify belonging genus Bacillus studied to identify the optimal condition for IAA production. The IAA production was studied by cultivating bacterial culture in nutrient-broad medium at different pH range from 4 until 8 and different concentration of tryptophan 0.5, 1.0, 1.5, 2.0, and 2.5 nanomolar. For the effect of endophytic inoculation of chili seedling, the seeds of chili were soaked in 10 ml of Bacillus suspension and then germinated into trays for 14 days. After that, the chili seedlings were soaked in 20 milliliter of Bacillus inoculant for 30 minutes and removed into polytube containing soil and organic manure. The Bacillus suspension was inoculated again 2 weeks after planting. Result and discussion. A total of 15 endophytic bacteria were isolated from the chili plants and screened for IAA production. The estimation of IAA production indicate that the 15 endophytic bacteria could produce IAA on the culture media supplemented with tryptophan. The amount of IAA produced in this study in range 0.18 until 1.37 microgram per milli. For isolate that is CB6, CB7, CB8, and CB11 show IA production more than 1 microgram per milli, and then selected for molecularly identification. The molecular analysis show one strain belong to the genus Pseudomonas and 3 strains belong to the genus Bacillus. Effect of tryptophan concentration show that the IA production of Bacillus subtilis CB11 increase at concentration 2.5 nanomolar. The IA production of Bacillus subtilis CB6 and CB11 reached a maximum production at tryptophan concentration 2.0 and decreased at concentration 2.5. Effect of pH on IAA concentration. In this study showed that no IAA production at pH 4, and then Bacillus subtilis CB11 and CB6 reached the maximum of IA production at pH 6. Bacillus subtilis Bacillus altitudinis CB8 reached the maximum IA production at pH 7. The IA production of isolate was decreased at pH 8. The three bacillus isolates CB6, 8, and 11 were selected for further assay to study their ability to promote the growth of chili seedling. The application of three bacillus strains will significantly enhance the growth of chili seedling. The inoculation of Bacillus subtilis CB11 showed the highest response on shoot length of seedling. The inoculation of Bacillus subtilis CB11 and Bacillus altitudinus CB6 affected to the root length of the seedling. As conclusion, 15 endophytic bacteria were isolated from the chili plant and they were screened for IA hormone production. 4 endophytic bacteria with IA production more than 1 microgram per milli were selected and molecularly identified. One identified as the genus Pseudomonas and the strain belonged to the genus Bacillus. The optimization of culture condition to maximize the IA production for the three Bacillus strain was indicated at pH 6 and 7 and tryptophan concentration at 2.0 and 2.5 nanomolar. The inoculation of Bacillus strain to chili seedling show increase in the shoot and root length of the seedling. That's my presentation. Thank you.
Thank you, Ibu Tiwi. And now we can start our discussion session. Please, all participants, if you have any comment or questions, I would encourage you to press the reaction button. So raise your hand, please, and then we can have an interaction discussion this afternoon. Otherwise, you can also put your question or comments in the chat column if you wish. Okay, now I see Ibu Siti Nur Rohma. Yes, Ibu Siti Nur Rohma. Please, you can deliver your comments or question.
Okay, thank you, moderator. I would like to ask to Ms. Ibu Wahyuni about slide before conclusion. Please explain this graph in the right side. Why this graph is in negative? Okay.
Okay, Bu Wahyuni, you can directly answer.
Yes. Should I share the screen or how is it the moderator? Can I share the screen?
Yes, that will be better if you can share the screen.
Okay. Yes. So this is the expression of genes. The minimum value is so this is the scale is log to scale. In log to scale, I use log to scale because to see the differences. If in the expression or in the in normal expression value, you cannot see very clearly the differences between the highly expressed and the low expression value of the genes. So this is just the matter of the presenting the data. If you want to put it in a kind of normal expression, you can only see small pictures or small bars here explaining that for example, the DBA gene expression was the lower compared to DBA gene in ADIRA4. I think that's my question. Ibu Siti Nurrohmah, do you have any question?
Ibu Siti Nurrohmah, are you happy with Ibu Wahyuni
Okay.
explanation?
Okay, I understand this explanation, but I still confused about this graph. Maybe why this this high standard error, or why this graph is in the under zero.
Oh,
So
yeah.
so
Well,
that's why this.
yeah. For presenting the data, you can choose if you want to use the relative gene expression, either the value in a kind of, for example, times 100 times or 200 times the city values, the differences of the city values. But I choose in log to scale just to see the differences between the high expression and the low expression. It's easier to see or to to conclude the expression, the gene expression. It didn't mean that the expression, the gene expression is in minus value, no, but it's just to put the scale. Maybe later we can discuss it about the presentation of the genes expression.
Thank you, Bu Wahyuni.
Yeah.
Yeah. Maybe if I'm not mistaken, Ibu Wahyuni would like to say that graph, maybe the purpose of the graph is just to compare between the gene expression. Is that correct, Ibu Wahyuni?
Yes, that's true. That's true.
Okay. Okay, Ibu Siti Nurrohmah, you are happy with her explanation?
Yes, I'm happy. Okay, thank you.
Thank you, Ibu Siti Nurrohmah and Ibu Wahyuni. And next, if other participants, if you have another comment or question, please you can post the reaction button. Raise your hand. Okay, I see here question from Bapak Riki Rumihat for Dr. Ramal. Are there any pre-treatment before using in the plant? Bapak Ramal, the question is, do you have any pre-treatment for the straw
Yeah,
moss?
hello.
Yeah, okay. Please, Bapak Ramal, you can directly answer.
Thank you, Pa, for the question. I think the only treatment we use is species for the straw. So just we cutting in order to make easy when we put in the pot. So that's the only treatment. There is no another one. Thank you.
Thank you, Pak Ramal. So all the pre-treatment are implemented to the all treatments. I mean, the pre-treatment is implemented to all treatments you have, right? Is that correct, Bapak Ramal?
Yes. Unless for the control, we don't put any straw on it. Only plain soil.
Okay. Okay. Thank you for explanation.
Yeah. So yeah. You're welcome. Sorry, my
Okay.
internet's not very good. Yeah.
Yeah. And participants, we still have 2 minutes left. If you have any other question, otherwise maybe we can finish this session. Thank you very much for your attention, and thank you for all presenters. Very nice to meet you all, and we will have 5-minute presentation from PT Wadia Prima Mulia and ITS after this session. I will return to the organizer. Thank you so much for your attention.
Okay, thank you very much, Ms. Surya Diantina. Now, before we moving to parallel session 2, we have a documentary video from our partner. Check it out.
Martin Chris, freeze drying system untuk penelitian, pilot project, hingga manufacturing produk Anda. Hubungi PT ITS Science Indonesia untuk informasi lebih lanjut. Plant Genetic Engineering and Breeding Workflow, supported by ITS Science Indonesia. Plant genetic engineering can be done in many ways. One of them can be done as follows. First, growing bacteria for transferring the targeted genes into the plant. By using incubator farm member, you can make sure the accuracy and stability of the optimum condition for bacteria to grow with simple user-friendly control button just like this. Second, DNA extraction and isolation. With nanodrop from brand Thermoscientific, you can be assured the extracted DNA is free from contamination such as phenols and others. With feature called Ecology, we can count the concentration of extracted DNA easily without hassle. Super easy. Third, cloning and designing genes. Targeted genes then cloned with real-time PCR, LightCycler 96 from Roche. We can monitoring the amplification of targeted genes directly. LightCycler 96 has 4 different channels and can differentiate 4 different targeted genes simultaneously. and then we can separate the applicants with electrophoresis. The targeted genes on agarose gel can be documented very well with gel dot ingenious from Syngene. This gel dot can capture fluorescence signal from dye DNA band, and then from there we could verify the targeted gene and splice the gene into plasmid.
Step 4. Transformation. Plasmid then are inserted into bacteria that we grow earlier. These bacteria will infect plant cells and insert recombinant plasmids into plants. Transformed cells then will be detected with flow cytometry. Meet Aurora from Cytec. This flow cytometry can help you to identify cells that are successfully transformed. Transformed cells with marker will be shown and counted by the software. Step 5. Cell and tissue culture. Transformed plant cells then are cultured into agar in petri dish. The growing transformed plant cell can be captured and documented with cell imaging and multimode reader. Citation 5 from Biotech.
Equipped with reliable microscopy module, Citation 5 can offer detection in fluorescence, bright field, HNA, and phase contrast applications. Citation 5 also equipped with magnificent lens up to 60 times. With Gen 5 software, image processing and analyzing are never been this powerful.
We can also measure the optimum parameters for the transformed plant.
Step 6. Plant breeding. Do not let your precious transformed plants die after all those steps above. Meet Conferent, a reliable climatic chamber for important applications such as growing tall plants, short plants, tissue culture, and incubation. With easy-to-use control panel and many other important features such as broad-spectrum LEDs and dimmable light. Alright, those are 6 steps of plant genetic engineering and breeding that ITS Science Indonesia can support with. Thank you for watching. Contact us on our socials for more info. Stay safe.
Halo, di video ini saya mau pamer alat lab canggih. Lihat deh, ini Magna Pure 96 dari Roche. Bisa buat bantu ekstraksi RNA virus COVID-19 secara otomatis. Sebanyak 96 sampel dalam waktu 2 jam. Setelah diekstraksi, sampel dapat diuji dengan metode real-time PCR menggunakan Light Cycler 96 ini. Hasilnya langsung terlihat positif atau negatif. Cepet kan? Hubungi teman-teman di ITS untuk info lebih lanjut. Bye-bye, stay safe.
Okay, welcome back in this parallel session 2. Before we start, I will invite Dr. Wahyuni. Are you ready?
Yes, Mbak Sandy.
Okay.
Yeah.
Okay, now time is yours.
Okay, thank you. Thank you, the organizing committee. Good afternoon, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. My name is Wahyuni and I will be moderating this session. This is the parallel session, the second parallel session in room 1 in this afternoon. We have 5 speakers in this session. The presenter 1 is Dr. Siti Nur Rahma from Research Center for Biotechnology, BRAIN. I can see already Dr. Siti Nur Rahma there. The second is Ms. Tri Handayani. I hope Ms. Tri Handayani also already opened her camera, her video. And then the third one is Mr. Rudianto from Research Center for Biotechnology, BRAIN. Okay, I can see already Mr. Rudianto, thank you. The fourth is Professor Dr. Endantri Margawati from Research Center for Biotechnology. Yes, I can already see Bu Endang. Thank you, Bu Endang. And the last presenter is Ms. Slametia Volkandari from Research Center Biotechnology BRAIN. Okay, before we begin, I have a few notes. The questions and answer session will be collected or open after all presenters deliver their presentation. And questions can be submitted via the chat feature during the presentation. And then you can also ask the questions lively at the end in the Q&A session by raising hand using the reaction box. Okay, without further ado, now let's please welcome the first presenter, Dr. Siti Nurrohma. Dr. Siti Nurrohma will present in vitro tissue culture of Artocarpus altilis or breadfruit. Okay, Dr. Siti, time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hello everybody. I would like to give presentation on my study about the effect of enzymidyl and sucrose concentration on growth of breadfruit shoot culture for in vitro preservation. Breadfruit is the member of family Moraceae that grows in tropical region of the Southeast Asian, including Indonesia and also in Pacific Island. This plant is one of the economically important trees, which is being used for food, medicine, timber, gum, dye, for textile and paper. The fruit has a high content of carbohydrate as an alternative for staple food. A research collaborative between LIPI and this young Chinese medical university has developed herbal medicine of breadfruit leaves, which are containing flavonoid and phytosterol that potential for treating cardiovascular disease. Conventionally, breadfruit is propagated by root or stem cutting. The shortage of this method is limited mass production and low quality of seedling. In vitro propagation of breadfruit has been reported in relation with a manipulation of nutrient composition. Climate change has aggravated the severity of environmental conditions, such as an extreme increase of annual precipitation that lead to soil flooding and drought as well, which is contributing to erosion of many plant species, including breadfruit germplasm. In vitro preservation offers several advantages, such as a very high system, reduction of space requirement, lower expenses of labor costs, and also prevention of genetic erosion. So, application to ex situ conservation through in vitro technique are required to support them. Material and method. I use in vitro shoot tip of breadfruit cultivar boni. I use a basal MS medium with a 5-level concentration and 3 level of sucrose. Our culture were incubated in a growth room, and data were recorded start from 4 to 20 weeks of cultivation. The parameter observed, including shoot length, leaf number, shoot number, and root number, and also after 28 weeks, length, diameter, number of shoot, length, and with leaves, and number of root were also recorded. This is the result. As we can see that in picture A, soot length decrease significantly as the concentration of antimidal increase. Using half strength or 1.5 fold greater of sucrose of a normal concentration of sucrose inhibit of soot length. The increase of antimidal was efficient in reducing of development of leaf, except for plantlet when the ground at the normal concentration led to reducing a leaf number. In the use of half strength or 1.5 fold greater lead to reducing of leaf number. The use of antimidal enhanced the number of soot. The maximum number of soot was obtained on the medium containing 1.5 mg per liter antimidal. Conversely, using half strength or 1.5 fold greater of normal concentration of sucrose led to reducing number of soot. The results show that low concentration of antimidal 0.75 until 1.5 mg per liter enhanced root formation. However, the use high concentration 3 until 6 mg per liter suppress root formation. In the right side, we can see that the soot culture in half strength concentration of sucrose were rootless. Table 1, we can say that the results show that increasing antimidal concentration suppress growth of soot length and also soot diameter. The concentration, the best maximum of number of soot was obtained at the 1.5 mg per liter antimidal. The use of half strength or 1.5 fold greater than normal concentration of sucrose reduced length of number of soot. Conversely, increasing sucrose concentration to 45 kg per liter significantly promotes soot diameter. This progressive decline in length and width of leaf occurred with increasing concentration of antimidal added in the MS medium. Effect of sucrose concentration at 30 until 45 gram per liter added in MS medium showed a significant difference in length and width of leaf compared with the half strength of a normal concentration of sucrose. The reduction of leaf number was greater in the presence of higher concentration. This performance of a breadfruit planet culture for 28 weeks in the left side is antimidol, in the right side uses sucrose. A conclusion, antimidol and sucrose could be used for in vitro preservation of breadfruit for 7 months. Antimidol at 1.5 mg per liter and 45 gram per liter added in the MS medium was the best concentration of breadfruit growth. In vitro preservation contributed to supporting existing collection as well as strengthening and expanding germplasm resources. Thank you for watching and listening.
Okay, thank you, Dr. Siti Nur Rahma, for the presentation. Now we move on to the next presenter is Ms. Trihandayani. Ms. Trihandayani will present about in vitro somatic embryogenesis of shallot. Please, Ms. Trihandayani, you can start the presentation.
Thank you for the opportunity and the time. My name is Triandayani. I'm from Research Center for Biology. In this seminar, I would like to presentation our research with the title In Vitro Somatic Embryogenesis from Thin Cell Layer Explant of Shallot. As we know, the shallot is the second major horticulture crop after pepper, and the largest production area of shallot is in Java. The current cultivation of shallot usually use safe bulk from the previous season, but the system has problem can transfer disease in the next season and reducing the potential yield. So the alternative, farmer can use TSS or fish or true seed shallot as planting material that free from the disease. However, the TSS system is unsuitable for non-sensitive flowering genotype, so we need to develop alternative propagation method that we can use somatic embryogenesis. Somatic embryogenesis is usually used to mass micropropagation and the one source of plant material used for genetic improvement, example for mutation breeding or somatic hybridization. Somatic embryogenesis in shallot and other olive species has been reported using various explants derived from bulbs, seed, flower bud, and from anther. Alternative source of explant we can using TCL or thin cell layer, which consists of few layers of cells, typically 0.1 to 2 mm in thick. And the use of TCL explant for micropropagation of shallot was reported and indicated that TCL was more efficient in producing plantlet than shoot-tip explant. So this research was intended to develop a simple protocol for inducing somatic embryogenesis from thin cell layer explant of shallot, including initiation of aseptic culture, embryonic callus induction and proliferation, maturation and regeneration of somatic embryo. In this research, we use healthy bulb from cultivar Bima Brebes and Rubaru Sumeneb. We have 4 different states of experiment. First is initiation of aseptic culture. The second is induction of embryonic callus. The third is proliferation of embryonic callus, and the last is maturation and regeneration of somatic embryo. It's of experiment. We arrange in complete randomized design with 4 to 6 replication. Some of observation data that we collected are the percentage of survival explant, percentage explant from callus or shoot, callus diameter, percentage of callus proliferation, and morphology of callus, and also percentage occurrence of embryonic callus that from globular embryo or mature embryo, and the occurrence of hyperhidric embryo. All data were evaluated using analysis of variance, and there are significant difference between means we compare using the MST. As preparation of TCL explant, the healthy bulb, we discard the dry outer layer, then washing using tap water. Then we soak the bulb in fungicide and bactericide solution. In laminar airflow, we conduct surface sterilization using ethanol, Clorox, and sterile water. Then we excise the outer layer and depart on the bulb. and we cut the tip part bulb and leaving the basal plate is about 0.5 to 1 centimeter in thick. And then we cut transversely into thin slice for about 0.5 to 2 millimeter in thickness. The slice section we use as TCL explant. For the flow of experiment, the TCL explant we inoculated in antibiotic containing medium for 5 to 7 days. Then we transfer to free antibiotic medium for another 2 until 3 weeks. The uncontaminated explant we inoculated in induction callus medium for about 6 to 8 weeks of culture. Then we transfer or subculture to proliferation callus medium for another 8 weeks. In proliferation medium, the callus that we that develop, we subculture in every 6 to 8 weeks in the fresh medium with same composition, or we can transfer to the medium for maturation and regeneration of somatic embryo. This is our result in initiation of aseptic culture when we use the bulk from the source of explant from underground usually incur a significant obstacle with high level of contamination that caused by bacteria and fungi. And to reduce contamination, the TCL explant we inoculated in media containing antibiotic, then we subculture to media free antibiotic to minimize the detrimental effect of antibiotic on the explant tissue. In initiation of aseptic culture, we compare to basal media MS and BDS with from Bima Brebes cultivar and Rubaru Semenep, and the ratio so that there is no different response in parameter of explant survival with no contaminant. With the rate of survival explant is about 40 to 60% in medium supplemented with antibiotic, and when the explant transferred to medium free antibiotic, the survival rate is about 60 to 80%. The TCL explant that culture in medium free antibiotic, some starting to forming a shoot that emerge from the basal explant. In induction of embryonic callus, we compare 6 different growth regulator treatment that consist of 2,4-D and combined with naphthalene acetic acid. And from the result, we have that a higher percentage of callus induction was obtained from the culture in the medium containing 2,4-D 1 milligram per liter combined with NAA 2 milligram per liter with no different response between cultivar. For callus induction in early formation of callus, we can see that callus start to division and the callus observe watery with transparent morphology, some starting for nodule and friable callus. In proliferation of embryonic callus, we compare for a medium that consists combination composition of 2,4-D, naphthalene acetic acid, and methyladenine. From the table 2, we can see that from some parameter that we observation, there is no different response between composition of growth regulator in proliferation the callus. But the data analysis based on the inoculum origin regarding the induction medium treatment so that the concentration of induction medium still affected the subsequent callus proliferation on the proliferation medium, and the concentration 2,4-D 1 milligram per liter seem optimal for embryonic callus formation and proliferation. In proliferation and embryonic callus, we can see callus with morphology friable with nodular structure, or callus with compact with nodular structure, or greenish friable callus with granular like structure. In maturation and regeneration of somatic embryo, we evaluated the concentration of sucrose and gel rat, and the results show that increasing gel rat concentration can increase the development of globular somatic embryo and decrease the hyperhidric somatic embryo. In development of somatic embryo, we can see that early formation we can find embryo with globular structure with the color yellowish to greenish, and some embryo show transparent and the other show opaque embryo. Embryo will develop to mature somatic embryo with we can see the coleoptile-like structure. The embryo somatic with coleoptile-like structure, we can see that some of them are transparent and some of them are opaque. But the somatic embryo that developed to normal embryo, normal plant is came from the somatic embryo that has opaque morphology. So this is our conclusion. We have developed a simple and efficient protocol for inducing somatic embryogenesis of salad, cultivar Mima Brebes, and Rugarusa Nanab from TCL as the explant. Thank you for your attention.
Okay. Okay. Thank you, Ms. Tri, for the presentation. It's very detailed. Sorry, my microphone is muted. Okay, thank you, Ms. Tri, for the presentation. The presentation was in detail. Now we continue the third speaker. The third speaker is Mr. Rudianto from Research Center for Biotechnology, PRIN. He will present the in vitro tissue culture of Moringa oleifera. Mr. Rudianto, the time is yours.
Thank you very much. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh. My name is Rudianto. I'm from Research Center for Biotechnology, National Research and Innovation Agency, or BRIN, Indonesia. Today, I would like to share with you one of my topic research titled In vitro shoot growth and root formation enhancement of Moringa oleifera leaf on DKW medium containing cytokinin and auxin. Before I jump into my main research, I would like to give you a brief introduction. As we know that Moringa oleifera belonging to Moringaceae family. This plant origin is from Himalayan mountain of southwest India. This plant has a lot of nutrition content such as vitamin and calcium, and a lot of secondary metabolites such as saponin, flavonoid, arginine, and glutamic acid. They are an obstacle to scaling up this seedling production because the conventional propagation of Moringa oleifera is very susceptible to disease, such as fungi, bacteria, and viruses. A limited number of explant for cutting and seed viability. Propagation through plant tissue culture can be applied to produce uniform seed, free from pest and disease, more stable, and applied quickly. One of the essential factors in plant tissue culture, especially in the proliferation state, is adding plant growth regulators such as cytokinin and auxin into the culture medium. So the aim of this research is to enhance the growth of shoot culture of Moringa oleifera and its root formation on the basic DKW medium, supplemented with different types of cytokinin and auxin. In methodology, we use a different type of cytokinin such as BIP, kinetin, IP, and zeatin, and different type of auxin such as IAA, NAA, IBA in combination with different concentration. These two types of plant growth regulator and then mixed to the DKW basic medium and then supplemented with 30 grams per liter of sucrose. The medium pH and then adjust to 5.8 and then solidified with 3 grams per liter of agar. Okay. After that, we sterilize the medium using autoclave at 121 degrees of Celsius for 20 minutes. After we make a medium, and then the plantlet leaf were cultured according to the treatment test, and then the culture were incubated at 24 plus minus 2 degrees of Celsius with continuous photoperiod with light intensity of 500 to 900 lux. This is the experimental design and data analysis that we use. We use completely random design with two factors. Factor number one is different type of cytokinin and auxin, and the second one is different concentration. The data analysis, we use ANOVA to determine effect between treatment, and then the significantly different variable were tested using Duncan multivariate test. Its treatment consists of 12 replicate, and then the variable observed were shoot height, number of shoot, number of petiole, and number of root. This is the result of our research. You can see that the different type of cytokinin give very significant effect on shoot height, number of petiole, and number of root. And different concentration also give a very significant and at all parameter observed. There is a interaction between cytokinin and different concentration on number of shoot, number of petiole, and a number of root. But there is no interaction between cytokinin and concentration on shoot height parameter. This is table 2. You can see that the best medium and the best treatment to enhance the Moringa alifera growth was produced in kinetin with 1 milligram per liter concentration. You can see that this treatment gave the highest shoot height, number of shoot, and number of petiole. And the lowest result was produced in BAP in 1 and 2 milligram per liter. So we can conclude that the best type of cytokinin is that's suitable to enhance the growth of Moringa alifera is kinetin with 1 milligram per liter concentration. This is the performance of Moringa alifera at 6 weeks after culture. You can see that in 1 milligram per liter of kinetin, the plantlet of Moringa looks more vigorous with greener leaf and figure stem and petiole, and significantly different with other treatment. This is the ANOVA of shoot height, number of shoot, number of petiole, and number of root on Moringa alifera using different type of auxin. You can see that the different type of auxin give a significant effect on shoot height, number of petiole, and number of root, and also root length. And the concentration also gave a very significant effect on all parameters observed. There is an interaction between auxin and concentration on parameter of shoot height, number of shoot, number of root, and root length. And in number of petiole, there is no interaction between the different type of auxin and concentration. This is table 4. We can see that the best treatment to produce the highest number of root was produced in IAA in 1 milligram per liter, and in control there is a few root formation result. This is figure 2. We can see the performance of Moringa alifera in different type of auxin. We can see that in 1 milligram per liter IAA and 2 milligram per liter IAA, the plantlet produce a lot of root, and in contrast, very few root formation. Produce root formation. So we can conclude our research that the type and level of cytokinin affect shoot height of Moringa alifera culture on KW medium. And the type and level of auxin also affected its root formation. Medium supplemented with 1 milligram per liter kinetin was the best for shoot height, number of shoot, and number of petiole. Auxin IAA at 1 and 2 milligram per liter produced the highest number of root and root length. The selection of plant growth regulator was beneficial to find out the best medium composition for growth and multiplication of Moringa aloe vera shoot, as well as for root formation. The author, I would like to thanks Prisna Chitra Andiani for her assistance in this research, and thank you very much.
Thank you, Mr. Rudianto. It's a lot of data presented today. And then we continue to the fourth presenter, Professor Dr. Endang Trimargawati. She will present the polymorphism determination of the FSCG genes in Indonesian bacteria. Professor Dr. Endang, please, the time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning or good afternoon to everyone and all my colleagues, friends, and all ISIBIO participants. This is my pleasure to deliver part of our research. This is the title, The Polymorphism Determination of the FACT-G, PST1, and FACT-X HIN1 genes in Indonesian bacteria. Here is our team, myself, Vidya Binta Bayu Putra, Herman Ratsma, Slamet Jahulkandari, and Indriyawati. What is the reason? The background is crossbreeding generally was aimed to improve the productivity of livestock through hybrid vigor heterogeneous effect. The background shifts of Mega, it was 75 merino and 25% carrot has been designed in 1999 for meat production purposes. Despite that purposes, both sheep, merino breeds, and carrot breeds have a potential of prolification traits. The prolification trait, we call it effect genes of sheep, could be selected by a molecular selection through some reproductive genes. The FACT gene of cross differentiation factor 9 and FACT-X bond for morphogenetic protein 15 has been used for the molecular selection to improve reproductive rate in sheep. In sheep, mutation point, there is a mutation point in gene 2019 G to transmission gene G to C in exon 1. While in gene FACT-X, there is a transition at 35 gene of gene C to B T in exon 2. Previous study in carrot sheep was reported showed polymorphic in carrot sheep has a form site in FACT-G exon 2. But there is no report yet in diversity of FACT-G, PST1, and FACT-X genes 1-3 in Indonesian sheep. So what is the purpose? The purpose is to determine the polymorphism of FACT-G and FACT-X genes in bacteria sheep using PCR RFLP technique. The material method, animal and DNA extraction, we use it to 75 megabacteria sheep. And DNA was collected from fresh blood, from jugular vein, using phenoject container containing ADDA. DNA amplification in total volume 10 microliter, containing 1 microliter of DNA template, 0.2 microliter of primers, 5 microliter of PCR master mix, and 3.6 microliter of free nucleus water. Here is the primer we are using on the top for the FACT-G gene and the bottom for the FACT-X gene. Amplification was conducted using a master cycler gradient from Eppendorf, Germany. This program was breed in saturation 95 degrees for 2 minutes, penetration 95 degrees for 30 seconds, and annealing at 656.6 degrees for 1 minute 30 seconds, extension 72% at 2 degrees and for 30 seconds, and final extension 72 degrees for 2 minutes. Visualization was conducted using GPOX documentation system. Genotyping, RFLP analysis of post-jet were conducted using a 10 microliter in volume 10 microliter containing 2 microliter PCR product, 0.2 microliter of resin enzyme in SP for FAG and hint 1 for the FAG-X, 1 microliter buffer and 6.8 microliter of PDW. Incubation was conducted at 37 degrees in water bath for 60 minutes. Visualization was conducted using GPOX documentation system. Statistical analysis we conducted for the genotypic and allelic frequency, number of effective allele, polymorphic informative content of PIC, and G-square test for Hardy-Weinberg equilibrium. Result and discussion. This figure showed the forward and reverse primers with the bold character, and then the top was FAG-G and the bottom was FAG-X, while the red color is the restriction side, and the transversion was in 2019 G2BC, and the transition for the FAG-X was 35 C2BT. Here the figure 3, the figure 2 was not exposure, is this containing the band of the gene of FAG-G and FAG-X. It was a 708 and the FAG-X was 141. Here is result of PCR RFLP. On the top was for FAG-G. So length 1 until 12 are polymorphic, which is and length 14, but length 13 are carrier with the genotype FAG plus and FAG minus. And the bottom is for FAG-X. All are monomorphic 10 We show here in the table actually a summary of the figure of number 3. This is we resume the FAG-G with the ST1 90.94 is polymorphic or wild type, and 0.06 is a carrier and no mutant there. And allele, this is we saw this 90.97 and for FAG negative 0.03. And NE 1.05, PIC was 0.05 with Hardy-Weinberg as 0.04, I guess. And the gene FAG-X hint 1 and 100% are monomorphic, so we got the wild type FAG plus and FAG plus genotype as 10%. Here, when we saw here, we can compare with another Egyptian SIP, Saidi and Osimi. this UC fact GPST1, all are monomorphic. This is different from our SIP. Then 10% are monomorphic, while our Pecros is 9.97 and 97% are polymorphic. While in Chinese, you SIP in Chinese, facts X in one significantly association with liter size. This has got the facts plus and facts plus genotype. This is the liter size 2.18 plus minus 0.06, while carrier is 1.66. Conclusion, fact GPST1 and fact X1 gene would not be suitable for molecular selection for megapetros. Future a deep investigation in other regions of fact G and fact X gene are important to be studied to obtain polymorphic site at least moderate PIC value. Then molecular selection for improving reproductive traits in SIP can be assessed. Here is I saw the SIP, Batur SIP, and this is actually for next our research. Thank you very much for your attention and your time.
Okay, thank you, Professor Endang. It's a lot of work, working with molecular markers in our population. Okay, and then we come to the last speaker, Ms. Slamadia Volkandari. She will present genotyping of insulin growth factor 1 gene in Lakor goat. Ms. Dia, please start the presentation.
Assalamualaikum warahmatullahi wabarakatuh. Firstly, I would like to thank for moderator, and then I would like to introduce myself. My name is Slamadia Volkandari. I'm from Research Center for Biotechnology, National Research and Innovation Agency. In this time, I would like to present about our genetic exploration in Indonesian goat with the title is genotyping of the insulin growth factor 1 or IGF-1 at the SNP 5752G2C on the Lakor goat from Southwest Maluku Regency. Introduction. Lakor goat is an indigenous breed of the Indonesian from Southwest Maluku Regency, especially at the Lakor Island. Here is the location. And then the Lakor goat are known to have a good adaptation with the high temperature and the pure feed quality. And the Lakor goat has been established with the decree of the Ministry of the Agriculture 2011 as a local goat from Southwest Maluku Regency. The Lakor goat is one of the protein source for the local community in the Southwest Maluku. Lakor goat is categorized included the large size of the goats, of the body goats, and then if we compare with the other local goat in Indonesia like the Etawa, Kacang, Costa, and Saanen goat. Growth performance in livestock is an important trait, and then the influence with the genetic and environmental factor. Genetically, growth was also influenced by polygenes, more than one genes. One of the gene is the insulin growth factor 1. Insulin growth factor 1 are essential for the normal growth and then the cell growth, development during embryonic stage, and then the tissue differentiation develop during development. So the IGF-1 genes is a good candidate related with the growth rate and then the meat production, milk production of the goat. The IGF-1 is encoded by a single gene located on chromosome 5 or BTA5 in goat.
In IGF-1 gene of the goat, they are the popular SNP. This is a G5752G2C, location in intron 4, and then the produce with the 3 type of the genotype is a GG, GC, and a CC. In previous study in Kecobong goat, a genotype GG had a significantly higher body weight and a body measurement than the genotype CC. In beta goat, the AA, or we can call CC, associated with the low body weight, and then the BB genotype or the GG genotype was significantly associated with the higher body weight. So the aim of this study was to identify the genotypic pattern of the IGF gain at the SNP position G5752G2C in Lakor goat Southwest Maluku Regency. Material and methods. In this study, we use 63 samples of the Lakor goats. This is a male of the Lakor goat and this female. And then we collected from the Lakor Island. This is the Lakor, a part of the Leyte Islands. And then we collect in the 3 villages, the Ketilepe, Berwawan and Zamluli, and Letoda village. And the sample in the study use the follicle hair. For the genotype analysis, we use the 3 steps: DNA extraction using the DNA extraction kit, and then the amplification of the IGF-1 gene targeted fragment using the PCR technique with the pair primer according Liu et al. 2012, and then with this program, PCR program, and then the visualization using the agarose gel 1%. For the genotyping, we use the PCR RFLP method, and with the detection of the SNP position, SNP 5752G2C, we detected using the restriction enzyme HA3. And then the visualization of the result of the RFLP, we use agarose gel 2%. This is the location of the fragment target and the location of the SNP. Result and discussion. Amplification of the DNA fragment containing the SNP, SNP G5752, G2C in the IGF-1 again successfully performs on the 363 base pair, and all of the amplicon were digested with the HA3, and the result we found the one pattern is a BB genotype. This is table we compare our research Lakor goat with the other goats from Indonesia, Kecobong, Gembrong, Samosir, Costa, and the others, and we compare with the others goat from the China, India, Pakistan, Iran, and Egypt. The monomorphic or the low genetic diversity in the Lakor goat, we assume that the geographic or the area and the rearing system influence of the low of the genetic diversity in the Lakor goat. Long distance from the Lakor goat to the big island like the Ambon. Ambon is a big consumption of meat. Causes several problems like the limited marketing access, and then the no market animal too in the Lakor Island. And then about the rearing system. The rearing system of the Lakor goat use the extensive rearing model. The disadvantage of this model is uncontrolled mating. The uncontrol of the mating is a big problem because in this case, increasing of the inbreeding level, and the effect of this inbreeding is the low genetic diversity. This is the extensive rearing model, and this is a good cage of the lacquer goat using the stone and the farmer called Lutur. So the conclusion, all of the lacquer goats from the three locations, the three villages in the lacquer island were detected monomorphic with the BB genotype of the IGF gen SNP 5752G2C. And then suggestion for the increasing of the genetic diversity is a changing of the rearing model, extensive rearing model, to the other semi-extensive or intensive rearing model, and using the mating control with the artificial insemination, and then also government regulation about the animal market. Perhaps with the suggestion, the genetic diversity can improve. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Ms. Dia, for the presentation. Okay, now we come to the question and answer session. I already see in the chat box there are 2 questions. For all the participants, if you want to ask lively, then you can raise your hand using the chat, the reaction box in the Zoom window. First, I would like to read the first question from Rael Rifki Firdian. The question is for Ms. Trihandayani. It was said that to avoid bacteria contamination, the eggplant was cultured on antibiotic media. Then, to deprive negative impact of antibiotic, the culture was transferred to antibiotic-free media. The question is how to determine that the culture was ready to transfer and how long? Ms. Tri, could you answer the question? Please open your mic.
Thank you for the question. Can you hear my voice? Hello?
Yeah.
Can you hear my voice?
Yes, very clearly.
Okay. Thank you for the question, Miss Elok. How we determine when we time we transfer to the new medium? Previously, we tried some experiment. The optimum time that we inoculated eggplant in the medium containing antibiotic, and when we transfer to the medium-free antibiotic. Usually, the time is depend on the size eggplant, type of eggplant, and source of eggplant. And usually for shallot, the eggplant that came from bulbs, we usually inoculated in antibiotic medium for 3 days until a week, until 1 week, and then we transfer to the medium-free antibiotic. This time is we get from our previously experiment. Thank you. Any question or is not clear enough?
Yeah, Miss Ella, do you want to respond?
Yes.
3 answers. Yeah, please.
This is based on my experiment using very much contamination on my culture. So I'm interested in using antibiotic for the medium. But you said that 3 days until 1 week for the explant above the ground. What about the explant under the ground? Or maybe you have some experiment using explant that is already infected from inside. How to manage that we could avoid the contamination. Thank you.
We usually sterilize first in laminar. We usually use to sterilize like Clorox, alcohol, and etc. We can use that. And for internal, usually for if the explant have internal contamination, we should usually inoculated first in the medium that containing antibiotic, or if the explant contain like fungus, we can add to the medium is fungicide like that, so that it can decrease the contamination. For the time, usually it depends. Some we can inoculate it in antibiotic for 1 week, or like if banana shoot from the ground, we usually until 1 month or for 4 weeks. That is depend on the explant that we can use. Thank you.
Okay. What about the fungus? Do you have any recommendation what kind of fungicide to be implemented in the medium?
We usually use fungicide that sell in the market, like Masalgin. It usually yes, it's because the form is in sorry, serbuk bahasa
powder, yeah.
Yeah. So it's usually it's easy to mixture in the medium.
In the medium. Oh,
yeah, like that.
I have read that Masalgin is apa ya? So we just mix the Masalgin into the medium weight like that.
Yes.
Okay.
I mix the Masalgin and antibiotic to several antibiotic. We put in the medium.
Oh.
Okay.
Thank you very much for the
Yeah. Thank you.
I think this is very helpful for me now. I'm now culturing Myristica, and I'm encountering massive contamination, especially fungus. Thank you.
Maybe for the you can also discuss it with Mystery after this session. Okay. Thank you for the
thank you.
questions. Yeah. Okay. Then there are a question for Miss Siti Nurrahma. I hope Miss Siti Nurrahma still join us. Yes, thank you, Miss Siti. It's from the question is from Mr. Handayani, and she asked how long the maximum time that we can culture the breadfruit shoot in medium containing ancidol and sucrose for in vitro preservation, and are there any effect of this chemical used to the growth of plantlet after we acclimatize in environment, ex vitro environment? Thank you.
Okay, thank you for your question, Ms. Trihandayani. I will ask this question. I kept plantlet in ensimidol and sucrose for 7 months, and after that I will recover to a medium MS and growth normal like other without treatment like this. Are you satisfied, Ms. Handayani?
Yes, thank you for the answer. But if we use ISIBIO, there is we can see the morphology to from the shoot if until we finish the culture.
Yes, of course. NC middle is like inhibit for elongation, for differentiation, something like this. This work opposite work with gibberellin biosynthesis. So the function is to inhibit growth for plant. So of course, this the optimum for cap this plant and until 1.5 milligram per liter. More than 1.5 milligram per liter ISIBIO is not recommended for preservation.
Okay. So I think there are still one question in the chat room from Ms. Dia to Professor Endang. I will quick read the question. So the polymorphism was detected, but low in FACG, FACG, and monomorphic in FACX. So not suitable for a marker. Then for the next exploration, what is your strategy to get a marker SNP for the trait in shape cross? Prof. Endang? Please unmute the microphone first, Prof. Maybe the organizing committee.
Okay, thank you. Thank you for the question. Thank you, Bu Yuni. Yes, we has already discussed about this with our team that we for the next research actually we will use for another ship that is Batur ship. There's also with some blood merino, so probably similar. So we may use another gene, also the population we may also to increase. That is our solution for to get the polymorphic. That is probably answer your question. Thank you for the question anyway. For the next homework, our next homework actually. Thank you.
Thank you, Prof. Endang, for the answers. I think if I can have 1 minute more for the two speakers, just to a quick questions and answer. First is for Mr. Rudy. It's just a short question, Mr. Rudy. Why did you choose the KW medium instead of the normal or basal medium in tissue culture such as MS basal medium? Mr. Rudy, are you with me? Are you still join with us?
Okay, thank you, Ms. Wayuni. You hear my voice?
Yes, very clearly. Yeah, very clear.
Okay, why I use DKW? Because in previous experiment, we use also a lot of basal medium such as MS medium, and then WTM, and then niche and Gamborg medium. And we compare between these kind of basal medium, and the result is DKW is the best result to enhance the growth of Moringa oleifera. I hope it give answer to your question.
Yes, it's very clear. Okay, thank you, Mr. Rodi. The last question is for Mbak Dia, Miss Dia. I see that your marker, the IGF, is not how to say it, it's not really represent the diversity of the lacquer goat. So what is the first is, could you relate your finding with your the aim of the experiment? I mean, what's next do you want to do in the future experiment if you could not get the diversity of this marker in the lacquer population?
Okay, thank you, Miss Wahyuni. In this study, we got monomorphic, and then the next step, we will exploration about the inbreeding level in the goat, in the lacquer goat, because the previous with our team, we explored in the mitochondria DNA and the Y chromosome, the genetic diversity is low. So the next step, we will evaluation about the inbreeding level using the microsatellite analysis. After that, we will get the conclusion for the next step to introducing the others breed or the others male from the others location to improve the genetic diversity in the lacquer goat. So it is my answer.
Okay, thank you, Ms. Dia, for the answers. Okay, ladies and gentlemen, this is our session. We finished our session. Thank you for the speakers and for the participants for the contribution of this session. Then we can just put the class for the presenters, and then I will now turn the session to the organizing committee. Okay, Mbak Stanley, Miss Stanley, thank you.
Okay, thank you very much, Ms. Wahyuni. Now we have around 4 minutes left to the next session. So we have a short break. See you later.
Coffee time. Coffee time. Coffee time.
Martin Chris, freeze drying system untuk penelitian, pilot project, hingga manufacturing produk Anda. Hubungi PT ITS Science Indonesia untuk informasi lebih lanjut.
Microtomy requires my full attention and dedication to make sure that I deliver the perfect slides. And with the Tissue Tech Auto section, I started getting the job done easier, faster, and with better quality. The automated block alignment makes sure precious tissue is preserved optimally. It is also a great help for making recuts, while the perfect alignment helps me produce even more slides. Trimming and sectioning are done with high precision, each section the same exact thickness. Thanks to the easy operation of the auto section, every technician in our laboratory is quickly up and running. Whatever the need, we are all confident to deliver the perfect slide each time. My name is Mark Shanti, and I'm empowered by Sakura.
Halo, kali ini saya mau memperkenalkan alat laboratorium dari Xinjin. Ingenious Tree. Sebelum pengenalan alat, apakah Anda pernah mengalami kesulitan saat mendokumentasikan hasil gel electrophoresis atau plot Anda di ruang gelap? Sistem dokumentasi gel secara manual terdiri dari ruang gelap, kamera, dan transluminator sederhana. Dokumentasi gel secara manual seperti ini kadang menyulitkan, dan belum lagi kalau hasil foto gel yang didapat ternyata blur. Dengan Ingenious Tree, Anda dapat memilih pencahayaan sinar UV, sinar biru, atau putih, sesuai dengan pewarna yang Anda gunakan. Selain itu, software Genesis akan membantu Anda memilih pengaturan cahaya dan filter yang tepat, sehingga sangat memudahkan untuk mendokumentasikan gel fluorescence ethidium bromida atau cybersafe DNA atau protein gel dengan composite blue atau silver staining. Untuk penggunaan mode otomatis, Anda hanya perlu memilih tipe gel yang sesuai, kemudian software akan secara otomatis mengatur aperture, zoom, dan fokus serta posisi gel yang sesuai sehingga gambar gel terekam secara optimum. Software Genesis dilengkapi dengan database tipe gel dan pewarna yang tersedia di pasaran. Jadi jangan khawatir tidak bisa menemukan pengaturan yang sesuai. Sangat mudah. Hubungi tim ITS Science Indonesia untuk info lebih lanjut. Terima kasih. Stay safe.
Martin Chris, freeze drying system untuk penelitian, pilot project, hingga manufacturing produk Anda. Hubungi PT ITS Science Indonesia untuk informasi lebih lanjut.
Okay, welcome back in the last session. So, Dr. Siti Nurrohmah, are you ready to guide in this session?
Okay, I'm ready.
Yes, okay, time is yours now.
Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Welcome to the panel session in ISIBIO 2021. We have to come to the second days of the conference. I would like to say many thanks for join us. Let me introduce myself. My name is Siti Nurrohmah as a moderator of this session. Okay, today this last para session, we have 5 presenters, and I will give time for showing the pre-recording each presenter during 10 minutes. If you have a question, you can please write in a chat, or you can raising your hand in a Q&A session. During presentation, please activate the videos and activated audios. So we move to the first presenter. The first presenter is Mr. Saiful Anwar, with a title Status of F94L mutation of the myostatin gene among cattle breed in Indonesia. For Mrs. Saiful Anwar, time is yours.
Okay, thank you for the time and opportunity. Assalamualaikum warahmatullahi wabarakatuh. My name is Saiful Anwar. I work at Research Center for Biotechnology, National Research and Innovation Agency. Today, I will be presenting the result of our study entitled Status of the F94L mutation of the myostatin gene in several cattle breeds in Indonesia. Myostatin, also known as growth and differentiation factor H, is a growth factor belonging to the transforming growth factor beta superfamily that play a key role in regulating the skeletal muscle growth. Myostatin was discovered as a causal mutation for double muscle phenotype, such in Belgian blue and Piedmontese cattle breeds. You can see in figure 1 an example Belgian blue child looks to have an extreme muscularity, especially in the fore and hindquarter extremity. These cattle have some advantages, such as the higher proportion of muscle, less bone, less fat, less saturated fatty acid, more tender meat, and a higher proportion of the most desirable cuts of beef compared with normal cattle. However, these cattle appear to have some disadvantages, such as reduced female fertility, susceptibility to respiratory illness, especially high frequency of dystocia than normal cattle. Non-mutation have been detected in coding region of myostatin gene associated with double muscle phenotype. 6 mutations are considered as disruptive mutation that inhibit the function of myostatin protein, including in exon 2 and exon 3. Besides that, 3 non-disruptive mutation have also detected. One of them is F94L mutation that very popular among limousine breeders. This mutation is located in exon 1, characterized by substitution of cytosine to adenine at nucleotide position of 282, which led to causing substitution of leucine for phenylalanine at the 94 amino acid in myostatin gene. Interestingly, this mutation has been found in high frequency in limousine breed and was not considered to cause a loss of function of myostatin gene. Many previous studies have proven this mutation significantly associated with increased muscle mass, carcass and meat yield and quality both in pure and crossbreed limousine cattle without any associated reproductive problems. So the F94L mutation is a promising candidate marker for meat yield and quality in beef cattle. The objective of this study is, in our previous study, the wild type C. allele was found to be fixed in Bali, Sumbawa, and Frisian Holstein cattle in Indonesia. But the mutant A allele was found in Pasundan, although in very low frequency, 1.2%. But we presume that it segregated from limousine bull through artificial insemination. Therefore, the objective of this study is to confirm the status of the F94L segregation in several cattle breeds in Indonesia. For methods, in this study we used a total of 388 animals, including Limousin Simmental, Holstein Friesian, Dali and Peranakan Ongol bulls, and PO cows to edit for PO samples. All bulls were from Lembang and Singosari, National Artificial Insemination Center of Indonesia, whereas PO cows were from smallholder farmer in Rembang Regency, Central Java Province. Frozen semen was collected from all bulls, whereas blood samples were collected from all PO cows. Genomic DNA was selected from the frozen semen and bull blood samples using a G-Sync DNA extraction kit. For PCR and genotyping were conducted according to our previous study. The PCR product was 630 base pairs, which amplified with an annealing temperature of 53 degrees Celsius. TAC1 restriction enzyme was used for genotyping, and the genotype data were then analyzed for genotype and allele frequency and Hardy-Weinberg equilibrium. Result and discussion. In the present study, all three genotypes were found. For AA genotype was characterized by one band, 630 decibel. It's similar with PCR product. For CA genotype was characterized by three bands, 630, 414, and 216 decibels. For CC genotype was characterized by two bands, 414 and 216 decibels. Genotype and allele frequencies of F94L in several cattle breeds in Indonesia are presented in Table 1. It can be seen that all three genotypes were found in Limousin bull with frequencies of 0.9, 9.3, and 89.7% for CC, CA, and AA genotype. The frequency of wild-type C allele was 6%, while mutant A allele was 94%. The genotype were in Hardy-Weinberg equilibrium. Interestingly, all samples other than Limousin breed have been found on the CC genotype. Thus, the C allele was fixed. The fixed C allele in Simmental, Holstein, and Bali cattle were still consistent with previous studies. The occurrence of the fixed C allele was also observed in both Indicus cattle, as represented by Peranakan Ongol in our study, Sumbawa, Brahman, and Nelore cattle breeds. So we presume that the presence of mutant A allele in cattle breeds in Indonesia most likely originated from Limousin breeds. To support these presumptions, in Table 2, a total of 58 representing 10 purebred cattle collected from Nights were also genotyped, and the results showed that all bulls were CC genotype. Although in inadequate samples, the results strengthen our presumption. In the present study, also the predominance of mutant A allele in Limousin bulls in Nights of Indonesia was in accordance in US and Australian Limousin cattle. Let's see in Figure 4. It can be seen that the frequency of the mutant A allele was 73 to 82% in US Limousin cattle and 95% in Australian Limousin cattle. The mutant A allele was also present in Limousin crossbreed cattle with various proportions, from 25 to 76%. The high frequency of mutant A allele in Australian Limousin might have contributed to the high frequency of the mutant A allele in Limousin bulls in two Nights of Indonesia, which are generally imported from Australia. In conclusion, The mutant A allele of F94L mutation in cattle breeds in Indonesia most likely originated from limousine breeds. The origin of the mutant A allele in Pasundan cattle may be derived from the limousine bulls of Knight's through artificial insemination. Limousine bulls are the only bull breeds as a source of mutant A allele of F94L mutation in Knight's of Indonesia, so it could be used as an alternative to produce Indonesian commercial beef cattle with high carcass and meat yield. However, this needs to be investigated further. In the last, we would like to thank my institution for funding this study through DIPA IPH for fiscal year 2021. We also thank Mr. Agus Wibowo, Mr. Agus Santoso, and Mr. Anwalu Fuad for blood sampling of the cows assistants, Lia and Rido for laboratory assistant, and all farmers in Rembang Regency for providing samples of their PO cows. That's our presentation. If any question, we are pleased to answer. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Mr. Saiful Anwar. It's nice, very presentation. Okay, we move on the next presenter. The next presenter is Mrs. Ecology Fitriana, with title "The Determination of Residual Fiber Composition from Agricultural Bioproduct After Being Treated with Solid-State Fermentation and Black Soldier Fly Larvae Rearing." So for Mrs. Eko, time is yours.
Hello everyone, let me introduce myself. My name is Ecology Fitriana. I am a student in Feed Nutrition and Science IPB University. Today, I would like to present about our research with title "The Determination of Residual Fiber Composition from Agricultural Byproduct After Being Treated with the Solid-State Fermentation and Black Soldier Fly Larvae Rearing." Today, the human population increasing its influence the agricultural exploitation for food demand, which had led to a significant impact on the environment. It was because the exploitation created several byproducts. The several agricultural byproducts such as cacao pod husk or CPH and oil palm fern or OPF are underutilized. Before, they are used as forage for livestock with the treatment but did not optimal. The accumulation of them may cause some problem. Examples: the accumulation CPH in the field can cause black pod disease and the tree and leaf cacao. Another problem is decreasing quantity and also quality of oil palm because no pruning of the oil palm frond. So therefore, this research have been focusing on enhance the value because of the high quantity of byproduct. Solid-state fermentation is one strategy for improving the nutritional value of agricultural byproduct, which have high fiber content. This technology uses low energy, affordable cost, and it's environmentally friendly. Meanwhile, another treatment is BSFL, or Black Soldier Fly Larva. The BSFL is an insect well very known for its ability in bioconversion and bioremediation of organic byproduct. They are an alternative for the management and evaluation of agricultural byproduct because of its ability to convert low to high value organic byproduct. It offers excellent adaptability to various organic substrates that unbalance nutrients such as coffee industry byproduct, vegetable waste, food waste, feces, and etc. So in this study, the combination of SSF or solid-state fermentation and Black Soldier Fly Larva or BSFL technology was evaluated, and it was hypothesized that the combination treatment can be changed in the nutrition cacao pod husk and oil palm fern with high lignocellulose content. The cacao pod husk and oil palm fern were collected from Kebun Penelitian Cibodas and Cikaba Field IPB. Then the three fungal strain, Paneruccia Cryptosporium, Pleurotus hajar-saju, and Transmetas versicolor were used to solid-state fermentation or SSF. The step 1 is substrate preparation. The cacao pot husk and oil pumper were chopped and sun-dried for 7 days. Then they are ground using a grinder, then sterilized using autoclave to inactivate a microbe within the byproduct. The step 2 is substrate fermentation. The starter for fungi was consist of fresh bran and molasses, and a single strain was added to the starter and adjusted to 70% moisture using sterile soaked rice water. The starter was incubated in a plastic bag with air circulation at room temperature for 7 days. Around 60 grams of the byproduct milk were taken in the plastic bag and adjusted to 70% moisture using sterile aqueducts. Then the single strain starter fungi was added and it was thoroughly mixed. It was incubated at room temperature for 21 days. The sample for fiber analysis was collected on final day. In addition, the substrate or media was prepared for substrate or BASF-L rearing. The step 3 is a BASF-L rearing. The method of rearing BASF-L was modified from Chai et al. 2017. About 11.8 grams of 7-old BASF-L were reared in 60 gram substrate solid-state fermentation. The substrate was kept wet, but not free water was in the box bottom. The rearing was stopped when the approximately 50% of larvae grew to become a prepup, and the leftover substrate or media was become a fiber analyzed. The fiber analyzed was determined neutral detergent fiber or NDF, acid detergent fiber or ADF, cellulose, hemicellulose, and the lignin. According to the official method, the official Francis method, the principle of the method is extract the target fiber fraction with the detergent solution. So, the figure of the screen show that the increasing percentage of NDF and IDF was due to the solid-state fermentation treatment. This was due to the decrease in the content of cell, such starch, protein, and lipid for a fungal development. They are such as nitrogen and carbon source. The hemicellulose content has decreased, but the cellulose content has increased. The decrease of hemicellulose in all experimental solid-state fermentation media reflect the fact that the fungi can produce hemicellulose degrading enzyme. Previous study was confirmed that the several WRF, such as Phanerochaeta chrysosporium and Transmetas versicolor, also produce hemicellulose enzyme like silanase. The cellulose content increased due to the free cellulose protected by lignin and hemicellulose. So, the solid-state fermentation treatment in CPH on pot cacao where 3 WRF RF can degrade the lignin content by 5.08% until 6.26%, while OPF, or oil palm fern, decrease around 1.87% until 2.35%. WRF can secrete cellulolytic and lignocellulosic enzyme, including lignin peroxidase, manganese peroxidase, and lacase. The most fermentation with the Phanerochaeta chrysosporium has the lowest lignin in the CPH, and the Transmetas versicolor has the lowest lignin in OPF. Each fungi is the lengthy time required for the fermentation. However, the characteristic component of media or substrate has influenced fermentation. BASF-L has two major digestion processes. The one is smooth part, and another is intestinal enzyme. A previous study showed that the substrate or media significantly influence the performance and the nutrient content, and also the microbe community in the gut of the BASF-L. Furthermore, the stable core group of bacteria can be present to guarantee dietary flexibility. They are Actinomycetes, Disgonomonas, and Enterococcus. They are present in at least 80% of the total bacteria. So one of them, Dysgonomonas, has mostly known for its role to degradation of lignocellulose. It was explained that the BASFL can reduce lignin in the CPH substrate, but adverse result in OPF may be suspect because of the effect of reactivation fungi in the substrate. Even BASFL can tolerate fungal development, but they can evade the nutrient substrate. Meanwhile, increasing lignin percentage in OPF was implicated by increasing NDF and ADF content. The combination of our fermented OPF and BASFL rearing decrease only hemicellulose content. Therefore, the BASFL can degrade hemicellulose and cellulose but cannot optimally absorb them to be increasing of biomass. The combination of CPH, Phanerochaetic Discosporium, and BASFL rearing use almost all fiber fraction except hemicellulose, while the CPH Strammates versicolor use all fiber fraction except cellulose. So, in this research, we can conclude the first, fungal fermentation enhance the efficiency of nutrient utilization for BASFL. And the second, the combination of solid-state fermentation and black solder Biorefinery ring technology increase the nutrient fiber and determine the lignin removal on CPH but not optimal on OPF. And the third, the main factor that need to be considered in the optimum time for the fungi fermentation and the time for BASFL rearing. So thank you. I would like to thank you to our beloved university, IPB University, and this work supported by Ristek Dikti within the PMDSU scholarship.
Thank you very much, Ms. Eko. This nice presentation. If you have a question, please collect in Q&A section. So we move in the third presenter. The third presenter is Mr. Rusli Fidrianto. Mr. Rusli Fidrianto will presentation with title Utilization of Fruit Byproduct as a Ruminant Feed in Vitro Digestibility and Methane Production. For Mr. Rusli Fidrianto, time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. First of all, I would like to introduce myself. My name is Rusli Fidrianto from Research Center for Biotechnology, National Research and Innovation Agency, or BRIN. In today's presentation, I would like to present our study about the utilization of fruit byproduct as ruminant feed, in vitro digestibility, and methane production. In recent years, fruit agriculture has been increasing continuously as a result of a growing demand. Along with the development of the fruit agriculture, agricultural waste such as wheat plant, peels, and seed are generated in large quantities. This agricultural waste become a tough environmental issue. In the case of fruit byproduct, usually the high amount of phenolic compound is found in peels, kernel, or seed. This byproduct have a potential use as a source of valuable antioxidant. One profitable strategy in treating waste is to recover the bioactive compound that can be utilized for food, pharmaceutical, or cosmetic industry. Many industry has been using fruit byproduct as raw material for extract product. However, this industry will generated a large quantity of biomass waste. There is no report yet on a valorization of this byproduct for ruminant feed. Therefore, the aim of this study was to evaluate the potential use of byproduct after the phenolic compound was extracted as ruminant feed by in vitro rumen fermentation analysis. In this study, 4 fruit waste was used. Fruit waste consists of the pomegranate peel, kaffir lime peel, mangosteen peel were obtained from a local supplier, and then the molastoma candidum fruit was obtained from PT Perkebunan Nusantara 8. All samples were collected in polyethylene bag. All samples were extracted the phenolic compound according to Makar 2003. Samples were dried at a room temperature for 72 hours, grinded, extracted with acetone 70% with ratio 150 using ultrasonic water bath for 1 hour. Filtered through the Wattmann filter paper, and then the solid paste was dried in a cabinet dryer at 42 degrees Celsius for 24 hours. All of the samples were analyzed using proximate analysis, consists of the dry matter, organic matter, crude protein, ether extract, crude fiber, NDF, ADF, hemicellulose, and NFE. All samples were also analyzed using FTIR and in vitro rumen fermentation. From in vitro rumen fermentation analysis, we get kinetic of gas production, methane gas production, pH, in vitro dry matter digestibility, in vitro organic matter digestibility, and partial volatile fatty acid using JCMS. The result of this study show that the Melastoma candidum fruit had the highest crude protein, followed with the kaffir lime peel, pomegranate peel, and mangosteen peel. In this study, the mangosteen peel had the highest crude fiber and the lowest ether extract. In this study, the NDF and ADF were showed the same pattern. The highest NDF and ADF were observed in mangosteen, followed with the Melastoma candidum fruit, pomegranate peel, and kaffir lime peel. There was no significant difference in hemicellulose content. This is the result from FTIR analysis. The result of this study showed that the absorption peak between 3,650 to 3,200 mainly reflects the OH stretching vibration, and then peak in range 3,000 to 2,800 correspond to the CH stretching bond of the methyl group. The peak around 1,317, 1,370, and 1,441 in all samples mainly provide the information of the CH bending vibration. The peak at 1,518 in mangosteen samples show the presence of more lignin in a fiber, and a peak around 1,032 centimeter correspond to this aromatic CH deformation. It means the presence of a lignin. The result from FTIR was also used to calculate the crystallinity of a sample, consists of the hydrogen bond index and total crystallinity index. The result of this study showed that mangosteen had the highest HBI value by 1.13, followed with the kaffir lime peel, Melastoma candidum fruit, and pomegranate peel. The result of the TCI analysis showed that the pomegranate peel and mangosteen peel had the highest TCI value than kaffir lime peel and Melastoma candidum fruit. The HBI was provide the information about the hydrogen bonding between the certain hydroxyl group in cellulose. Generally, crystallinity decreased with the increasing of the HBI value. The low HBI value and high TCI value in pomegranate peel were indicate that a pomegranate peel contain a high crystallinity. This is gas production, kinetic, and digestibility of food byproduct that we get from in vitro rumen fermentation. The result of this study show that the kaffir lime peel had the highest potential gas production rate, dry matter digestibility, organic matter digestibility, follow with the pomegranate peel. Moreover, the kaffir lime peel was observed had the lowest pH as a result of the highest digestibility and gas production. The higher potential gas production and digestibility of kaffir lime peel and pomegranate peel in our study may be due to the high protein content and NFV. Nitrogen V extract represent the soluble carbohydrate in the feed that easy to digest in a rumen. The lowest potential gas production and digestibility were observed in a mangosteen by 18.84 ml, 12.32% and 7.28% respectively. Even though the Melastoma candidum fruit had the highest protein content and the potential gas production and the digestibility were significantly lower than kaffir lime peel and pomegranate peel. The high NDF, ADF, and the crystallinity in MCF were also affected in the low digestibility. In the case of a methane production, large difference was observed among all samples. The mangosteen and Melastoma candidum fruit saw the similar result with a no methane production at 12 hours, 48 hours, and 24 hours. The methane production of kaffir lime peel at 24 hours, and 48 hours were the highest among the others. The highest methane production in kaffir lime peel in this study may due to the high amount of acetate and butyrate production that saw in the table 5 from the fiber digestion. This is the result of the volatile fatty acid from samples. The result of this study showed that the highest short-chain fatty acid was observed in a kaffir lime peel, followed with pomegranate peel, Melastoma candidum fruit, and mangosteen peel. Moreover, the only acetic acid and propionic acid were detected in mangosteen. In this study, the similar pattern was observed between gas production kinetic, digestibility, and short-chain fatty acid. The previous study reported that the concentration of SCFA short-chain fatty acid had a positive correlation with organic matter digestibility. Furthermore, high A per P was observed in kaffir lime peel and Melastoma candidum fruit as a result of a high portion of acetic acid and low propionic acid. The total short-chain fatty acid had a positive correlation with the total gas production if the fermentation of the feed substrates led to high portion of acetate. And the conclusion are in this study found that even though the phenolic compound had been extracted from the fruit byproduct, it still can be used as a ruminant feed. The kaffir lime peel and pomegranate peel had a good potential use as ruminant feed due to the high protein content, digestibility, gas production, and short-chain fatty acid compared with the Melastoma candidum fruit and the mangosteen peel. And this research was supported by the Program Research National or PRN 2020 with a contact number 44A1 PRN 2020 and DIPA IPH LIPI 2021 of Research Center for Biotechnology, Indonesian Institute of Sciences. Thank you.
Okay, thank you, Mr. Rusli. Very nice presentation. Before go to the fourth presenter, I remind you to the field attendance register have already circulated in the chat, and so you can go to the link and make sure to register yourself today's event. So we move to the fourth presenter. The fourth presenter is Miss Sinta Maharani. Miss Sinta Maharani will a presentation with title Substitution Effect of Chili Leaf powder for palm kernel meal in a ruminant feed on the in vitro rumen fermentation characteristic. So for Ms. Sinta Marahani, the time for yours.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Let me introduce myself. My name is Sinta Maharani, and I am a junior researcher in the Research Center for Biology, National Research and Innovation Agency. In this occasion, I would like to present our research with the title Substitution Effect of Chili Leaf Powder for Copra Meal in Ruminant Feed on In Vitro Rumen Fermentation Characteristic. As we know that the human population in the world is constantly growing and there is a necessity to increase food production from plants or animals. Copra meal is one of the alternative byproduct produced mainly in some tropical area of the world. Copra meal is a byproduct of the coconut industry that usually used as a cheap source of nutrients compared to those normally used to supply part of the requirement of animal diets. One valuable possibility in the field of animal nutrition is recycling this material as alternative feedstuff, particularly for herbivore. Capsicum pepper are among the oldest cultivated plants in the world. After harvesting, the chili leaves are left as main residue. These wastes are known as a byproduct and their disposal may represent an environmental problem. Natural substance extracted from plants can be used as a safe dietary additive to modify rumen fermentation and microbe in terms of pollution mitigation, for example, methane and ammonia, and improve feed efficiency. There have been a limited number of studies on the effect of diet substitution with chili leaf powder for copra meal for ruminants. Thus, this present study aims as a preliminary study to assess and evaluate the substitution effect of chili leaf powder for copra meal in ruminant feed on in vitro rumen fermentation. And material and methods: chili leaf powder preparation. Chili leaf were obtained from local farm in Bogor, West Java, and chili leaf were washed 4 times with distilled water to remove the impurities and dried in a cabinet dryer at 40 degrees Celsius for 24 hours. Before analysis, the dried sample was milled into 18 mesh flour and stored in polyethylene bags at 4 degrees Celsius prior to analysis. All concentrate feed ingredients were obtained from local supplier in Bogor, West Java. Feed ingredients were formulated according to the formulation shown in Table 1. Samples were analyzed for proximate analysis according to AOAC method. The experiment was arranged in a randomized complete block design with 5 different chili leaf powder concentration. This is DC0, DC25, DC50, DC75, and DC100% as copra meal substitution in concentrate feed as a treatment. In vitro rumen fermentation was measured by method of Theodoro et al. With modification, and in vitro dry matter digestibility and in vitro organic matter digestibility were determined according to Ley et al. The sample of partial volatile fatty acid, gas chromatography, mass spectrophotometry or GCMS analysis were performed using GCMS Shimatsu and MegaWax MS column. Data were analyzed by one-way analysis for variant using SPSS24. Significant effect of treatment were determined using Duncan multiple rings test method. And then result and discussion. For the first time, a chemical component analysis. The chemical composition of chili leaf powder, copra meal, and concentrate shown in Table 2 and Table 3. We can show in Table 2 that the chemical composition of chili leaf powder and copra meal were significantly different in dry matter, organic matter, extract ether, crude fiber, and nitrogen-free extract. However, there was no different crude protein composition between chili leaf powder and copra meal. In this study, the dry matter content of chili leaf powder is higher than copra meal. However, the organic matter, extract ether, crude fiber, and nitrogen-free extract content of copra meal is higher than chili leaf powder. Jayanegara et al. State that the extract ether content observed in copra meal were high, indicating inefficient oil extraction from the original materials. The chemical composition of concentrate was reported in Table 3. Except nitrogen-free extract content, all chemical parameters showed significantly different. Dry matter, organic matter, crude fiber, and nitrogen-free extract composition of DC0 was higher than other variable. Crude fiber composition of DC100 was significantly higher than other by 14.77%. Gas production, pH analysis, and digestibility was shown in Table 4. In vitro gas production kinetic, methane, pH, and digestibility of effect of chili leaf powder substitution, we can show the result that potential gas production and gas production rate were not significantly affected by GLP substitution. In this study, the range of pH, all treatment were 6.75 to 6.85. This research showed that the substitution of chili leaf powder for copra meal did not affect the pH rumen, and in vitro dry matter digestibility and in vitro organic matter digestibility value were significantly increased after GLP treatments. The increase of dry matter digestibility and organic matter digestibility were proposed due to the decrease of crude fiber and increase of crude protein. Digestibility had positive correlation with crude protein content. Substitution of chili leaf powder for copra meal was expected to be a beneficial rumen function based on its stimulating effect on fermentation by increasing degradability of crude protein and plant cell wall constituent. Phenol concentration in ruminal fluid was also significantly increased related to the increasing of GLP concentration, with DC100 as the highest phenol by 2.94 millimole per deciliter. The increasing of phenol content in rumen fluid was not followed by the decreasing of methane production. It may be due to the phenolic content in the concentrate substitution of copra meal with chili leaf powder was not enough to decrease methane. The ratio of acetic acid to propionic acid showed a significant effect as shown in Table 5. The lowest A per P value was produced by DC100, and treatment of DC25, 50, and 75 showed the highest A per P ratio value. Furthermore, the SCFA value was not significantly affected by all treatment. There was a research confirmed that less nutrient digestibility causing less volatile fatty acid production and less gas production. In conclusion, the result of the current study indicated the increasing chili leaf powder concentration was followed by increasing crude fiber and protein. However, ether extract and nitrogen-free extract content were decreased. In addition, the chili leaf powder can substitute copra meal up to 100% without decreasing SGFA production. However, more detailed studies are needed to evaluate the addition of chili leaf powder in ruminant diet. So I would like to say thank you, and this research was supported by Prioritas Riset Nasional 2020 and DIPA IPH LIPI 2021 of Research Center for Biotechnology, Indonesian Institute of Sciences. Thank you.
Okay, thank you, Ms. Sinta Maharani. Very nice presentation. So we move to the last presenter. The last presenter is Mr. Muhammad Zaki Alifian with title Comparison of Selenium Levels of Source and Laying Hands Performance and Eggs Quality, a meta-analysis. So for Mr. Muhammad Zaki Alifian, the time is yours.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Zaki Alifian. I want to tell you about my research, comparison of selenium level and source on laying hands, performance, and eggs quality of meta-analysis. Introduction. The current trend of the poultry industry by meat laying hens diet with selenium that is suitable for increasing the concentration of selenium in meat and eggs. Conventionally, sodium selenate is the most frequently used source of selenium in laying hens feed. Organic forms such as selenium fortified yeast and selenium methionine have been suggested to be used as selenium additive to maintain healthy laying hens and increase selenium concentration in meat and eggs in recent years. Organic selenium has antioxidant properties and high bioavailability and lower rate of product accumulation and toxicity compared to the inorganic form. This increase efficiency the use of selenium. In this study, aims to evaluate the influence of dietary level selenium organic and inorganic on the performance and eggs quality of laying hen by employing a meta-analysis method. Development of database. A database was built from published studies which have report on the addition of various levels of selenium laying hens diet. The criteria of articles to be included in the database were that they were published in English, treatment include the addition of selenium source to certain basal feeds, and experiments were conducted in vivo on laying hen. A total of 28 articles were initially found after searching using the above mentioned keyword. This is table of a database. Analysis of data. The database was statistically analyzed using mixed model methodology, in which the model has been widely used in meta-analysis research related to animal nutrition. Different studies were considered as a random effect, and the dose of flock were treated as fixed effect. The table regression equation on the influence of selenium in addition in milligram per kilogram of diet as feed on performance of laying hen. The addition of selenium dose didn't significantly affect the laying hen rate. The addition of selenium increased egg weight. Egg mass was not affected with the addition of selenium. Feed intake was not affected by increase in selenium dose. Feed conversion ratio was influenced by selenium dose with linear model. Selenium dose did not affect the mortality of laying hen. The addition of selenium had no effect on egg quality, but the thickness has to decrease of selenium, but the thickness of the cell decreased the thickness at high dose. Perform of different selenium on laying hen. Different addition of selenium affect the performance egg weight and mortality of laying hen. This eggs quality of different selenium on laying hen. Albumin has and tendency to increase with different selenium addition. Selenium X has a tendency to increase with the addition of different types of selenium. The quality of the X had no effect with the different in type selenium source. The absorption pathway may be a possible explanation for the accumulation of product. Selenium when laying hens are fed diet of different selenium concentration. Organic selenium in selenium yes is dominated by selenium methionine, which can be incorporated in eggs as effectively as a methionine. Organic selenium source such as selenium methionine or selenium US were actively absorbed and can be directly incorporated into protein, whereas inorganic selenium source such as sodium selenate are passively absorbed by the body. Source of selenium inorganic such as selenium selenate are required for selenocysteine synthesis. The conclusion, the meta-analysis has discovered that increasing selenium level laying hens diet leads to an improvement egg weight and decreasing feed conversion ratio, but in eggs quality high level of selenium decreasing egg shell thickness. Organic and inorganic selenium increasing egg weight and decreasing mortality on laying hen. Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Muhammad Zaki Alfian for nice presentation. So we move to the Q&A session. I have already checked in the chat this question for Mrs. Sinta Maharani from Prof. Endang. How much you can supply chili leaves to the feed? What the function of chili leaf for the rumen of cattle? For Mrs. Sinta Maharani, please answer.
Okay, thank you, Ms. Siti Nurrohma, and thank you to Prof. Endang Trimarkawati for the question. I will try to answer the question. Our research is the big topic of our research is actually nutrition characteristic nutrition and bioactive compound in this chili leaf. But when we try to collect the sample, there are so many farmer here just throw the chili leaf when they have been harvested, and we try to identified the nutrition characteristic of chili leaf, and here we found that in this varietas the protein content of chili leaf is 18%, and 18% to until 26%. And in this in vitro research, we use the lower protein content, that is 18%. And we will for the next research we will try to identify the other variety to check maybe the other variety can show the effect of mitigation of methane. So, and for the rumen function, the function of chili leaf for the rumen for cattle, we hope that our research chili leaf powder here can be a protein sources for the cattle ransom, cattle ratio. That is my answer. How about Prof. Endang?
Thank you, Sinta. Sukses
Thank you,
ya.
Prof.
Dr. Siti, I think, yeah.
Okay, thank you. Thank you. Okay, we will wrap up the last session of oral presentation in this oral presentation. So I return to the host, and I would like to give a very time for join this oral presentation and say goodbye. Thank you.
Thank you. Thank you very much. Yeah, thank you very much for all the moderators, and also Bu Lilis as the invited speakers, and also for the all participants for the active participation in this last session of ISIBIO 2021. And then before we close our session, please kindly return to the main room for closing ceremony. And thank you very much.
Recording in progress.
National Research and Innovation Agency, a synergy in an orchestra.
Ladies and gentlemen, welcome back
Established through
to
the
the main room
Presidential
of ISIBIO
Regulation number
2021. We would like to thank all the speakers and moderators for very insightful session. Okay, so for the next, we will announce the best presenter of ISIBIO 2021. And the best presenter are chosen based on the highest score of the presenter in each room during day 1 and day 2 of parallel sessions. Now we would like to announce for the first day, the best presenters of the first day of ISIBIO 2021 are Room 1, Nikadek Erosi Undaharta. I'm sorry for the wrong spelling. And room 2, congratulations. Room 2 is Mr. Aryo Beta Juan Silvero. Congratulations. And room 3, the winner is Fitria Dwi Ayuning Bias. Congratulations for the winners of the best presenter of the first day of ISIBIO. Now, move on to the second day. The best presenter of the second day of ISIBIO 2021 are Room 1, Mrs. Wayuni. Congratulations. Room 2, La Ode MM Davis. Congratulations. And last but not least, room 3, Andri Wardiana. Congratulations once again. Let us give a round of applause to the winners, and you can use the reaction feature in the Zoom to congratulate the winners. Once again, we'll say congratulations to the best presenter for delivering such remarkable presentation in ISIBIO 2021. Ladies and gentlemen, we will have closing remarks by Dr. Mastur, Chair of Indonesian Biotechnology Consortium. Please welcome Dr. Mastur.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. Distinguished invited participants, organizers, ladies and gentlemen. First, let us express our praise and gratitude to Allah the Almighty for His grace and guidance that today we can successfully finish the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering. On behalf of everyone involved in this conference, I will also express our gratitude to the Chairman of National Research and Innovation Agency, Dr. Laksanatri Handoko, for his support, direction, and opening remark. His support motivates and boosts our energy to enhance research and application of biotechnology, to support national development in all regard sector and field. Biotechnology at the front line of science and technology is required to increase productivity, quality, efficiency on biodiversity, sustainable environment management and use, medical and industrial sectors, product diversification, and new innovation. Biotechnology is also expected to overcome the climate change through mitigation and adaptation to bring great benefit to human life and environment. During these 2 days, we had a fruitful discussion and sharing information. Thank you so much for all resource persons for your excellent ideas and sharing information. For all participants, thank you so much for your support in scientific papers. We highly appreciate all sponsor and support for this international seminar, especially for BRINS and Biotechnology Enterprises Partners. KBI management and all members believe everyone gain a huge benefit from this program. Therefore, KBI management will commit to promote biotechnology through the similar program, especially during period 2021-2024. We will improve collaboration with other stakeholders, universities, research institute and bureaucrat, and industry and other biotechnology practitioners. Synergy to support research, education, and industrial practices in Indonesia. At this opportunity, we will also invite all elements, universities, research institute, government institution, industry, and business related to biotechnology to join KBI. KBI management commits to bring real benefits to all members. KBI will facilitate research, engineer, university lecturers, and students to improve their research and publication. For bureaucrat, KBI will participate in promoting regulation. For industry and business sector, you will get opportunity to advertise your product. Finally, We hope collaboration between KBI members and all stakeholders will be strengthened and intensified. Lastly, thank you Research Center for Biotechnology BRIN for great collaboration. For all participants, on behalf of the conference organizer, please accept our sincere apologies for inadequate services or any shortage during the conference. We hope this conference will bring advantage for everyone who join for our organizations and national development. Praise to God. Alhamdulillah, I close this conference. Thank you and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Masur, for officially closing the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering. We would like to give gratitude to Chairman of Research Organization for Life Sciences and Chair of Indonesian Biotechnology Consortium, all the keynote speakers, invited speakers, moderators, and presenters of the International Symposium. And not to forget the participants, thank you for the participation in ISIBIO 2021. Don't forget to fill out the list of attendees, and the certificate will be sent by the committee through email maximum 15 working days. Tomorrow, we will also hold the 7th Indonesian Biotechnology Conference, or IBC 2021. There would be talk shows with many great speakers, so we hope you can join the event tomorrow. And on the behalf of the official committee, I would like to apologize for any inconvenience matters during the event. So see you on ISIBIO next year. Have a good day. Goodbye.
Oke, thank you Mbak Wiwit. Thank you semuanya.
Thank you very much.
Thank you.
Thank you for all. See you in next year.
Thank you semua panitia, seluruh komite. Live ya.
Iya, Bu.
Asep, Bayu, makasih ya.
Makasih.
Sama-sama,
Selamat
Ibu.
panitia.
Panitia
Siap,
semua.
Bu.
Ayu, terima kasih banyak. Ayo, sukses semuanya. Terima kasih.
Sukses Perfeni. Terima kasih.
Yen.
Sama. Hey, Mbak Endang, apa kabar?
Baik.
Ayo
Ada
ketemuan.
Ayo dong.
Hah?
Besok IBC.
Oh iya, besok IBC ketemu.
Oke.